Good morning to our virtual audience at home. We hope you guys are up and ready to roll today. A big, long, exciting day. A lot of good information, some about medical research, some about just living life and making things better as we deal with the issues that we deal with. But a lot of good information, and we're glad that you're here with us. We wish you were physically here with us. We love to visit and sit and talk, but knowing that you can't be here, it's good that you can do this with us virtually. Don't know about you, but it was kind of a long night here, a good night. We finished last night, had a chance to visit with Collin from CPATH and the CPATH crew. We finished that about 9:30 and headed up to the room. Norma got there and our keys wouldn't get us in our room, so we spent half the night in the hallway. So we're recovering from a very good long day and good long night, and we hope you were with us most of the day yesterday. This morning's going to be a good day. I want to share some information with you. First and foremost, you can't do these kinds of things without wonderful sponsors and contributors. And in your program that I hope you're looking at online, on page 4 is a listing of all of our sponsors, and I just want to share those with you one by one quickly this morning. But Joshua Cochran and Toby and Becky, the Conlon Foundation, the Tool Shed Training, and you'll be hearing from them today and tomorrow, Regions Bank, Mary Kay Robinson, Sionic, I'll be talking about the Sionic sleeve tomorrow, A-Linker, Cadence Shoes, Your Medical Store, First Community Bank of the Heartland, that's our bank at home, who's been a sponsor about every year we've done this, and we appreciate that. Chick-fil-A, Deep Tissue Massage by Ray Presnell, who will be here doing some of that work today and tomorrow. World Bird Sanctuary, Missouri Botanical Garden, and the St. Louis Zoo. And we have an anonymous contributor that for the past few years, including this year, has said to us, we want to pay for the bags that you hand out there. And so we appreciate that very much. And I'm going to show you the bag and then tell you a little bit about the symbolism that's on it, because you may not have seen this at home. But of course, America is celebrating its 250th anniversary just around the corner. And regardless of where you stand on a lot of public issues, it's a birthday and we're celebrating America's 250th. But on the bag, you'll see a map. And you'll see pins on that map. And that map just symbolizes that we're on a pathway. We're going to the gateway. We're going to find cures and treatments down the road, and that's part of what this process is about. And you'll see those red pins on there. Nora says those red pins represent each one of us on this trail as we try to find cures and treatments that make a difference. And what part do you play at home? You can contribute financially in any way that you possibly can. We know you get a lot of emails about that, but without the resources that are raised, we could not do the research. We could not do the annual conventions. We could not do the SPF talks and the other things that you guys are involved with and are part of. So thank you for the contributions that you make. Thank you for your physical presence when you can be physically present on meetings that we have and those SPF talks. Your presence, your involvement, your contributions in any way make a great difference in what we're trying to do here with this foundation and moving toward treatments and cures. Again, we're about to get started in just a few minutes. We appreciate you guys at home with us. It's going to be a very tight schedule. If you've done this with us before virtually, you know that about every couple of hours we take a break so folks here can move around, get to a restroom and so on. But this agenda is so full with good speakers, there really aren't breaks in our agenda. So we'll be back to see you. Let me tell you that today it's not on the agenda that you have, but we do have a lunchtime speaker. So when time comes for lunch, you may want to grab your food tray and get in front of your TV so you can hear that speaker. He's going to be talking about clinical trials, particularly where PLS is concerned. So please join us then for that lunchtime speaker. Again, a lot going on here. If you need us, you know how to get in touch by text or email, and we'll certainly try to assist with anything in any way we can. I'm going to say goodbye for a moment, head to the front to get this thing kicked off this morning, and we'll talk to you sometime later today. Thanks for being with us. We appreciate you very much. Good morning. My watch says it's a minute or two after 8 o'clock, and I know we have a very aggressive agenda. If you've been with us at past meetings, you know that most mornings and afternoon there's a spot that says break. You may have noticed in this agenda that's not the case, and there's a reason for that. We've got more researchers, clinicians, speakers who wanted to participate and be part of this meeting, and we certainly wanted to fit all of them in. So I'll just tell you now, and Tim will remind you later, if you need to take a restroom break or a stretch break or any other kind of break any time of the day, feel free to do that on your own, and we will all understand because we'll all have to do that. But a very full agenda today and a lot of good information over the next 2 days. So good morning to you. I don't know what kind of night you had. When the meeting was over, I'll share with you my night and Norma's night. When we finished with the meeting, we went down to the restaurant, I don't know, about 7 o'clock, 7:30. Had dinner with Collin and the CPATH crew and continued to work a little bit about information we heard yesterday. Wrapped that up about 9:30, quarter to 10, and I'm thinking, okay, going to get back to the room, chance for a shower and a good night's rest. You would know this would happen. We get to our room and our keys won't unlock the door. So we go get back on the elevator, we go back down to the floor, and I sit there and wait for Norma to go to the front desk, get the keys reprogrammed. We're good now. To the room, get there, the reprogrammed keys will not open the door. So Norma gets on the phone. 30, 45 minutes later, we find out what the problem is. The batteries have died during the day. So we had to have batteries. So it was late when we got in, a late shower, but a good night's sleep. And I hope all of you guys had a restful night and did well. But to every patient, family member, caregiver, clinician, researcher, sponsor, volunteer, and friend joining us in person and virtually from around the world, we thank you for being here. The first thing I want to do, and I can't see them all, but I want to introduce to you your board of directors who are here, and then share with you that every other board of director, a member of the board, is participating with us virtually from home. So I can't see you, but Jim Sheehan, are you in here? Yeah, there's Jim. Jim is our vice president, and he's chair of our fundraising committee. You may get more emails from fundraising folks than you'd like to see, but again, it's because we need that to make all this work. So thank you, Jim. Frank Davis. Frank is our president emeritus. He served as president for 10 years, and he continues to work hard with us and on your behalf. Dina Landfair. Dina. Here's Dina. Dina is from Iowa and serves on the board. Tina Croghan. Okay, Tina's out in the hall. Becky Cochran. Becky here at center table. And, of course, Dr. Fink is our medical advisor, and I know he's busy this morning. But those are the members of the board of directors who are here. Those who are watching and participating virtually are our secretary, Hank Chupi, Jackie Wellman, John Cobb, Sarah Kramer, Karina Thurgood. And so we appreciate all the work that all of them do. And they all wanted to be here, but for different reasons weren't able to do that, but they're joining us virtually. Well, as you saw yesterday, there was energy in this room, a lot of great information, a sense of momentum, a sense that our community and our board and this work is entering a new chapter. With more than 200 people expected to participate and many more joining through our live stream and future recordings on the SPF YouTube channel, this has become one of the largest and most important gatherings in SPF history. And let me say to you about the YouTube channel. Many of you, I'm sure, have watched some of those programs. Every program from every past annual conference recently is on there. If you missed part of a session here or you'd like to re-listen and rehear some of what you've heard after we get past this meeting, just a few weeks down the line, it'll all be on the YouTube channel, and you'll have the chance to go back and listen again if you'd like to do that. So many years, people living with HSP and PLS have felt isolated, unheard, invisible, and many times forgotten. Many families have spent years. I've heard stories from some of you about spending years for a proper diagnosis. Many were told there were no treatments, no roadmap, and very few people who even understood these diseases. But look around this room today. You know, our registration information told us that 72 people who have registered to be here are here for their first annual conference. And getting to know one another is so critically important. You know, I've got friends here that I never would have known, would have never met had it not been for HSP. And, you know, in one sense of the word, I certainly wish I didn't have this to deal with. But on the other side of that coin, I've met so many wonderful people who care for one another. And these days we can do that by Zoom or by email or by cell phone. Norma shares a story, and I've had similar stories.
The first conference that she managed was in Atlanta in 2017. And she came around the corner, and there was a woman standing there alone with tears going down her cheek, and Norma thought something was wrong. She was hurt or had a major problem. Norma walked up to her and said, "Ma'am, are you okay? What's wrong? Can I help you?" And her response was, "I have dealt with this for so long, and this is the first time I've ever been with somebody else who looked like I look, who walked like I walk, who has these same problems and understands this disease." So get to know one another as friends. When you go back home, when we go back home, we can continue to support one another in so many different ways. But you're not alone. This conference represents a growing global movement of people determined to change the future of HSP and PLS. If you were here and listening yesterday, you heard speakers from all over the world that we collectively are trying to bring together along with them. We're not the organization that's leading the world charge, but we are a key part of it in trying to bring those world researchers, those world patient organizations together collectively to make a difference in this process. Over the next 2 days, we are bringing together an extraordinary delegation of clinicians, scientists, therapists, innovators, caregivers, advocates, and families, all united around one shared mission: to improve quality of life, accelerate therapies, and build a future filled with greater hope, health, wellness, and possibility. And while science and medicine are incredibly important, this conference is also about something much deeper. It's about humanity, because these diseases affect every part of life. They've taken away mobility from some of us, careers, independence, maybe financial security, dreams you may have had, time with family, the ability to travel freely, the simple things that I used to, and many people these days take for granted every day. But despite all of that hardship, this community continues to show remarkable courage. Patients continue the fight. Families continue caring. Researchers continue searching. And together we continue building something stronger than despair. We have hope with direction. This year's conference is more than words. It's a call forward for every one of us. The bag that you got, a pathway to the gateway, and that if you think about those words, we're trying to work with all of you and everyone involved to create a pathway to a gateway of treatment, therapies, and a better life. We're witnessing advances in genetics, biomarkers, rehabilitation science, assistive technologies, AI-driven discovery, wearable technologies, outcome measures, and therapeutic development that were almost unimaginable a few years ago. The conversations taking place this week are not theoretical anymore. They are becoming practical, translational, forward-moving. And what happens here matters far beyond this hotel ballroom and this conference. Because our sessions are being shared virtually across the world, families who would not travel are watching, researchers are watching, young clinicians are watching, and future collaborators are watching. The whole world is watching what we're collectively trying to build together. And what we build here must continue after this conference. We must continue connecting globally, supporting one another, sharing knowledge, creating partnerships, advocating for research, demanding urgency, and preparing for the therapies we believe are coming. We cannot afford to waste this moment and this opportunity. The window before us is real. The momentum is real. The opportunity to change lives is real. More than 250 years ago, a great nation was built around the belief that people working together could overcome impossible challenges because they shared more common interests than differences. That same spirit is alive in this room today with the things we're working with and dealing with. No matter where we come from, no matter our background, no matter our diagnosis, we are united by something powerful. That belief that tomorrow can be better than today. And I believe with all my heart that future generations will look back on gatherings like this that you are part of as moments where this community chose not to accept limits, but instead chose collaboration, innovation, compassion, and action. So on behalf of the foundation, thank you for being here. Thank you for being part of this mission. Thank you for being our friend. You know, it means so much to us. And there are many of us, and we won't get a chance to visit long with everyone. But it means so much to us because I want to introduce a couple of other people, and they may not be in here. But at our table, of course, all of you, almost everybody knows Norma, and I don't have to tell you, when I say something like this in front of you guys, you all laugh because you know it's true. For Norma and me, with the exception of the time we deal with family and deal with issues on our farm, from the time Norma wakes up till the time Norma goes to bed all year long, her focus, her thought is on SPF and how can we bring more people to the table. How can we make this work quicker? She's aggressive. She wants to find a treatment, a therapy that can help not me, maybe me, but for many of you, we have a daughter who has HSP, and we have 5 beautiful grandchildren that I watch every step they take. And you and them are the reason that we do what we do. And while I spend my time, I tell the board of directors, the best thing I can do for you is do all I can to keep Norma from wearing out. But she's in this every day because of her deep compassion and deep love for what we're doing. At the table with her this year is a person who's working with us. She's involved in fundraising and relationship development. Her name is Linda Arnold. You may have met her at the registration table, so if you haven't met her, stop and shake hands with her. But so many people across the board who are working daily to try to find an answer for the issues that we live with on a daily basis. Thank you for being here. Take in all the information you can. Take time to invest in new friendships. And as we leave this convention tomorrow afternoon, take information with you. Stay in touch. Some of you may be involved. Norma does routinely what we call SPF talks. If you've not ever participated in those, when you see one that's a topic that you might be interested in, plug in, get involved, and stay in touch with what's going on. We love you. We appreciate you. And with all that, I'm going to ask our MC. You guys know Tim Croghan. Tim's got a big job, and that's to keep us on schedule, keep us moving. So join me in welcoming our MC, Mr. Tim Croghan.
Thank you, Greg. I really appreciate that introduction. Usually I'm doing all the introductions. So this is very kind. So first of all, we want to thank everybody for being here. And real quick, I want to do, I just want to thank our sponsors, a few of our sponsors. I'm going to do this throughout the day, excuse me, but this is all being put together because of their assistance and their generosity for us. You got me, big guy? Sorry, just lost it. Excuse me. You guys like all those technical issues yesterday? Good, we're going to have a lot more of those today for you, okay? Just wanted to get you. Are we good on number 1, my man? I should be busting this room down pretty loud, okay? Anyway, everybody can hear me now. Fantastic. If not, you know I can carry this room. I don't need a mic. You guys have been watching me for many years. But real quick, I just wanted to say thank you to some of our sponsors that have been really helping us help put this together. And we want to start off with the Collin Foundation. Okay, thank you very much for your help and your assistance. And by the way, all these are listed in your programs, okay? So we want to thank the Collin Foundation. Regions Bank has helped us, and they work with us, and they have contributed with us. We like them. Also, you're going to meet this guy today, but his organization, the Tool Shed Training. You're going to meet Javier today. Great guy. You're going to like that. And then we're also going to go with our anonymous donors. There is a large group of anonymous donors that like to be anonymous, but they like to donate, and they like to help the foundation, and they like to help us put this event on. So for all of our anonymous donations, we thank you. Okay, so now we're going to get the show started, okay? So here's the official opening. Good morning, everyone, and welcome to the 2026 SPF HSP PLS Patient and Family Conference. Whether you have traveled from across town or across the world, whether you are attending in person or joining virtually, thank you for being a part of this extraordinary gathering. This year's theme is Pathway to the Gateway, a roadmap to health, therapies, and wellness. For more than 2 decades, SPF has brought together patients, families, clinicians, researchers, and advocates to learn from one another and build hope. But this year feels different. What began as a patient and family conference has evolved into something much larger, a convergence of patients, families, researchers, clinicians, international networks, NIH leaders, translational scientists, regulatory experts, and industry partners, all focused on a single question: how do we move from discovery to therapies? The convergence of the critical building blocks for therapeutic development is creating opportunities that would have seemed unimaginable only a decade ago. The challenge before us is no longer simply understanding these diseases. The challenge is organizing ourselves well enough to deliver therapies before patients lose function that can never be regained. Over the next 2 days, you will hear from some of the world's leading experts. Each speaker represents an important piece of the roadmap. Some are helping us understand disease biology. Others are building biomarkers, collaborative networks, assistive technologies, clinical trial readiness programs, rehabilitation approaches, and future therapeutic strategies. Together, they are helping create the pathway that may one day lead to treatments and ultimately cures for HSP and PLS and related neurodegenerative strategies. Disorders. Excuse me, skipped a page. As we begin today's scientific journey, it is fitting that we start with someone who represents one of the most important developments in HSP research in recent years. One of the recurring themes of this conference is the meaningful progress requires infrastructure. Scientific discoveries alone do not become therapies. They require coordinated networks, patient participation, natural history studies, biobanks, outcome measures, collaborative investigators, and long-term commitment. So, our first speaker today, I'm actually ahead of schedule by 8 minutes. Let's see if we can keep that up, okay, guys? We're going to try that. Hopefully, our speaker is waiting. We're going to have a virtual presentation. As principal investigator, here we go. Dr. Darius Ebrahimi-Fakhari has helped build exactly that foundation, as I mentioned before. As principal investigator of SP-CERN, the NIH-funded Spastic Paraplegia Centers of Excellence Research Network, Dr. Ebrahimi-Fakhari is helping unite patients, clinicians, and researchers across institutions to accelerate discovery and prepare the field for future clinical trials. Many people in this room may ultimately benefit from the infrastructure being built today through SPF-CERN. Please join me in welcoming Dr. Darius Ebrahimi-Fakhari virtually presenting SP-CERN and Collaborative Research Networks, how collaborative networks are accelerating HSP and PLS progress.
We got to change.
Okay, we're going to pivot.
Yeah, we got to pivot just a little. Dr. Darius hasn't joined our Zoom yet.
Okay.
So if you don't mind, if you'll move to the next speaker, which is Dr. Blackstone.
Okay, well, I can fill for 8 minutes. I've got a song and dance number ready to go.
We've got so much on the schedule.
Okay,
Let's
you
do
don't.
that on
Okay,
the
we'll keep that till midnight.
So, Dr.
All right, we're gonna skip ahead. Okay, and this is what we do. And here we go. So we're gonna skip ahead, and our next speaker. I'm gonna read what's on the script. You're gonna love this. If Darius helped explain how we build the infrastructure, our next speaker helps explain why we need it. Oh, hey, guess what? We're going back. This is great. Hey, guys, there's a thing in video. We'll fix this all in post. Nope, we're going back. Nope. Forward, backwards. Okay, we're going. We're going, okay, here we go. Now we're committed. Okay. Guys, it's only Tuesday morning. And guess what? We usually do this on the weekends, so this is great for a Tuesday. Okay. Throughout the history of HSP and PLS research, few individuals have contributed more to understanding disease mechanisms than Dr. Craig Blackstone. Many of the biological pathways being discussed throughout this conference, including axonal transport, organelle dynamics, membrane trafficking, endoplasmic reticulum function, and cellular stress responses, have been illuminated by discoveries from his laboratory and others working in these areas. One of the themes emerging across neurology is that many disorders may have common biological pathways. Understanding those pathways may ultimately reveal therapeutic opportunities that extend across multiple patient populations. Dr. Blackstone's work has helped create much of the scientific foundation upon which those opportunities now rest. Please join me in welcoming Dr. Craig Blackstone, presenting Disease Mechanisms and Therapeutic Targets, Understanding Disease Biology and Emerging Therapeutic Opportunities. Dr. Blackstone. And you got the two. Fantastic, sir.
Yes.
Thank you very much. And we got your slides right down here for you.
Perfect. Thank you so much.
You bet. Thank you, sir.
It won't sound that complicated when I talk about what I'm actually do. Can everyone hear me okay? Or is this one I have to really get close, huh? Okay, I'll be sure to do that. So it's like I'm performing or something, like which I can't do normally. So yeah, it's a real pleasure to be here again. Is it okay now? Is this close enough? Oh boy, you really do have to get right up on this. Well, it's a real pleasure to be here. And again, I think that I can't remember how many of these I've been to over the years. And like everyone that spoke this morning, like Greg and Tim, it's really been, you just feel the momentum in the HSP field moving closer and closer to therapies. And that's what excites us all. And that's why we're all here today, is for you. So I'm going to talk a bit about disease mechanisms and therapeutic targets for upper motor neuron diseases. I'm mostly going to talk about HSP because that's what I work on, but a lot of the same principles apply to PLS, and I think it's a really exciting time. And I'm going to talk a little bit more general than most of the speakers because I know many of you are new, and some of the words we throw around are kind of confusing. So I'm going to try to keep it relatively high level in terms of what I talk about, but of course I'm very willing to delve into more detail if that helps any of you. So my day job now is I oversee probably the world's largest neuroscience institute. I think if you add everybody together, it's over 1,000 faculty. It's a health system of 82,000 people. So my job is mostly to answer emails during the day, but it's also to try to bring people together. And that's what's so great, as I think all day long about how I can bring people together, how we can do science, how we can take care of patients better. And when I see all of you, you're already doing that so well. So just keep going because I think you'll see that the next phase of really bringing therapies to patients is really about teamwork and communicating and working together. And again, that's my day job. And I know it when it's done well, when I see it, and you're doing it very well. And again, just continue to do that. And again, when you think about trials and how hard they are to do, I can tell you in our institute, we're doing 800 trials at a given time, just in neuroscience. So it can happen. It happens in all different diseases, and HSP is going to be there soon, more and more. I'm very confident about that. So again, I have lots of disclosures. I get funding from different places. I consult a lot, and I work with companies. I won't talk about any of the companies, but I will sort of juice some of my general insights because I think it can be very important to think that as we move closer to therapies, we're going to have newer types of partners than we're used to working with. And that's an important part of getting this over the finish line. So one of the most important things about clinical trials, and this was a big problem with the early Alzheimer's trials, is making sure that when you test a therapy, you're testing it on the disease you think you are. Many of the early Alzheimer's trials, 30 to 40% of the people didn't have Alzheimer's disease. The trial has zero chance of success in that area. So it's very important that we understand what diseases you have. Now, HSP is great in that it has a genetic basis, and that allows us to be more sure and also to to divide people into different groups that might benefit from different therapies. The only reason I'm listing some of the other diagnoses here is I know many of you have probably been tested for them. They have different treatments. So it's important that we're sure you have HSP, that you don't have any one of those other diseases that's listed there, mainly because they have different treatments and you would benefit from different approaches if you have them. Now at the top, you'll notice that 30 to 40% of people we think have HSP don't test positive for a genetic, in one of the genetic studies. So that's a very important group of people that we have to do a better job of. We have to find more genes, and you'd think, geez, we have 100, isn't that enough? Well, it's not. We need to make sure that everybody with HSP gets a diagnosis, and that helps move closer to therapy. And that's why Darius and others talked about SP-CERN has a specific program within it for genetic analysis of people who don't yet have a diagnosis. And I know that applies to many of you. It applies to many of the people I see in my own clinics, and it's an area that we have to continue to work on. So again, one of the really key things is to understand, what the disease you have is as detailed as we can, because that ultimately helps us in terms of our therapeutic testing. Another feature is, if you'll notice on the right, is that it's a sort of a schematic, but the disease neurons that are involved in this disease are really long, like a meter long. And that meter long neuron can be sort of impacted in many different ways. That's another reason for us to be sure that you have HSP and not something else, because there's a lot of other disorders that can impact those nerves and that can look like HSP. So again, very important to make sure we have that diagnosis right. Now, when we think about HSP and PLS and the brain pathways, and I sort of alluded to a little bit of one of the key features is it's a neurons called upper motor neurons. In HSP, it's just the longest ones. That's why your legs are affected. In PLS, it can be shorter ones too. That's why your voice might be affected. That's why your arms might be affected. But another difference, of course, is the numbers of patients with these. HSP is far more common. HSP is mostly genetic. PLS, doesn't seem to be. So again, there's important distinctions, but it's also important to recognize the overlap. They are the same basic cell type, and hopefully that therapies that work for one have the potential, depending upon what they're targeting, to work for the other as well. It's most likely that a lot of the early studies will be done in HSP just because there's a lot more people and there's a lot more basic understanding of the pathways. But certainly I think there's a lot of hope for PLS as well as we make progress in the HSPs. As many of you know, there's many, many different genes, and that can get confusing. But as I'll show you, we don't have to think about it as 100 different diseases. It's a smaller group of diseases that all link together in common themes. And those common themes hopefully will benefit people with multiple different forms. But I think it's also important for us to not think of HSP as one disease, that it's probably a small number that we can coalesce around certain mechanisms, and that actually might make it more effective for therapy. If you think about common diseases like depression, high blood pressure, those are mixtures of lots of different diseases, and that can sometimes delay the development of specific therapies. But because we have this genetic sort of anchor for a lot of the HSPs, that's going to give us a lot of ability to, I think, to target the right therapy to the right patients at an earlier stage than most diseases. Now, if you think about the treatment, we always talk about that. We talk about the new ones we're trying to develop, but we have to remember that we have a lot of treatments for different symptoms. The key is that they're for symptoms. They're not changing the disease course. So if you look at the top, there's physical and occupational therapy, stretching, activity. That makes a huge difference in patients. There's medicines to decrease the spasticity. Some of them are oral. Some of them are through a pump in baclofen. There's some medicines that are used for other diseases that sometimes help. And there's medicines that have side effects and that can limit their use. There's injections like Botox that can help people in certain cases. And then there's a lot of other symptoms that come along with HSP and PLS, whether it's depression, anxiety, whether it's dystonia or other types of muscle tone abnormalities. It can affect your bladder. It can cause spasms that hurt and cause pain. And there's different things that we can do for that. And you'll see at the bottom, there's even neurostimulation paradigms. We have several patients that use the Bioness device or various types of stimulatory sleeves that they can put on their legs. But all of these, as far as we know, are therapies that target symptoms and not the underlying cause. So the goal, of course, as you can see in the bottom, is what we want, what you all want, is something that changes the disease course in a positive way. So how do we develop these therapies? We heard a lot yesterday about different approaches, but if I step back and take a very simple approach, and I know this is a lot of you know this already, but I figure some of you might not. When you have a mutation in a disease like HSP, that's a mutation in your DNA. Now, when your DNA is converted to RNA, that mutation kind of goes along with it. The RNA is abnormal too, and then often that leads to an abnormal protein or a absent protein. Now, when we talk about different therapies, when you hear the word gene therapy or all these RNA therapies, that's on the left. That's the DNA and RNA section. When you talk about a lot of the other therapies, you're really talking about the protein and the cells themselves. So in the next slide, and we talked about this yesterday, is how do we think about HSP as a smaller number of diseases? It's really overwhelming to think, how would we treat 100 different diseases? But when we have to group it down into a smaller number, it becomes more manageable. And this is a picture I drew like 15 years ago, and it was sort of trying to do the same thing we talked about yesterday. How do we group different HSP genes into various categories in a cell that we can treat? So we heard yesterday, like if you look in the bottom left, well, some of those diseases are SPG 11 and 15, and we heard that, well, there's a potential drug that's helping for lysosomes, and you can see it's in that little group. There's other groups like tubular ER, endoplasmic reticulum, microtubules, and you can see when we think about how all these proteins work, we do know a lot about what the basic biology is. We don't always understand how the mutation is causing a disease or causing these long axons to be dysfunctional, but we do have groupings that we can start with, and the technology is much better now than it used to be. So I think we're going to get better and better at sort of defining a small group of areas that we can focus our therapeutic efforts on. Something like this, but even a modern version of it. Now, when we think about what we already have, and I know that this can get confusing, so I wanted to just write it as simply as I could. We have a lot already. We have model systems. We have flies, we have worms, there's tadpoles, there's human stem cell-based models, there's mice and rats, and there's different ways we can read these out. What's important to remember is none of these are HSP. Only people have HSP. These are models that model certain aspects of the disease, and that's not the same as the disease itself. And it's important for us to remember that. We can make flies better, we can make a cell better. That doesn't always translate to making people better, but it's a start. And the more evidence we have in these early models, the faster we can move things to you. The reason we use these models is they're cheap and they're easy in many cases. In clinical studies in people are very expensive and time consuming. We want to make sure when we do those, we have the greatest chance of success. And that's what these models help us with. And I always put at the bottom technology. There's all these new approaches that we can continue to use, whether it's AI, whether it's better machines, better ways of analyzing data. All of that technology can be applied not only to these model systems, but also to help understand, the disease in people, not just in model systems. Now, we heard yesterday, I think it was Dick that brought up, well, why are we talking about trial readiness? Why are we talking about networks? Why aren't we talking about targets? Well, there are targets, and one of my long-term collaborators, June Lee and Peter Boss and many others that are here today, Honda, are studying specific pathways. It's just we didn't talk about them yesterday, and you'll hear about them today and probably tomorrow as well. But I just listed a few, but there's many others. So each of those pathways is being tested in different model systems, and the more evidence that's supportive of some improvement, the closer those will get to go to people. They won't all make it to people, but by doing all these early studies, the ones with the most promise will. So I think there's a lot of areas that we are targeting for therapy, and that these will ultimately get through sort of the gauntlet and ultimately end up in human clinical trials. Now, when I think about the therapeutic approaches for HSP, the number one is small molecules, and that's the one that you're most familiar with. That's a pill that you take. And that's, of course, going to be effective in a lot of people. I mean, one of the great things about small molecules is you can give them to anyone throughout the world. When we talk about complex gene therapy, that can't be given everywhere, because I work with a lot of populations throughout the world in different diseases, small islands in the Philippines and places like that. And gene therapy is not going there anytime soon, but a pill could. So we always need to think about small molecules, how we can develop those faster, better, more effectively. One of the exciting things many of you have heard about is gene therapies. How can we take these wonderful sort of therapies that make a bad gene go away or make a protein that you're missing appear? And these are working, and they're working in a lot of neurologic diseases, and patients are getting a lot better. They're hard to do, they're expensive, but they're working. And I think you're going to see a lot of innovation in this area, not just in HSP, but in other diseases as well. Now, there's another thing you can do. Instead of delivering a protein that's missing, you can knock down a bad one. And many of you might have autosomal dominant forms where they tell you, well, the protein, there's a normal protein, but there's a bad one too. And well, the normal one is fine, but we got to get rid of the bad one. So that's what knocking it down is. Let's get rid of it. And those are things called ASOs, or antisense oligonucleotides, or siRNAs. And many of you might even be involved in some of these sort of N of one, where they just do a couple people studies. One of the really exciting things that we're doing a little bit with now is repairing individual mutations. So you can go right into the genome, right at the DNA, and fix the mutation. And you can even fix it in the RNA. That's that next step. And there's a lot of potential in this area. Not easy to do, but again, it has amazing potential. So you'll see when you look at those top 4 options, there's a lot of ways we can treat this disease. We just have to make sure that we all work together, pick the right methods, and really put those resources into the most promising ones. Because each step of the way, as you get closer and closer to people, it gets a lot more expensive, and you don't get to do quite as many different options. You really have to sort of coalesce around a small number. But there's a lot of promise out there. And at the bottom is what we've been talking about yesterday. How do we figure out, how do we design our trials? How are we ready for when the therapies are ready to make sure we can get them going right away? And how do we innovate to make things easier for patients with rare diseases? Common diseases that have tens of millions of people, a lot of the trials, the big trials, are sort of focused on them. How do we do the same in a group of patients where the diseases are much rarer? And we've heard that. And of course, many of the forms of HSP fit into this category. So there's a lot of really positive things going on. And by communicating and working together, I think we'll gradually sort of narrow those down and be able to do this very effectively. So I wanted to give you one example of my own work and with June and several other people that we've done over the years, just to give you a flavor of what we do. I mean, you'll hear this from many people throughout the day, but I just wanted to take one form of HSP and sort of talk you through how we think about it as scientists and how we ultimately hope this leads to specific types of therapy and what our rationale is, how we get there. So SPG3A is one of the most common forms, and it was identified by John Fink in 2001. I started working on it soon after. It was the very first project I work on in HSP. It's mostly childhood onset, mostly pure, which means doesn't have a lot of other symptoms. But as we heard yesterday, it might be a mistake for us to think about these diseases as pure. They're not only upper motor neurons. There's other parts of the brain that are affected, and sometimes other parts of the body. Many of these proteins are not just expressed in the brain. They're expressed other places too. So it might not be so useful for us to think about things as being pure and to think more about, maybe the predominant symptoms, but not get hung up as much with pure versus, complex. The mutations are missense, which means an amino acid is changed one to another one or a small deletion. So, and these can be inherited. You can get them from your parents or grandparents and down the line, or they can be de novo. They can just appear. And some of you might have a genetic change that wasn't present present in your parents and it just came out of the blue. And sometimes that mechanism is helpful for us in terms of therapy and can guide us in different ways. Most patients slow progression, but there's a couple people, a small group that have very severe presentations. And that's an important thing to remember, is that just because you have the same type doesn't mean you have the same symptoms. And in some diseases, the variation can be tremendously large. And that can be helpful for us in terms of understanding the biology and also planning our trials. So when there's this variation, it can be very helpful, again, for us to think about that. How do we incorporate that into our trial design? How do we use that information to move things forward quicker? Now, in the case of SPG3A, and I won't show many of these sort of science slides. Basically, it's a protein in the endoplasmic reticulum. It's just an organelle in your cell. But what you'll notice, and I just show a picture on the right that's blown up, you'll notice that there's a whole bunch of genes that fit into that same category, and they're all binding to each other. So that gets to this idea is maybe there's a cluster of genes or conditions that all can cause the same type of basic pathology and ultimately treated the same way. So this is a case where you might think that these 7 or 8, or it might even be more than this now, this is one of those groups of coming together and making HSP a little bit more manageable as we think about how many different forms we have out there. Now, this is from Darius's work many years ago. He just took a whole bunch of SPG3A mutations. They're all listed along the bottom. And you'll notice a couple in red, and they're the ones that cause more severe disease. And you'll notice that they're all clustered together. So that's teaching us something. There's a specific part of this protein that if you change it in a certain way, you get a more severe symptoms, and that can be very helpful to us. So you don't have to look for your own mutation on here. It's probably there, though. There's 50 or 60 of them, and there's just a few that cause very severe disease. So if you think about the ones that don't cause severe disease, this is what you'd see typical HSP 3A. You have a mutation. It's one amino acid to another. We can understand the structure, how it changes. This is an enzyme. We can understand if it changes the enzyme activity. And we understand a lot about what's causing this to lead to disease. And in model systems, and this is June Lee's work that she did in collaboration with us many years ago, we can take cells from patients, skin cells, make them into stem cells, and make those stem cells into nerve cells. And we can see abnormalities. Like if you'll notice on the far left at the top, those axons, that's that long part of the cell, are normal length. You'll notice in the patient, and this is from that young girl I showed, they're short. We give a drug that's vinblastine or Taxol, which are microtubule binding drugs, and that length goes back to normal. Now, this is just one small step of what we need to do to get a drug to patients, but it shows that some of these abnormalities are reversible if we give medications. And to the extent that we can keep doing this kind of study and understand as we get these cells closer and closer to normal, that's what we want to do. So that's sort of the small molecule. That's the drug aspect of how we treat these. We use model systems to test our ideas and ultimately move them forward. However, we have other patients who have very severe forms. And I can tell you, I don't have the data here, but we've been working with a group in Ireland, Niamh O'Sullivan, and in working in flies, she's able to show something different about this mutation than the one I just showed you. So in other words, in some of our model systems, we're able to see why one form might be less severe than another, and importantly, why we might need to treat them differently. So even though they're the same HSP, different therapies might work for the different mutations. And I know that sounds very complicated, but it's good that we have models that can start sort of teasing the reasons for the clinical variation apart. And that would be very important for therapy. And again, I just wanted to show you a mouse, just because you've seen these. This is a mouse model on the bottom. It has what looks like HSP, and on the top is normal. So you can sort of see what kind of symptoms we're looking at. And then when we look at these symptoms and quantitate them, this is how we test our drugs. We see what abnormal is versus normal, and we try to make the abnormal mice behave normally by giving them gene therapies, medications, and so on. So I won't go through all these, but just like for people, we have clinical measures for our model systems, especially our in vivo or animal models, whereas in humans, of course, we have different types of assessments, but they focus on the same thing. In this case, they focus on how well the mouse can walk, how fast they walk. So different tests, of course, but some of the same basic principles that we apply for our clinical studies. And again, the closer we get to people, the more likely we are, I think, to succeed in terms of a therapy that works well for patients. Now, the way we design mouse studies is very much the way we design human studies, is we do them prospectively. We design it at the beginning, and then we start giving the mouse a medication over a period of time, and at the very end, we look at what the effect was. And some of the mice don't get the drug, and some of them do. And the people who are testing them don't know which one's which. So that's sort of this prospective, double-blinded, nobody really knows what's going on. We've started doing a lot of our tests with Jackson Laboratory and other laboratories, and that becomes important when you start thinking about regulatory agencies. They like to see these tests done by unbiased companies. So it gets expensive, but you start to use what they call CROs. We've started doing that for several of our trials because it makes it easier to move forward in the regulatory framework. Now, another thing I mentioned that we have is technology. And when we think about how we assess model systems, we can certainly do it behaviorally, but we want to really drill down to see what's that mutation, what's that abnormal gene doing to that cell or mouse or human. And this is a new technique of a different type of electron microscopy. And I won't go into the details of it, but what it is it allows us to see things better than we've ever seen them before at higher resolution. And here's just an example of what data looks like when we get it out. So you'll notice on the left and the right, we have thousands and thousands of sections. No human can go through this. So even though we did this 7 or 8 years ago, we used AI. We used the AI to find the organelle and circumscribe it and mark it, and then ultimately assemble all these sections into something like what you see here. So on the left is what ER in an axon, in a corticospinal axon, should look like, and on the right is what happens in people with SPG3A, one of the models. So you'll notice there's all these like little ladder-like structures that were never there. So now we have a cellular readout for what this disease might look like. We don't know if making that better makes people better, but it gives us things to test. And what was interesting, and this is years ago, of course, the AI couldn't do this initially because it was so abnormal. I bet the AI today could, but back then it was just sort of emerging, and it thought the ER would go one direction, and when it went the other direction, it didn't recognize it. So we still need people for some of these studies, but ultimately, once we taught the AI program how to do it, it was fine. So it still needs us too. So one of the things I think you can think about, and this is true for this ER one too, and I wanted to emphasize it, is that many times we see an abnormal cell process that applies to multiple diseases. So if you think about the ER being not shaped properly, that applies to some forms of ALS, some forms of sensory neuropathy, and HSP. And that gives us a huge advantage because that's more people. And more people means drug companies, funding agencies are more interested in supporting it. So I think that, and it gives us more options for how to test it. So I think that this is sort of just, again, I just wanted to take you through one HSP, but when you think about all the other ones, a lot of these same principles apply. We can learn things from our model systems, we understand the basic biology, and we can also see how these pathways might impact other diseases and how that sort of enlarges the tent of people who might benefit. So we have not only multiple HSPs that can benefit from a therapy, but potentially people living with other disorders as well. And the more we find those kind of relationships, the easier it is, I think, for us to move ideas forward because more and more patients are involved. So as a conclusion, I think this is a really exciting time for HSP therapeutics research. I think each year that we come, we're closer and closer. And again, many diseases, as you've seen in the nervous system, have had remarkable outcomes recently with different types of therapies, even traditional ones, or some of the new gene therapies. And all that momentum in these other diseases benefits HSP as well. So I think it's a really exciting time. I think SPF has done an amazing job of bringing people together, keeping them together, getting everyone working toward that same goal of better therapies for people with HSP, and not just ones that control symptoms, but ones that change the course of the disease. And again, each year, I know we are getting closer and closer. So I'm very optimistic, and I hope all you are as well. So thank you. Do you want me to answer questions? Yeah. I guess you want me to answer questions? Yeah. Yeah, we have a few minutes before Darius. If anyone has any questions, I could probably answer them. No?
Yep, I'll come to you. Hang on. I'm really good at this.
When do you think we'll actually have HSP being clinically tested?
I hope soon. Well, I mean, it is already. I mean, some of these gene therapies are already in children especially. They're already being tested. For the more traditional small molecules, a lot of it depends on how far along it is in the process. If it's a drug that's already FDA approved, it can move almost instantly. If it's a drug that has to be developed and tested for safety, it'll take, several years. So a lot of it just depends on what works in a lot of these model systems and where the funding is. Because again, it's very expensive to do trials. And so I think a lot of it is groups like this coming together and saying, where's the most potential? Where can we put our resources? Is there a company that's willing to back this or pursue it? But there's ideas that are out there. They're being, these trials are ongoing now for limited numbers of patients with some of the genetic forms. I think the small molecule forms, it could benefit a lot of other people. It really just does depend on what the most promising candidate is. If it's a drug that is already out there, it's very easy to move that to a trial almost instantly. Again, if it's one that has to be developed, it will take longer.
Is that happening now?
Sure. Again, when I showed all those pathways, and other people will speak about that today, these are being tested in model systems, and depending upon what the drug is, it will depend on how quickly it can be moved into people. So I think that you're going to see over the next several years that we're going to start seeing more and more HSP trials, and I think that's another reason that so much of yesterday and some of today is about trial readiness and how we organize, because these ideas are already coming. And again, just to emphasize, these trials are already ongoing, just for relatively limited numbers of patients.
Thank you.
Sure.
We got, okay, we'll go to the other side of the room. We got time for one more. Hang on, wait. Nope. Gotta wait. Nope. There's people out there in the world who want to hear your question.
So my question is, on one of your slides, you mentioned that late onset Friedreich ataxia mimics the SPG7. Could you just talk a little bit about that?
Yeah, I was going to mention that to some of the clinicians, and maybe John or other people have seen the same. We've had a couple of people come to Mass General recently through our ataxia program, which is led by Jeremy Schmaman, who's one of the world's experts in ataxia. And he sees quite a few of the people with SPG7 who have more of an ataxic presentation. The ones with spasticity come to me, the ones with ataxia go to him. But he's shown me a few people recently that for all the world looked like SPG7 to me, and it turned out they had late onset Friedreich's ataxia. So I think it is to emphasize that panels don't always catch all the possible diseases, and that was sort of my reason for emphasizing getting that diagnosis correct. But I think that some of the 30 to 40% of people that we think have HSP might have an unusual presentation of another disease like Friedreich's. So I've started looking at it in a lot more in people that I think have HSP but test negative with some of the traditional panels that don't include Friedreich's. Because Friedreich's normally is a different disease early, but when you get it late, late or very late, it can look very much like Parkinson's, like HSP, like other things. Now, I actually just put that on there this morning because I was going to remind myself to talk to some of the other physicians here to see if they're seeing that as well. Because again, literally, I saw 2 people recently in Jeremy's program that looked like they had SPG7 but tested negative, but they had Friedreich's, late onset Friedreich's. And of course, that has different therapeutic options. So again, I think that emphasizes in that early slide, we need to know what people have because there are different types of therapies being developed, and we want to make sure the right patient gets the right therapy. And that's if we don't do that, then almost no matter how good our drug is, we might not see a benefit. It's really critical to make sure that, again, we make sure everybody has the right diagnosis and they end up in the right study. Sure. Oh, sorry. Thanks. Thank you.
Thank you, Dr. Blackstone. We're going to take a couple of seconds here to get kind of reorganized, and we're looking good. Norma, do I have a thumbs up in the back of the house? I have thumbs up. Here we go. Okay, now we're gonna start kind of semi over again a little bit, but Dr. Darius Ebrahimi-Fakhari has helped build exactly the foundation we talked about earlier, about how all these efforts are all coming together. As principal investigator of SP-CERN, the NIH-funded Spastic Paraplegia Centers of Excellence Research Network, Dr. Ebrahimi-Fakhari is helping unite patients, clinicians, and researchers across institutions to accelerate discovery and prepare the field for future clinical trials. Many people in this room may ultimately benefit from the infrastructure being built today through SP-CERN. Please join me in welcoming Dr. Darius Ebrahimi-Fakhari virtually presenting SP-CERN and Collaborative Research Networks, how collaborative networks are accelerating HSP and PLS progress. Dr. Fakhari.
Thank you so much for this very kind introduction. I hope you can hear me okay.
Yes.
And it's a pleasure to be here today. I regret that I can't be there in person, but I hope to be back next year. Dr. Blackstone's talk was a perfect segue, I think, into the second talk. And my main mission today, this morning, is to introduce you to an initiative that we've built over the last 3 years that is hopefully accelerating a lot of these developments into a clinical stage. So my name is Darius Ebrahimi-Fakhari. I'm a pediatric neurologist and neuroscientist at Boston Children's Hospital and Harvard Medical School. My team and I, we conduct research on hereditary spastic paraplegia, and we look after about 700 families or so with HSP. So I'm speaking on behalf of the SP-CERN investigators, which is a group of 10 different sites, and I'll introduce you to the Spastic Paraplegia Centers of Excellence Research Network, or SP-CERN for short. So a quick outline for the next 30 to 40 minutes or so. I'll first give you a little bit of a background and talk about the unmet need. I'll then switch gears and talk about SP-CERN. I'll tell you about the rationale for building this consortium and its structure. I'll show you how this is positioned within the wider network of rare disease networks funded by the NIH. I'll then introduce you to the key 3 clinical research projects. The first is a natural history study, the second is a genomics platform, and the third is a platform for biomarkers. And then I'll talk a little bit more specifically about what we're hoping to achieve together, the patient communities we work with. I'll show you some work on blood-based biomarkers, how we structure this, and then talk a little bit about next steps as well. Okay, so with this, I just wanted to remind everybody that we have tremendous challenges ahead of us, but at the same time, there's also a positive development overall. In 2026, and this talk gave me, again, an opportunity to reflect on this, we have more clinical expertise than we ever had. We are seeing for the first time in adjacent fields, and hopefully soon in HSP as well, that we move from using genetics as a diagnostic tool to genetic approaches to therapies or gene therapy. We are building the infrastructure for clinical trial readiness. We have a lot of translational research that you'll hear about in the next days. We have, on the pediatric side, growing experience with first-in-human clinical trials. We are investing in the next generation, and we're building the community. So there's a lot to be proud of and a lot that is developing, but obviously we need to do more. So our team's mission, the SPCERN's mission, also my lab's mission, is to advance patient-centered translational research in childhood onset movement disorders by linking clinical insights to mechanistic discovery and therapeutic development, accelerating the path from molecular understanding to first-in-human studies and meaningful clinical impact. That is a long sentence. Basically, what we're trying to achieve is we're trying to accelerate the time from concept to proof of concept to a first-in-human clinical trial. That's the main mission. Now, this is easier said than done. The reality is drug development in the United States and most places in the world looks something like this. You have first, an unmet clinical need, and we as a community and as clinicians are very good at defining this. We can understand the symptoms, we can measure the symptoms in many different ways. We can think about therapies, and Dr. Blackstone talked about some of the pathways and targets that have already arisen from prior work. We can choose a therapeutic entity, put it forward as a candidate, test it first in cell models and animal models, do all the necessary safety studies, and then get it to a clinical trial. I think we're good at this part, at the left side of the slide. What we need to become good at is the right side of the slide, which is doing actual clinical trials. And this is not because the science isn't there. In many cases, it's because the funding is a problem. Because in HSP, as we all know, we talk about very, very small patient populations with limited abilities to attract industry funding and industry partners. Hopefully that's changing in the time. SPCERN is built to change that, basically. Now, how do we get to this stage? Reflecting on my own experience in Boston, we started this program 5 years ago, specifically with the mission of understanding childhood onset forms of hereditary spastic paraplegia, because we recognized there wasn't a lot of work being done at the time. So we started this natural history study, and we've had a remarkable experience in that all the families that we see in clinic are very supportive of our research, and we've had an opportunity, as you can see here, this is actually data from last year, we've had an opportunity to see over 700 families at this point in time, and many of these families come back for a second, third, fourth visit, and this gives us an understanding of the many symptoms that these children and young adults experience. So I'm not commenting on these videos in very much detail, but I want to show you that HSP, although there's convergence on one symptom, is not one disease. It can present in many, many different ways, including in very young children, as you can see in the top panel. It can present with more milder symptoms, relatively speaking, as you see in the second and third panel, or it can present with very severe symptoms, as you can see in the individual in the fourth panel. So it's a challenge to understand this and put this all into one basket, but the more patients we see, the better we become at this. So just in this very short period of time, we've made a lot of, I think, important insights. I won't go through this in detail, but I'll just say that this type of patient-centered work has really allowed us to close a few gaps for specific forms of HSP, and more work along these lines is needed. But what we want to get to is a platform where we can test therapies. And I would argue the HSPs have many advantages in that sense, because while we understand the symptoms better now, and while there's convergence on one set of symptoms, which is lower limb spasticity and weakness, we have all the different genetic mechanisms that we know of today represented in the different forms of HSP. So there are biallelic loss of function conditions. The prime example is SPG11 for that group. There are conditions with dominant negative or gain of function mechanisms. Certain patients, certain mutations in SPAS might work through this mechanism. There are conditions where haploinsufficiency is the mechanism. There are conditions where you have specific variants that can be targeted with specific modalities, such as so-called antisense oligonucleotides. So we have all of this represented. The task now is to attract people who develop these therapies, maybe not for HSP in the first run, but for HSP in the second and third run, and really give them a platform to test these therapies. So with this idea in mind, we expanded this natural history study and the network to 11 sites across the United States. You see them listed here. We have worked very hard to find people who are motivated, who are willing to donate, volunteer their time to help us build this network. We've had amazing support from the Spastic Paraplegia Foundations in the first 2 years, which really got us started. And then we were very fortunate to receive one of 7 large NIH grants that will fund us for the next 5 years to conduct this work. And this is no small achievement. It's the work of many, many people. I think the application had 1,100 pages at the end. So it took a lot of work and time to get here, but I think now we have something that we can use. I always compare this to building an engine and then hopefully driving the car as fast as we can. And I think we're at a good position now where HSP is finally at the table with many other rare diseases. And the Rare Disease Clinical Research Network, which is this NIH-funded structure, helps many rare diseases, and HSP is part of this. This was the first meeting that we had that we participated in representing HSP in our consortium. Now, what is the SP CERN? This is a little bit dry, but I want everybody to know what we know. This is a consortium that is now NIH funded for 5 years. There is an opportunity to extend that by another 5 years at the end of this cycle. We are one of now about 20 consortia for rare diseases, and we have a shared infrastructure, meaning that we all work with the same data management and coordinating center. We have standardized forms, we have standardized project management, we have a single ethics board, we provide training to young investigators, and we use these infrastructure really to achieve a number of things. The first is to develop disease area expertise in both pediatric and adult onset HSP, to develop the first multi-site biorepository and long-read genomics data set, to harmonize outcomes and study the natural history in a harmonized way, and then to develop career development pathways for clinician scientists, specifically in HSP, so that we have the next generation working on the problems of tomorrow. So how do we do this? We have basically a central infrastructure, and as you see listed here on the left. We have 3 clinical research projects, a natural history study, a genomics platform, a digital biomarker platform, and we have a central biorepository. We work very closely with the Spastic Paraplegia Foundation and other patient advocacy organizations. We work very hard, and I'll come back to this at the end, to build bridges with consortia in Europe, South America, in Canada, and in South Korea. Again, this is all to really share data and to disseminate this information as widely as possible. So what are these sites? We have 10 active sites, as you can see listed here. We already have 2 sites who are joining this network very soon, hopefully, Northwestern in Chicago and the Children's Hospital of Philadelphia in Philadelphia. Since October, we've enrolled 144 participants, so we've had a relatively slow start, but the numbers are picking up very quickly. I presented this to our friends in Canada just 2 months ago, and I think the number was closer to 80. So we're picking up speed, and that's really wonderful to see. And this is across the country. So you see the first enrollments here in Cincinnati and in Iowa, for example. This is a busy slide, shows you how we're organized. We're sort of orchestrating this network here from our team in Boston, but work with many around the country. This is just some of the core infrastructure. I won't go through the details here, but just to say that we built in 2 pieces that will be helpful, I think, for many ideas in the HSP field. The first is a so-called career enhancement core, where over the course of 5 years, we'll be able to support the salary of 5 young fellows who are coming to train specifically in the care and research for HSP. This is an investment that is, I think, very forward-looking, because if we attract talented people at this stage of their career, this will benefit the field of HSP for decades to come. So that is something that we really wanted to emphasize, and we have 2 excellent first fellows this year. At the same time, we also acknowledge that there are sometimes, ideas out of left field that are exciting, that are high risk, potentially high reward, and it's incredibly and increasingly difficult to get funding for these kinds of ideas. With the funding constraints of the current climate and with the changes introduced by the current administration, it is very difficult to get funding for high risk, high reward type of ideas. So we wanted to mitigate this risk and introduce the program where every year we give out 2 small grants of $50,000 for high risk, high reward research projects that are meant to generate data that can then be used for a larger grant application. I think this is very important because sometimes these high risk, high reward ideas, they catch fire and they really change the course of the field. So that's something that is very important to us, and that will be rolled out every year. So speaking about this, our first 2 fellows are here, Dr. I and Dr. Aguianda. You see their projects listed here. Dr. I is studying genetic modifiers of age at onset in SPG4. If you have SPG4 and have multiple family members affected and have noticed that between different family members, there is a difference in terms of symptom severity and when the symptoms started, please contact us because we're specifically interested in these kind of families because they allow us to maybe understand why some people have milder symptoms and why some people have more severe symptoms, despite the fact that they come from the same family and have the same mutation. So please reach out to us. I think Dr. I has been able to recruit 60 or 70 family members already, or members of families. And we need more because this is probably a needle in the haystack situation where we need to look at many, many families to find these genetic modifiers. The second project is one that I think is very timely, and I was very pleased that the independent advisory committee awarded this grant to Dr. Aguianda because she's going to work on biomarkers, which is the bottleneck for almost all forms of HSP. What are biomarkers? Biomarkers, simply put, are proteins or other molecules in blood that you can measure to know if somebody has a disease or to know if somebody's disease has progressed. You probably are all familiar with hemoglobin A1C for diabetes. It's a simple screening and monitoring tool. We need something like that for HSP, and she's going to try and find that using the latest of technology. So that's a really important project, I think. Moving on to the pilot project core, this year's pilot project was awarded to Dr. Sanderson from the University of Washington for a project that has relevance to patients today, which is to look at transcutaneous spinal cord stimulation for individuals with hereditary spastic paraplegia. This is a study that studies non-invasive neuromodulation in adults with HSP. So if you're interested in participating in this study, please contact the SPCERN and we'll make sure Dr. Sanderson knows about your interest. Again, he's just starting, but this is an important avenue. And again, it's kind of one of those high risk, high reward projects, hopefully with useful data for the years to come. I've mentioned already that we work very closely with the Spastic Paraplegia Foundation. We wouldn't be here without the initial support that got us started. But we also work with many other smaller foundations and really try to bring everybody together around this shared cause. So in terms of other research projects, we have a natural history study where we meet with affected individuals and their families on a yearly basis. We conduct a clinic visit. Some of you in the audience may have come and seen us for one of these. Basically, you undergo a standard visit, a standardized visit, a standard neurological examination, different tests. We obtain blood samples and really use all of this information to map out how HSP changes over time. At the same time, we and others recognize that not everybody who has HSP receives a genetic diagnosis with standard clinical testing. So in order to close this gap, but also in order to find new genes and maybe genetic modifiers, we are able to offer a limited set of advanced genomic testing using the latest of technology. This is an effort led by our colleague, Dr. Zirkner at the University of Miami. So if you have HSP, but you don't know which gene you have, this is a study that you can participate in. And lastly, We all know that a clinic visit is a short 60 or 90 minutes. Most of your life happens outside of these 90 minutes. So in order to be able to understand symptoms better in your native environment, in your home environment, we've introduced a third arm in the study where we use wearable sensors, the types of sensors you wear around your hips, around your ankles, around your wrists, and that capture data 24/7, because we think that this might be a useful approach to really map out symptoms outside of the clinic. So who can participate? Anybody with a diagnosis of HSP can participate in different arms. So if you have SPG4, SPG3A, SPG5A, you are in our sort of one of our core cohorts. We call it cohort A. If you have a childhood onset form of HSP, you're in cohort B. If you have any other type of HSP, you're in cohort C. If you don't have a genetic diagnosis yet, you're in cohort D. And cohort E is after we found something using research genetic testing, you then enter the other cohorts. So we have something for everybody is the message. And if you're interested in participating, reach out to us by email or through the website. It's very easy to find. If you put in SPCERN in Google, you'll find it. And that way we can help you find the center that is closest to you. So this is what a visit looks like. We do all of these tests. We've trained all the operators in doing these tests. We ask you to complete a couple of questionnaires which help us gain your perspective. What is really impacting your life? What problems and challenges do you face because of your symptoms? So this is really important to us. This is just a little bit of how it works, how our biobank works. All of the samples are collected in the central biobank and are accessible. They're also accessible to anybody who wants to study them. So they're not meant to sit in the freezer, they're meant to be used. So we share them with everybody who wants to study HSP. This is just an example of one of our little patients who's completed one of his visits. You can see here we have a little checklist. And then for Research Project 2, which is the genomics platform, we also collect samples and then do a number of very sophisticated genetic tests. Again, this is for people who don't have a genetic diagnosis yet. These are some of the examples of sensors that we give out. You wear these sensors for a week or in clinic with us. This way we gather information. This is led by our colleagues at St. Jude Children's Research Hospital in Memphis by Dr. Burns and the team. So really experts in this area. And we hope that we'll gain a lot of insights from this. So what is our target? Our target is 200 individuals with SPG4, 100 individuals with some of the other core HSPs. We really want to include everybody, but have a specific focus on the types of HSP that you see listed here for various reasons. But again, this is open to anybody of any age. So please participate, please support us. And hopefully we'll see more examples of clinical trials reaching a phase 1, phase 2, or even later stage. These are two examples of two gene therapies for rare forms of HSP that have been successfully developed to a clinical stage. This is for SPG50 and SPG47. We need more examples like this. And I think SPCERN hopefully will help us with that, but a lot of additional help is needed. So building on these two examples, we are conducting a very critical gap analysis of all the HSPs to see which ones are ready, where are the gaps, and this is something that we'll hopefully share later this year with the community. We want this to be transparent. So when a company comes along and says, oh, well, how about HSP for our therapeutic entity? They can look at this map and say, oh, okay, for SPG4, these opportunities exist. For SPG7, these opportunities exist. For a different type of HSP, there's everything there, but we're missing a biomarker. This kind of information needs to be accessible, and we're hoping to, in the first year, define this for everybody to see. A specific emphasis is on SPG4, simply because SPG4, based on our data, affects about 50% of all individuals with HSP. The numbers are variable, but it's sufficing to say that's the most common type of HSP, and therefore I think really stands out. We are, I think, we have built a very good engine in the United States, but this is a rare disease. We all benefit from working together with international partners. So we've used the last 6 months to build these bridges, and I'm very happy to say that we've built wonderful collaborations with the partners in Canada, Europe, South America, and in South Korea. And we're looking to work with them using a very nimble, but I think a very efficient strategy, which is to say, we're all collecting the same minimal data set that we can then share between the different consortia. This will give us a lot more power, and I think is the right way to do it. And this can develop obviously in many different ways, and hopefully also in clinical trials that we do in parallel in all of these different locations. That's of course a much larger undertaking, but I think if we build the foundation for it, that is possible. So I'll summarize, and I appreciate you all listening to me. I wanted to show you that there are tremendous opportunities in our field. I think childhood onset HSP is a little bit of a paradigm for rare diseases in general. We face the same challenges and the same opportunities we face for many rare neurological diseases. Clinical trial readiness programs are essential for any meaningful clinical trial design. So we really need to invest and have invested in this. We have a very strong central administrative core, which organizes the SPCER network. We're thinking about the future with these career enhancement and pilot project initiatives for emerging ideas and early career investigators. There is an opportunity here for others to help us. Sometimes we have more good ideas that we could fund. So there's an opportunity to work together to bring these ideas to fruition. I've mentioned our natural history study, which is Project One. This will be the central clinical trial readiness platform. And then we are very committed to international collaboration, which is the best way forward for HSP patients around the world. So with this, I have to thank the many SPCERN collaborators, the many investigators who are again donating their time to make this happen. This includes the individuals listed on the right: Dr. Blackstone, Dr. Cashman from Mass General Hospital here in Boston, Dr. Zuckner from the University of Miami, Dr. Gilbert from Cincinnati Children's Hospital, Dr. Fink from the University of Michigan, Dr. Finkel and Burns from St. Jude Children's Hospital in Memphis, Dr. Shai from Iowa, Dr. Calamy from Houston, Dr. Christie from Dallas, Dr. Davies from Seattle, and Dr. Mingborn-Chisuk from Seattle. So we're all committed to this, and it's really a wonderful group, and I hope we can make big strides in the years to come. Thank you again for listening, and thank you all for your support. If you would like to participate in our research program, please Google SPCERN. I'm sure the website and the email are easily accessible. So thank you for listening, and I'll be happy to take any questions.
Hey guys, we got time for maybe one question. Doctor, let me get that for you. Okay, here we go.
When will PLS research be included?
Yeah, it's a great question. So one challenge that we have faced is the Rare Disease Consortium. I hope you can hear me, but the Rare Disease
Hold on just a
Consortium.
second, Dr. Fakhari. Hold on just a second, Tim.
Second, our sound guys working on another issue. Here we go. The question was, Doctor, when will PLS research be included?
Yeah, it's a great question. It's something that we're working on. The challenge that we had in the beginning is, unlike HSP, PLS is not defined by a set of genes. So the consortium that we've applied for funding for is specifically for genetic conditions. That's not to say that we can't include PLS. So we would be very happy to support PLS, work together with the natural history study at Columbia University that exists, And specifically also, I think if there's a question of whether someone has HSP or PLS, I think the second research project where you have advanced genetic testing is a potential way to answer that question. So I would encourage you to participate. We include PLS the same way we include any form of HSP, but we have the challenge that we don't have a gene for PLS, whereas we have genes for HSP.
Thank you, Doctor. So we're going to move on. Doctor, we appreciate you being with us this morning.
Thank you.
I think we have one more question.
Does the funding for your 5-year funding, does that end this year? Should we start lobbying our Congress people, or how can we help advocate to get that renewed?
Yeah, thank you for that, and thank you for offering your support. The 5-year cycle ends in 2030. So we have just been awarded this grant in October of 2025. So we have a little bit of runway. I would say 3 years in, we need to think about the renewal. So we have a little bit of time. That's not to say that it isn't helpful to have support now, because as you will see, we have more good ideas and more things we can do that we can with the resources of this consortium. The consortium doesn't solve everything, but it provides a basic infrastructure to tackle a lot of problems. So if something comes around that we think is highly valuable and is high yield, high reward, supporting that through other avenues is something that we hope to achieve. And I'll say that other consortia in this network for other conditions have done this very successfully. So this joint partnership has really enabled a lot of things in other fields.
Thank you, Doctor. We really appreciate you being with us this morning, and we hope to hear great more things from you. Have a great day.
Thank you very much. Thank you.
Hey guys, a couple of quick notes. Somebody left a cell phone on a table a little while ago that was ringing, and it ended up, there we go, okay? I'm going to set this right here. Couple other quick notes. In your welcome bags, there's a really great piece of important information in there. It's the pathway manuscript. Make sure you read that and understand it. It's got a lot of great information in there. Also, in the our big conference booklet, on page 4 has our list of donors in them. So make sure you read over that and support them if you can. Also, in your bag, we have some coupons there's one for Cadence shoes there should be in your book bag or in your bag, and in the once again in the big booklet there is a coupon for the A-Linker is in that. And another quick note about Dr. Fakhari's presentation: you can get those links that he was talking about earlier. They're on our website under the research tab, under the SP CERN tab, under research. Okay, you guys all know how to drop down menus and click, click, click. You can find those there, okay? And also remember, we also have the Be Healthy Expo going along in the other, across the hallway in those rooms over there. Make sure you get a chance to go over there. And also, there's still time to do, putting, all your green and red little pieces of paper in all the baskets over there. We got that going on later this evening, so make sure you get a chance to go across the hall. We are now ready for Dr. Fink. So as we continue building the roadmap towards therapies, our next speaker helps bridge one of the most important gaps in medicine, the distance between scientific discovery and meaningful patient impact. For decades, Dr. John Fink has been one of the leading clinical voices in HSP. His work has helped define how these disorders are diagnosed, understood, and evaluated in patients. Importantly, he has also helped the field think critically about what will be required to move discoveries from the laboratory into disease, into the real-world therapeutic development. One lesson emerging across neurology is that discoveries alone are not enough. Therapies require clinical infrastructure, measurable outcomes, natural history data, and clear understanding of how disease progresses in patients. Today, Dr. Fink will discuss how we move from scientific understanding towards therapeutic readiness. Please join me in welcoming Dr. John Fink presenting Clinical Transition and Therapeutic Readiness, Bridging Scientific Discovery into Real Therapeutic Pathways. Dr. Fink. Let me go get your clicker for you.
I've got it.
You got it with you? Wonderful. And you can see that right down there?
That bright light is very bright. Does that need to be that way?
Yes.
I hope I survive.
You'll be fine.
We'll see. I may get a migraine. Okay, good morning, everybody. I'm very pleased and honored to participate today. And I say this every time I've come. I learn from everybody here. This is a learning for me. Every time I see somebody in clinic, every time I see somebody in the hall, I'm learning about PLS. I'm learning about HSP. You know, you collectively, individually, are the experts. And we, as physicians and scientists, are students. And it's humbling to know, it's humbling to realize how little we know, how we have to translate your lived experience into scientific knowledge, into scientific jargon that we can manipulate and understand and translate. So I'm glad to be here and I'm taking notes. Now, we are doing today, as you know, a gait study. Anybody can participate, and it's across the hall. It's always a sign up and wait. It's inconvenient, and I appreciate everybody's patience. We'll see as many people as we can. We're talking today about, about clinical translation. That is, to go from scientific discovery into a clinical trial, or to go from a clinical measurement into a scientific data collection. It goes both from science into a clinical trial and from clinic into a scientific study. It goes both ways. I am always talking to many different people at once. I'm aware of that. We're talking to people with advanced scientific knowledge. I am talking to people now with advanced scientific knowledge and to people without advanced scientific knowledge. I'm talking also to leaders in the field, both scientific leaders, foundation leaders. So I'm talking to many different people, the foundation, leaders, families who don't have the condition, individuals who do have the conditions, plural, scientists, I recognize that. And I'm going to speak mainly in generalities, trying to address different audiences simultaneously. So, let's go forward. I want to make a couple philosophical points. First of all chemical reactions, every chemical reaction, and physiologic processes proceed in two directions. All of them do. Here's a reaction, H2O, to form water. It actually proceeds in both directions. The dissolution or the reversion of water back into its elements, hydrogen and oxygen, is actually occurring, but under most conditions is occurring at a very, very, very slow rate. So this is an equilibrium. All chemical reactions, let's talk about all physiologic reactions. Well, you know, a cell is at a very undifferentiated state, then becomes differentiated, for example, into skin or into a nerve or into a lung cell or kidney, whatever. And we used to think that that was a terminal differentiation. It occurred in one direction. Now we've realized, actually, philosophers and and plant biologists have known for plant biologists have known for hundreds of years that that there's de-differentiation and it goes both ways. And now we realize that even fully differentiated cells, highly specialized cells, can, under various circumstances, completely, or to a lesser extent, somewhat de-differentiate. Why am I talking about this? Because healing is possible. Degeneration, now I didn't say quick, I didn't say simple, I didn't say I know how to do it. I'm just talking in theory. that healing is possible. Regeneration is possible. Now, once a cell dies, that cell is dead. It's not going to come back to life. So, but there are, in some circumstances, additional neurostem cells that can repopulate, albeit at this moment in our scientific knowledge, at a very, very minimal and inadequate manner. But the cells that are dead are not going to come back to life, but there are other resident cells that might be able, under some circumstances, to multiply. Or at any rate, without talking about cell death, just talking about cell injury and cell degeneration, those processes are potentially reversible. If the cell is alive, there's a potential for healing and regeneration. Now, I didn't say quick, I didn't say it's available today, but these are possible. So just think about this concept that all physiologic processes, including degeneration and regeneration, are reversible. They're at equilibriums. Sometimes the rate of the reaction favors degeneration, and we need to learn how to shift the circumstance so that it favors regeneration and healing. These are possible. They're possible in experimental models. They're demonstrated in experimental models. I also want to talk about another philosophical point, and that is the concept, it's a dual concept of a catalyst and a disruptor. What's the difference? It's the same thing. A catalyst induces change. Changes the dynamics of a chemical reaction or physiologic process or other situation. That's a catalyst, induces change. And a disruptor induces change. It's just a judgment about how you view, the person could be a catalyst to induce change, but people that want to maintain the status quo view that as a disruptor. It's just a different perspective of the same. We need some catalysts. Catalyst is a judgment-free term. Disruptor is a judgment attached to it. I'm not going to talk about the judgment, but we need catalysts. We need to induce change. We need to expedite. What we're doing now is good and too slow. And what we're doing now is I don't want to say uninspired. It is very inspired, but it is the path forward that we know it will work for drug discovery, for advances. It's all good, and it's way too slow. And we need to be catalysts for change, to expedite the reactions collectively to facilitate drug discovery and clinical trials. And so those are two philosophical concepts. Now let's talk about the issues about facilitating clinical trials and trial readiness. Okay, this is important. What are the goals of therapy? What do we want? Well, we want to prevent the disorder, okay? We want to stop the worsening of the disorder. We want to reverse the neurologic injury. We want to reduce the symptoms. We want to improve function. We want to restore independence. None of these are duplicates. Improving function, improving the ability to walk, it's fabulous. We want to restore independence. We want beyond improved ability to walk. We want beyond improving speech and swallowing. We want it to be we want to be independent of the burden of impaired speech or swallowing or dexterity. So these are the goals of treatment. Okay. Now, why do I talk about that? Because for each of these goals of treatment, now let's don't use the word treatment, let's talk about a clinical trial. A treatment is an established approach to reduce the symptoms or the progression or the burden of the condition. That's a treatment, and every treatment has a limitation in its effectiveness, and every treatment has a statement about side effects and safety. No treatments are completely effective, and no treatments are completely without side effects. Those are treatments. They're known to be effective under certain circumstances, and they're known to be safe within certain range. That's treatment. But we're not talking about treatments today. We're talking about trials. Trials are unproven. We need to establish their effectiveness. We need to establish their safety. These are trials. Okay. Now, when I talked before about the goals of treatment, to prevent the condition, stop the worsening, reverse the injury, reduce the symptoms, improve function, and restore independence, each of these goals of a trial requires different outcome measurements. Okay? Outcome measurement. How are we going to judge if a trial prevents the disorder? How are we going to judge that if it prevents the disorder? Well, for example, Dr. Fakhari yesterday showed a slide about some improved rate of gait acquisition in children who are on the gene therapy trial. Okay, so gait was delayed, but it was less delayed, substantially less delayed, in some children who received the gene therapy trial. So if you say that the symptom onset of gait worsening was potentially delayed, that is we didn't prevent the disorder, but we delayed the onset of symptoms. So when we talk about preventing the disorder, let's get down into the weeds a bit. We can prevent the symptom onset. That may be a goal. I'm not saying we can, but that may be a goal, to prevent the symptom onset. However, the subject in whom we prevented the symptom onset that has a genetic condition is still capable of passing on that genetic condition to the next generation. So we might succeed in a clinical trial in stopping the symptoms from coming on. Somebody is identified as having SPG4, and we can intervene at some way, God willing, and stop the symptoms from coming on. The person still has SPG4 and could still transmit it under those circumstances. So one way would be to stop the symptom onset. Another way would be to correct the gene mutation. So not only did we stop the symptom onset, but we corrected the gene mutation, and now the person, if we could correct it in stem cells, in the sperm or egg producing cells, then they would no longer even have the condition to pass on. So there's various levels. So my point is, each of the goals of treatment, when we talk about a clinical trial that we're trying to establish, a trial we have to think about what are we trying to achieve. Okay, let's move on, because these determine what are we going to measure. Let's talk about stopping worsening or reversing the neurologic injury. We could treat the underlying disease mechanism. We know biochemically what the problem is, or we can try to figure out what the problem is. If we have an identified gene, then we have great insight into what the biochemistry and molecular processes are. We can try to intervene to stop that process. Okay. But then we can try to intervene to change the secondary injury. The primary problem affects, for example, the endoplasmic reticulum, or it affects let's say the axons. The axons are involved in transport it involves the microtubules, I meant to say. Let's say primary process affects microtubules. Microtubules are involved in moving cargo up and down the length of the axon. Microtubules are a cellular structure involved in moving cargo up and down the length of long nerves, besides giving structure to the nerves. And one of the cargoes that are transported are mitochondria. And so if there's a primary problem in the microtubules, that is the tracks that are necessary for carrying cargo up and down the long nerves, and one of the things that's transported on those microtubule tracks, those rails, is mitochondria, then there can be mitochondrial impairment, functional mitochondrial impairment, even though the mitochondria are healthy, they're not distributed where they should be. So there's a functional mitochondrial impairment as a secondary consequence of an axonal transport problem. So we can treat the underlying problem, that's one thing. We may also want to treat the secondary molecular or cellular consequences. And each of these that is a simple explanation of a very complicated field. And not complicated in terms of difficult to understand, but complex because there are many, many participating factors. It's not complicated intellectually, it's complex because of the numerous factors that are participating. And so we can treat the underlying disease mechanism, we can treat the secondary consequences of that underlying problem, and then importantly, we have opportunities to augment the regenerative or compensatory processes. When a cell is injured, it reacts to injury by compensation, molecular compensation. Pathways are augmented to keep the cell alive and healthy and functioning as best it can. So while there's injury ongoing, there's already compensation. And it's in every cell. And we need to learn how to, we can treat the underlying process, or we can treat the compensatory process to augment it, to facilitate recovery. Okay. Now, let's talk about reducing symptoms. All right. We want to promote healing. We want to augment regeneration. Now, let's talk about increasing strength, reducing spasticity, increasing speed of movement. These are all achievable goals. Achievable targets. But I want to point out that improving function is a different category than reducing symptoms. For example, let's say that a person has severe spasticity, marked spasticity in their legs, a very difficult time moving their legs. And we use a baclofen pump, and the legs have never felt better, never felt more relaxed. It's so wonderful feeling. But there is such profound weakness that the person can't stand. Now, that's not always with baclofen pumps. That's a downside and something we have to watch out for and make sure who is a good candidate for baclofen pumps. I'm not trying to make any comment about that approach to treatment, which has helped many, many people. I'm just saying sometimes reducing the symptom, for example, spasticity, to a large extent, may not improve the function. It did not improve gait. It reduced the symptom, but didn't improve the function. So we want to be careful. Are we trying to reduce symptoms? And we can measure spasticity, and we can measure strength, but what we really care about, we do care about those symptoms. But what we also care about is the function. We want to make sure that, we want to know what our targets of trials are. Why am I talking about this? It's because each of these targets is going to dictate what kind of outcome parameters we look for. Certainly, we want to restore independence. That is a big goal. That's an overarching goal. To make a person's spasticity less is important. But that's not the total goal. That's one goal. Really, we want to restore independence. We want to erase, or we start out with diminish. We want to ultimately erase the personal and family burden of not just the person who has it, but to their family, of having HSP or PLS. Now, when we talk about these, why am I going into detail? Because each of these, am I shouting? Each of these determines what outcome parameters we're going to look at. There are different outcome parameters depending on the goals of the treatment. Now, if you say, well, we're going to just give this treatment and see what happens. Okay, we can do that. But we really should say, what is the treatment that we're doing? What is it designed to do? Is it designed to treat a molecular process? I'll get into this in a minute. I'm getting ahead of myself. But if the treatment is designed to interfere with a certain molecular process, then the outcome parameter we're going to look at are going to be biomarkers reporting as best we can that molecular process. If the treatment we're going to be giving is to change a symptom, then we're going to need inventories and self-assessments about the magnitude of that symptom. If the treatment is going to be a functional treatment, then we're going to look at functional outcomes. But so my point is, we could say we don't really know what to expect. This is a difficult place to be, and that's actually where we are. We don't really know what to expect when we give this trial of new agent X. What do we expect? We don't know. So we're going to have to collect, think about this, because we don't really know necessarily what parameter the agent X is going to affect. Is it going to affect symptoms, function, neurologic performance, so forth? We have to collect then data in every one of these areas as a massive data collection. It's not a targeted data collection. So we have to be careful of that, because that is not difficult. It just increases the complexity, time, cost, reduces the number of participants in the study. The more focused is our question. We're giving a drug or a gene therapy or something to affect this parameter, and we're going to measure this parameter. The clearer, the more efficient the study can be. So let me back up. I talk about this. Any clinical trial, these are elementary points, any clinical trial is done, you make a measurement of something, you do an intervention of something. The intervention could just be time. We're going to measure somebody, and then in their walking, for example, or their speaking, and then time will pass, and then we'll measure them again. If there's no other administration of a study agent, just time alone, that's an observational study or a natural history study. But if we're going to do, it could be interventional, where we give something, and then we repeat the measurement. And then importantly, we have to make a comparison, either to the person before and the person afterwards, or the person of a treated group to an untreated group, between subjects or within subjects, so forth. That's basic. Now, if we're talking about what are the outcomes that we're going to use, let's just go into the details here for a minute. If we're going to try to, let's say we're going to try to stop the worsening. That's the goal of this trial. We're going to stop the worsening, okay? Well, worsening would be measured on neurologic terms. So for example, what's the spinal cord conduction velocity? How does the spinal cord work? And how efficient is it in transmitting nerve impulses? Well, if that's the goal, we're going to try something. We're going to try Ampyra to see if it makes the nerves conduct faster in the spinal cord. Then we'd have to do a spinal cord conduction, for example, a spinal cord conduction study beforehand, a spinal cord conduction study afterwards. That would be an important, or something like that. Or if we think that this would help peripheral nerve function, we would do a nerve conduction study at the beginning and nerve conduction study at the end. If we're looking for neurologic outcome parameters, or if we're looking at, for example, MRI, that's also a neurologic structural outcome parameter. Where there's tract abnormalities in the nerve fibers in the brain, spinal cord, and we're going to see, or the corpus callosum shape and so forth. We're going to make these measurements at the beginning, and then we're going to do a study, and then we're going to make these measurements afterwards. Those are for neurologic change. And there's also this business about neurologic examination, and that's in measuring neurologic strength and spasticity and reflexes and so forth. That could be part of measuring neurologic change. What if we're trying to, as I say, if we're trying to treat the underlying condition, we'd of course have to measure biomarkers, molecular chemical changes. If we're trying to reduce symptoms, then the outcome parameters would be in questionnaires. If we're trying to improve function, then the outcome parameters would be functional tests. What's a functional test? Well, how easily can a person get out of a chair? How easily can a person how much walking does a person do? How much are they restricted in their day-to-day functions because of walking or because of using their hands or with their speaking? Can you speak on a phone or do you have to have a special diet to accommodate swallowing problems and so forth? These are functional tests. And so what we're trying to achieve in a study will dictate the kind of outcomes that we select for study. Now, just to talk a little bit about these outcome parameters. The outcome parameters, and we could talk for days about outcome parameters. Well, outcome parameters, these are what we're going to measure. We have to consider the variability of an outcome parameter. That is, if a person comes in for the study and they're having a neurologic exam at the beginning and then they're going to get a study drug or study something, some kind of treatment, and then we're going to repeat the neurologic exam, we have to look at how variable are the findings on neurologic exam. Okay, so for example, let's say a person comes in and I examine them. And then they go out to the next room and Dr. Blackstone examines them. Okay, well, Dr. Blackstone is a much kinder person than I am, and people may warm up to Dr. Blackstone, and he may get better data or different data. And then they walk into another room and they see Dr. Fakhari, who's an outstanding physician, and they may get different. How close are the repeated examinations done on the same day by different examiners on the same person? They're not completely in agreement. There's a variation. The person hasn't changed, but the manifestation of the symptom or the appreciation of the magnitude of the condition is different in different examiners. That's an issue. We need to define that for every neurologic parameter. When the reflexes, what's the variability in reflex testing between examiners? What's the strength testing? What's the modified Ashworth scale assessment of spasticity? How variable is that? Every one of these things needs to be standardized between examiners. But besides that, the condition itself is variable, especially, for example, in cold weather. In Michigan, when people are driving 2 hours to clinic in the cold weather and they get out of the car where it was warm and now they have to walk in the parking lot in the cold and their muscles get very, very tight, and then they come into clinic and they're very stiff and they say, well, you don't understand, but at home I'm much better than this. It's situational. So there's variability. So if we're measuring a neurologic exam, we got to measure it, for example, multiple times, multiple examiners. Okay. Now, what about if a person doesn't sleep? What about if a person's in pain, and so forth and so on? There are situations besides the cold weather that can affect symptoms tremendously. And I just want to say that years and years ago, maybe 15 years ago, I did a study where I looked at very structured neurologic exams. Now I'm actually speaking to other investigators. But I did a study where I looked at people with hereditary spastic paraplegia and I examined them for over each year for 3 years, and I think I had 35 people. And it was a 100-point exam. How strong was the quadriceps muscle? How strong was the right hamstring? What was the spasticity at the left ankle compared to the right ankle? All this kind of stuff. And what I found was that a person would come back and the exam would change. Sometimes the right hamstring would be weaker than the left, and sometimes it would be stronger than the left, and sometimes spasticity distribution would be different, but the person was the same. Their walking, 25-foot walking, 6-minute walking, was the same, even though the exam changed. And I recognize then that the point, this is my opinion, that the neurologic exam is very helpful in describing the distribution of the injury in the nervous system, but was not very helpful because of its variability, and it was not very helpful because of its variability and the fact that variability did not correlate with change in function. It was that the neurologic exam has a role in characterizing the impact of the condition on the nervous system, but was not very helpful in general terms in terms of assessing serial change. It might be helpful if the magnitude of the change was dramatic. If a person was full strength and then they became paralyzed, well, those things would be great. But, I mean, the exam would be very helpful under those circumstances. But within the range that we would see over the course of a year with hereditary spastic paraplegia, it was not helpful. Certainly, validity for any outcome measure is important. That is, validity means the outcome measure is a surrogate marker. The biomarker is a surrogate marker of the condition. It has to, so how well does it correlate with functional change? We heard yesterday a discussion about neurofilament light chain, a chemical that can be measured in blood and is abnormal in many different nerve degenerative conditions. It's abnormal, often, but doesn't necessarily correlate with worsening. So it is a biomarker, but it doesn't correlate with it's not necessarily higher in people that have more advanced symptoms. And it doesn't seem to change necessarily with the chronicity of the condition. So it's a biomarker that there's something wrong in the nervous system, but it doesn't really correlate with symptom change or duration. And then there's the issue about functional relevance. If we're trying to assess function is the impact of the condition on some getting out of a chair, swallowing, doing laundry, doing household activities, driving to work, whatever the function is. Now, personally, I'm more and more focused on functional measurements. We're measuring gait and so forth. But I think that is extremely important because, for example, if a person says as an outcome parameter, well, I think I feel a little more comfortable walking up and down the stairs. You've given me this treatment. Spastic paraplegia rating scale has stair climbing ability as one of its parameters. And so if there's a sensation or a feeling, I feel more comfortable and stable walking up and down the stairs. That's an important patient outcome parameter. Okay. The FDA is going to want to see what was the stair climbing ability. If the outcome of the study was that most people or a lot of people said, I feel more comfortable climbing stairs, we need to, that's good, that's important. I don't want to say that's of no relevance, but what we really need to measure is what was stair climbing ability? Did stair climbing ability actually change? So if someone says, I feel that I'm able to walk better, okay, but we measure the walking and there's no more increase in number of steps per day or number of steps per month or no change in stride length or anything we can measure in walking. If someone says, I feel my walking is better, and we see absolutely no change despite our as intensive of measurements we could say in measuring walking, then we're going to say, well, there was no measurable functional improvement in walking. Okay? No change in the need for a walker or a cane or use of a wheelchair, no fewer falls, so forth and so on. No measurable functional change in walking. And people could say, I feel my walking is better. We would probably conclude that the study did not have a functional impact. And so would we go forward? Would we go further down that path to amplify that drug? Probably not. If we saw no functional change, just only a self-reported symptom improvement, a sense of symptom improvement, we probably would not. Okay, now I'm speaking now to leadership. And I want to comment, what should we do to facilitate clinical trials? What are we doing and what should we do? This is John Fink's opinion. Right now, we are promoting, let me give an example here. In the 80s and 90s, there was basically beginning in the 70s, there was a dramatic, miraculous success in treating childhood leukemia. What was previously a nearly always fatal condition became a treatable, and now childhood leukemia is not one condition, it's many, many conditions. But it went from largely untreatable to tremendous treatment success in the 70s and 80s. And how did that happen? It did not happen by centralized clinical trials. There were centralized clinical trials, but the way that I'm not saying there weren't. There absolutely were many centralized, university-based clinical trials of childhood leukemia. What happened more was that it was crowdsourced. Many oncologists, pediatric oncologists, hematology oncologists, participated by, I have a person with this particular kind of leukemia, I'm starting them on this regimen, I'm going to measure their outcome in various ways, and I'm going to put that information into a database. But it was crowdsourced, and different clinicians were doing different things. And they were learning from each other, and it was all a public database, and it was all analyzed frequently, and you could see trends and so forth. And so when trends were observed, people would go in that direction. And different combinations. It was crowdsourced. So let me just point that out. Second, I want to point out something that people might not realize. But where is the army of 1,000 scientists in the United States studying PLS and HSP? Where is that army? It doesn't exist. There are very few scientists in the United States whose full-time research focus is PLS and HSP. Maybe a dozen. Okay? I'm sorry. That's what it is. They're not enough. We need an army. Okay? This is a big task. HSP is more than 80 conditions. PLS is a half a dozen conditions at least. We don't even know, for most people, maybe for 6% we have ideas in PLS, but most people with PLS, we don't know the cause. So where's the army of scientists and physicians? It doesn't exist. But what does exist are highly trained clinicians who could be drafted into an army. Now, what we're talking about here is having a central, this is a catalyst, or you could say disruptor concept. I'm not saying we shouldn't do centralized clinical trials. They have their place. But what I'm proposing is that we have standardized data collection that any physician, neurologist, physical medicine doctor can be trained to use for data collection on outcome parameters, on gait, on symptoms. We have developed consensus outcome parameters, and then we teach people how to use these, not just people participating in 10 centers, but people's primary physicians are trained in administering these outcome parameters, and data is collected to a central place. And so, what would this do? It would greatly increase participation. But it requires the use of common assessment parameters. Well, these are largely already created and been validated. The primary lateral sclerosis functional rating scale has been developed for years and tested and validated and available, and people can be trained in doing it. It's not complicated. Any neurologist, any physical medicine doctor can be trained in how to administer this. And we can collect data. And so these scales, the functional rating scales, have been collected already and validated. And same with the spastic paraplegia rating scale, that's been consensus designed and validated, used in many studies. It's available. So when we say that the crowdsourcing to draft other people into participating in clinical trials would require the use of standardized assessment parameters, these standardized assessment these have already been created. We don't have to convene a committee to create these. Now, what can the Spastic Paraplegia Foundation do to facilitate clinical trials? Okay, and I recognize that this may be a catalyst. But right now we're facilitating clinical trials in a central, I'm getting the look from Tim that says I'm over time, but just trying to carry on a tradition here. So right now we in the Spastic Paraplegia Foundation are facilitating centralized trial design, which is important, but I don't think it's sufficient. I would propose that we in the Spastic Paraplegia Foundation develop the ability to maintain a portal for data collection and distribution that is not maintained. I mean, it can be HIPAA compliant and so forth. That's not maintained by a single investigator or a committee of investigators, and therefore is not the purview of one team, but is open for everybody who is, anytime you go to see your neurologist or physical medicine doctor, they could collect information and they could upload that to this portal, which would then be ongoing. It could be symptoms, questionnaires, quality of life, functional measurements, gait testing, everything could be at a crowdsource, and people have to be, of course, be trained in these measures, but it could be uploaded to a nonpartisan, by nonpartisan I don't mean political party, but not affiliated with any university's research portfolio. So it would support anyone who has wanted this data. And how would we do this? We'd have to get funding and session training to online and in person for every physician who needs to come up to speed in use of these outcome measure assessments. I think we could do that. I think that would be a goal and a good investment of resources. So, go blue.
Thank you, sir. Yes. Sorry, guys, we're 15 minutes behind. We got lunch coming. We got to go. By the way, Dr. Fink is here, and he's doing his gait study across the room. He's going to be here all day today and all day tomorrow. Make sure you walk up to him and ask him a question. You can do that, okay? He's not going anywhere for a while. I've got his plane tickets. I'm not going to give them back for a couple of more days, okay?
We can just go.
We're going to hope this goes. All right.
I'll just tell them where I am.
All right. Here we go. We're moving right on, guys.
We're going to move. I'm from a small town in the Midwest, and I'm going to be a very different speaker for you today. And so for sake of time, we're going to go through things, but we're going to go through an interpretation version, and you can always have my slides or talk to me after this is done. See if I can get my slides up. Okay. We're going to tie some of these things together today. We're going to talk more about like the regulatory science piece to this. And we're going to update you on some of the work that we're doing with SPF to really create a strategic strategy strategic plan that would be a roadmap to really take into all this information that you all have learned today, but really try to create it into a cohesive plan. There's lots and lots of stuff going on. We're going to add a little bit different lens to this in that there are terms that are called like fit for use or qualified or whatnot. These are things that regulators do to look at scientific evidence to allow into things that would be interpretable for use to decide whether or not a treatment is safe or effective. And I'm going to go through these hopefully really quickly, but give you some cohesive insights into some of the things you've heard, but then update you on the discussions that we had yesterday. Now, you've heard today, preclinical data is really important. You know, when you think about that, why is that important? I want to define a drug target. I want to understand the pathophysiology of the disease so that I can better ameliorate it and change it and be informed. That's very important for this. And as you've heard today, there's lots of discussions around model systems, in vitro work, like omics information. And these information obviously are useful for companies oftentimes or sponsors to identify rapid ways to screen things that are multi-scale. And so you can become a lot more efficient in doing drug discovery. You heard a lot about SB-CERN and clinical trial sites. I'm not going to go into that detail for sake of time. We'll come back to that. Drug development pipeline, I know that's an evolving in many rare diseases, obviously sponsors. Drug development tools, I can't tell you how important those are, because what happens in drug development in rare diseases that's a lot more challenging in compared to like more common diseases is that you may have a company that has one asset. They run the gauntlet for drug approval. If they fail, all that information is lost. They have to create everything from scratch. So they have to go do the regulatory argument, they have to do their statistical plan, they have to argue the regulatory discussion, they have to go through and model the work and do all these things. And if they don't succeed, there's no other resource. That information, you start from pretty much ground zero again. And so the government and it has endorsement pathways, but they're all around the development of what we call drug development tools. So in a sense, if I'm a sponsor, I can go and I can take this off the shelf and say, hey, this has already been negotiated. I can use it. It's useful. It's just like something else, a resource or a tool. And there are formal endorsement pathways for that to occur. And so as you heard throughout today, I mean, we discussed data warehouse, we talked about biomarkers, we'll talk about those in a minute. A little bit more clinical outcome assessments, master protocol, and prioritization framework, and of course funding. Now, why is regulatory consent into finding a cure and to beginning to look at research for this? Because again, like I was an academic researcher, I was a clinical researcher. One of the things that I didn't fully appreciate in my career was I didn't quite understand exactly how regulators thought. And so we're not trained that way. That's not the lens that we speak to. And so when I listen to science, lots of times I have two brains going on. One is looking at, hey, this is cool science. It's a great paper, may get a Lancet article. And then I'm the regulator. And my regulator takes a very different lens to that same scientific discussion. And the rigor and the complexity of that sometimes can be quite different because the way the regulator thinks it can be very different. But the goal of regulatory agencies are always to determine a public health initiative. They want to make sure a drug is safe and effective for what its intended use is. And so that's why regulatory science is important. I'm going to breeze through these, and if people have questions at the end of the day, we can come back to this. I'm going to translate them in a minute. Efficacy is established for a clinical benefit, and that's described by you. Individuals impacted the disease define that framework. We try to assess this in what we call clinical outcomes, and we develop instruments to actually measure that. And there's lots of ones. They're clinically reported, they're patient reported, there's digital health technologies potentially, and then there's biomarkers who really act as an indirect measure, and not necessarily of benefit, but whether or not a drug ameliorates the progression of a particular biomarker, mechanistic pathway or pathological process, et cetera. Now, one of the things that when you look at FDA, they have a series of guidances that are a little bit like instruction manuals to determine, did this clinical outcome assessment designed and instructed to produce something of the scientific rigor that needs to be. And so the first steps of this is really understanding the disease or the condition. Then it's contextualizing things that around, how that should be measured. What are the specific concepts? For example, you heard out today, motor concepts are important, right? Speech concepts are probably important. Cognitive concepts are important. But what that does is it allows that persons with the condition suggest areas that they want to have benefit. Because you can't determine efficacy if you don't know what benefit is, right? You've got to flesh this out. And that measurement is very important. And there's certain psychometric analysis are done. There are things called IRT. There's lots of methodologies that are undergone, and we can talk about that if we have more time someday. But what ends up happening then from that is you assess how well you're measuring those things that are important, and you put that in an instrument. And that instrument, as it's gone through scientific rigor, develops what they call a context of use, which you may have learned today. Sometimes you need to modify that instrument over time. ALS-FRS right now, for example, in ALS right now, we're spending a lot of time revamping that instrument right now. And we have others in like GM1 right now, we're looking at an instrument, developing one de novo in that space. And so, so that's our framework. Biomarkers, I'm going to talk about this just briefly. You heard about these. For drug development, there are some ones that are really more attractive. Pharmacodynamic biomarkers, they're important because they look at dose response. They also look at, did you hit the drug target that you need to hit? There's prognostic ones that say, hey, if I look at the entire population of a disease that, and I try to look at a biomarker, what are the ones that may benefit most from that treatment and related to that biomarker? And then predictive ones really look at sequences of event over time and look at disease progression over time. And so oftentimes those are used and prioritized in drug development because they have utility. And then there's the holy grail of biomarkers, what's called a reasonably likely surrogate endpoint. And why that's important, it's more of a bit of a predictive biomarker, but it's demonstrated to act as a surrogate for clinical outcome assessment. And what that means in the US in regulatory strategy is because you can get accelerated approval. And if those of you aware of the Biogen study for tofersen, NFL was one of the biomarkers that was judged as a reasonably likely surrogate endpoint. Now, context of use really quickly, I mean, when you hear they say that, it's basically saying whatever these instruments are for, what parameters, it's like your indication, like you pick up your drug box right there, or what your instruction manual is, it says this is what this thing can be used for and there's scientific evidence to be used for. And so people use that a lot. And so each of these things that we've talked about in drug development tools really function in that manner. Now, how is that information used? Why are endpoints important? Endpoints are important, and the rigor of the endpoints are important is because anything that's unknown or doesn't measure anything that's meaningful produces risk. It means you try an experiment hoping that you're measuring the right thing to generate a clinical benefit. So it's a really good idea to make sure you're measuring the right things and you understand the performance of that measurement. So for example, there's lots of variability. You need to figure out how to manage that variability, right? Either through training or longer follow-up, increase your sample size. There's lots of ways to mitigate that. And so with rare diseases, oftentimes if we don't look at the natural history study data, we may not understand that. When you look at biomarkers, oftentimes the biomarkers in a regulatory package support things much more in depth in early signal detection. Primary endpoints typically are clinical outcome assessments, but the biomarkers act as supportive evidence to suggest we're working together to move towards the right things. Now I'm going to breeze through data because of time, but I want to say, we talked about data standards. There's a lot of things with harmonization. What I really want to talk about, and we see this happen a lot in different conditions and rare diseases, is that there's a lot of data being collected, but it's not cleaned and QC'd and standardized, and it's heterogenic, and there's stuff that's missing, and it's not in a common data dictionary. And so when you try to look at that, it's like when you go and you spill a whole bunch of stuff on the floor and try to count it. You can't do that. It's not effective. You can't pool it. And so good quality data and having really traceability and documentation is really, really important, because existing data is all of your time, all the resources and all the utility of that. And then, that's probably the area that's most underdeveloped. And let's see, I'm going to go through because we're time. Now, CPATH, we started a partnership with SPF. We've been involved in lots of different rare diseases and common diseases. We were established over 20 years ago, really aimed at creating drug development solutions. We're a neutral convener. We don't come in with particular biases on pretty much anything. And we work with sponsors, academics, foundations, regulators, really to define where they define their needs, like to figure out, okay, what do they need to advance drug development? We have existing staff in all those areas and expertise, and particularly giving in the context of regulatory grade infrastructures. Now this is probably the most important point. Sorry, hold on, everybody, if you're shaking your chairs from that whirlwind tour. I want to talk about a little bit more on what we talked about and what we've been working on with the board. The first one, is looking at project feasibility, like determining what existing efforts we're going there. So we talked a lot to investigators. We did our own research. We worked with the foundation to review things. In fact, looked at grants, mapped out all the industry partners and talk to regulators and begin to really scope, okay, what are people thinking that we need? And then yesterday we held a research roundtable meeting. And the goal of that was to create a democratic process where people could come and say, these are the priority areas that we need to invest in. These are the gaps that we clearly need to define. And so what we listened today is we had speakers come in and explain, okay, this is where we think we are, and then these are the things that we still think are gaps. What's going to happen from this is we're going to generate a report, a roadmap, and we'll get feedback from the community because we want to continue this to be a transparent process. And then, potential recommendations for the board to consider in the next steps of their activities as an organization. Our outreach is completed, data landscape's completed, the grant review, we've looked at grants and whatnot to understand what's been done and what's recently and what's passed. And then we're actually trying to work on determining what data is out there and what harmonization processes will be and clean that data and try to do QC on it if folks will let us, and we'll give it back to them so that it's in better forms and standardization in ways that can be used by industry and regulators in a more cohesive way. And then again, we'll talk through the roadmap. What I want to talk about, we'll go through since we're almost at time. This is what they wrote. Everybody can have my slides. I want to update on what we heard. This is slides I made this morning, so it's not complete. I haven't gone through all my notes again. And people, I believe that was videoed, so people can probably get that recording. Greg, don't shoot me if I said that wrong. Mechanistically, I think what we heard is there was a need for broad omics discovery and understanding on model systems. That was a clear message that we heard. But also, taking AI to begin to look at existing data. That's going to require a data warehouse to begin to explore that and to identify both targets but pathways. And there are things called ontologies that can be done also to link things that are in shared diseases. So Mondo ontologies. It wouldn't be just information within SPF or PLS. There's a lot of information that can be compared to other diseases to find pathways of disease, but also potentially treatments that would be there. Clinical trial sites, I think you've heard this from SP CERN today. I think they're doing an amazing job of trying to create clinical trial infrastructure to support the community. I think they're all well on their way. I think the natural history and biospecimen imaging are complete. What we'd like to really understand is the quality of the data that's being generated. We see for certain modalities, for example, the missingness or whether or not the context of use that you're potentially planning with the data needs to have a lens. I work with a lot of foundations where they're collecting information, but sometimes it's not in a way that answers the question ultimately that would be suitable for drug development. I'd like to understand that more. I love the fact that they're looking at junior investigator training as part of that and applaud that as part of their award, and also applaud NIH for funding that because it's critically needed. Drug development pipeline, we heard specifically about 2 assets yesterday. Unfortunately, we're still really limited here, and I think there were discussions about the need for gene-specific drugs and then genotype-independent strategies, so independent of genotype. And what that means, and one of the things that we'll be looking at more closely is that for common mechanisms of disease, you'll have right here close proximity to the pathogenesis, middle, and then early, probably a high level. Most traditional therapies are really up here. They're nonspecific. Gene therapies are right on the button. I think what we'll need to start considering is where do we go here at the mid-level and the higher, because those might be more likely to be the quick win, particularly for drug repurposing, which was also mentioned at the discussion yesterday. And we have funded one investigator at CPATH working on some preclinical work right now and whatnot. And we hope to mentor and continue to support that program and whatnot. Drug development tools, I think there's a acute need for summarizing lots of this data. There's a wealth of data that's floating out there in the SPF and PLS reader system. I think for HSP, we found 30 data sets, I think, that were out there, and about half of that-ish PLS. And so we've actually made outreach to those people to look at data sharing. We've sent agreements. Our data platform is funded by Food and Drug Administration, and it specializes in rare diseases. And that allows it to be shared broadly with folks, but also based on the permission of the owner, but also for regulators to begin to look at it. Because oftentimes regulators are trying to understand, okay, when they get a sponsor coming in, they want to look at that information to start understanding how they should respond to those questions, particularly for diseases for which there's not been an approved therapy. That's an important point. When you're first, it's hard. And so having that information shared so that it can be interpreted and people work together is really, really important. Biomarkers, I think that one thing that obviously we heard a little bit NFL. I think there was some pros and cons there. And then I think that there's some discussions around more genotype-specific biomarkers, and I won't go into those specifically. I think there was an idea that we needed biomarkers of multiple types. I think pharmacodynamic were ones that were one of the things that were prioritized, but also enrichment biomarkers or prognostic biomarkers were there. Digital health technologies, when we talked about them, and you heard this, and Dr. Fink's actually working on this right now, is looking at motor and as one of the primaries, because obviously it's pretty broad spanning for both HSP and PLS. And then, but not to forget, is that speech and cognition we need to look at. One of the things that is really important when you do DHT work is that you're collecting lots of information. And that's where you all come involved, because one of the challenges that we've seen in working in other rare diseases right now with digital health technologies is that the clinical meaningfulness hasn't been defined. Because you're collecting after information. So if I do this, is that meaningful? If I walk, is that meaningful? How do I measure that? Because I'm tracking all the sensor information. In fact, it's like, I think we just talked to a natural history study that told us they had 30 terabytes of data for one disease. That's a lot of data. And so you have to learn to process that and identify it, but also linking it to clinical meaningfulness. DMD for Duchenne muscular dystrophy, they just recently went through some approvals around stride velocity and looking at how gait, but that relies on the sensor technology that you're using, the assessment frequency, defining clinical meaningfulness, the validation steps. All those things are really needed to be useful for regulatory decision making. And so even though you're collecting data, it's got to be focused and aimed at the right things. Clinical outcome assessments, I think we heard the standard batteries defined. We went a little bit into the guidance, for sake of time, we abbreviated that. I think we need to take a little bit finer tuned comb there. We also need to look at the clinical input of that and whether or not how involved persons with lived experience were identifying the concepts, because it's really hard to get a drug approved if your target isn't measuring things that are exactly meaningful and measurable to the clinical population. I think that I'm excited about what I heard, but I want a deeper dive there. And then master protocol. I think we're pointed in that direction. I think where we need to do work, probably a working group to really look at this, but also take data to start simulating clinical trials. So before we enroll people, we start looking at what does this trial really need to look like? Like, how fast does this progression happen? What is the effect size? How much difference should we expect? And those take parameters both in data simulation, but also take in your vote, because we need to understand that as well and part of that. Next steps, and we'll keep everybody updated on this, and sorry I rushed these things for sake of time and such, but I wanted to get through all the talk, and hopefully it'll give me 5 minutes for questions. But really to take this and put it into a report. And we want to make this, and we're thankful for the foundation. Like I said, we came here, we've worked in a lot of diseases. We've been very fortunate as an organization to be involved in first drug approvals for many indications, and both rare and non-rare. But it was creation of regulatory tools that help them advance things. The data that we've used as a platform is being used for alternative controls for sponsors. And we've had things that we're sponsors for simulation tools that have been approved for FDA and EMA. We have biomarkers. I mean, there's lots of things now. It's fortunate that where that industry, back to the industry example, anybody from industry and any of these can take these and use them free. They're usable. I don't have to start from scratch. I build on the science that came before me. And I think that's the goal of what we all want to do here, because we don't want to restart everything every time we do this. And time is precious, and wasting that is not on anybody's minds right now. Sorry for the really talking fast. I think that caught us about 2/3 way up. And Raul will take questions, and I apologize for the rapid manic talk. Any questions real quickly? If not, yes. Yeah. How are we going to look at, I think if I heard the question correctly, it says, how are we going to assess safety? Is that what I heard? And then monitor safety? There's a couple things. As an aside, one of the programs that we're launching right now is called 1 to Millions, where we're taking preclinical data and beginning to look at for things like gene therapies, including like vector, deliverable, everything, but then looking at predictive safety modeling. Because we hear things like ASO backbones have issues, obviously AV and AVV vectors have their own challenges, but what we want to do is predict that before we get to humans. Second part of that is that for those that view that that are in gene therapy studies or have considered them, there's a long period of follow-up that occurs as part of those. There's what they call a post-marketing requirement. People like me who are SGs for FDA get on these advisory committees, and then we review that information to see if there's a safety signal for determinations. And then they'll make recommendations from that. But there is a requirement typically for those products to be followed up long term. And so it's important, for individuals that participate in those to have that long-term follow-up. The limitation of those studies is that they are smaller sample size and whatnot. And so we lean on the risk-benefit of those trials to be big effect and then a little bit more tolerability when it comes to safety. And people will continue to monitor those to make sure that there needs to be some mitigation happening from that.
But if the long term, if they're followed up long term, I assume at that point, if there is any harm, the harm has already occurred. So how are you going to assess harm before you enroll patients?
You can. Usually it's through preclinical studies, and then experience. For sometimes it can be the experience with the delivery technology, are the two things. Like with viral vectors, I think there's two parts of this, and I apologize if I'm getting too much to the weeds, and people can tell me to shut up if they need to. But one of the things that when you're doing with viral or antisense oligonucleotides, they're characteristics of the delivery system themselves that you're having to look at the safety of. Typically, when molecules like gene therapies go into studies, they're unlike other molecules. There's additional batteries of tests. Sometimes, things as level as non-human primates, for example, or that, and which makes drug development sometimes very expensive for gene therapies. The other part is that when you're looking at these, there's extensive batteries and what they call not on-target and off-target toxicities. And then part of those off-target toxicities specifically is inappropriate insertion of the gene into where it shouldn't go. However, that's usually now, the technologies continue to evolve quite well. And from my understanding and talking to lots of different advanced researchers in those spaces, the specificity is really good. That doesn't, usually what you'll see on the news, and this is where you're probably going to see things, is sometimes you'll see blips in like hepatic toxicities or in rare cases cancers or things like that. But that's far between. But again, it just also supports that people are looking at that and not letting that just assume that that's okay. Anything, Bueller? Sorry for the, okay. I'm going to pass the baton to Srikanth, who's in addition to, he's the important person to speak today. He's a program officer at NNDS and is a good friend, and he is very passionate about HSP. Actually, the discussion we had yesterday was something he and I, because we work in other disease states, had talked about before, changes that happened within the government, but we had planned to try to compare notes about how preclinical science and regulatory science could mirror to create a cohesive strategy. And that was just about the time that right before SP CERN was a brainchild for the early submission. And so we're very fortunate to be good colleagues, and he's going to give you some more insight to things that I don't understand. No, good. Thanks, Colin.
So good morning, all. I'm supposed to check this clicker. Okay. All right. The only change to what Collin said, thanks, Collin, for the intro, is I'm not the most important person. Collin is, and all of you are. So thank you for being here. And I'll start off by saying thanks to Greg, Norma, the entire community to have me here, to invite me here. This is my first time here. This is definitely not going to be my last time. I will definitely be here because I think it's important. One of the points that Dr. Fink said, I'm here to learn, and you guys are the teachers for us. So I would like to have those conversations, learn more from you all, from all the researchers, from all the clinicians, because that's the only way we can build for the future and build a community, an ecosystem for HSP, for PLS. And I think I want to start off with an optimistic point of view, especially with everything that we heard yesterday, the healthy discussions on what we know already and how much we know already and how to build up from that, the convergence from other closely related neuromuscular diseases and other neurodegenerative diseases, what we can learn from those to build a forte for HSP and PLS. So appreciate everyone who participated in those discussions last afternoon and the continued discussions today and tomorrow also. I do want to also say that the field is clearly moving forward because we are constantly, if there are a few key terminologies or key words that you want to say have been oft repeated over the last 24 hours are biomarkers, clinical trials, gene therapies, outcome measures. So it's, and one of the other important points that I want to reiterate from Dr. Fink's talk is how do we build the next cohort of researchers and clinicians to not just the next cohort, but also expand that cohort and have them excited to work on HSP and PLS. I mean, they can work on other closely related diseases because I think we learn a lot from the convergence of biology from various other diseases. So, but we do want to emphasize the fact that we need more trainees and fellows in the field going forward. So that was my preamble, and I'll try to catch up on time. Most of these slides have all the information there. It's public information for the most part. I do not have anything to disclose, and if I come out saying something or opinionate on something, that's purely my opinions and nothing reflects off that of NIH, opinions-wise. But information-wise, it's all NIH information. NIND is information. NINDS is the National Institute for Neurological Disorders and Stroke. And I've told Norma and Greg, I will share the slides, but I need to get the approval from downtown before that. So, but in time, I will share the slides. So I'll try to catch up on time. I'm not going to repeat a lot of things that are on the slides. To start with, I've put on frequent miles on my scientific journey, obviously I come from India originally, basic education. Then a few decades back, I moved to this country for grad school, came to Georgia Tech, go Yellow Jackets, and took my journey up towards the north, came to Baltimore, came to start my journey into learning the neuroscience and the nervous system by working in a brain tumor lab before I started my PhD on ALS biomarkers and ALS molecular mechanisms at Pittsburgh, then moved on to postdoctoral work on polyglutamine diseases, Kennedy's disease with Dr. Fishbeck. Then decided that I needed to travel to Europe and see some places. So I went to Sweden for my second postdoctoral fellowship, working on Huntington's and Parkinson's disease. Then I got the opportunity to work as a chief scientific advisor for a biotech firm in southern Sweden. And all of this led me back to NIH about 10 plus years ago. I started with the Center for Scientific Review as a scientific review officer, which is the first phase of peer review that happens to every application that comes into NIH through grants.gov. And then about 3+ years back, I joined NINDS. I'm so glad to be doing what I'm doing now, which is I oversee a grant portfolio that involves HSP along with ataxias and other rare neuromuscular diseases. So I am in the rare space, but sorry. I guess that will go away. So in any case, I'll keep moving forward. I just want to, I'm sure you all know, technical question. Could you just remove the, oh, it went, never mind. So NIH is basically there are 27 institutes, and right at the bottom you'll see there are 3 that are boxed. Those like the Center for Scientific Review, Center for Information Technology, and the NIH Clinical Center, they do not have any funding authority, but the rest of the institutes have funding authority. And I'm just circling the 2 institutes that I've been associated with, grateful for being associated with them over the last 10+ years. So I just want to reiterate the mission statement for NINDS. It is to seek fundamental knowledge about the brain and the nervous system, and to use that knowledge to reduce the burden of neurological diseases for all people. And it's 2 things that, 2 pointers here is NINDS mission includes fundamental neuroscience, understanding fundamental biology of the nervous system, but at the same time to help reduce the burden of neurological diseases. So again, all of you are well aware that as Dr. Walter Koroshetz transitioned from over a decade of excellent stewardship and leadership at NINDS and to the broader neuroscience enterprise, he has definitely left behind a strong foundation on which we are going to survive as a community. And importantly, I want to say that the transition has been seamless with Dr. Amy Bani Adams being the acting director now for NINDS. And it reflects the strength and continuity of the Institute's leadership over time. And this also reflects its enduring commitment to the neuroscience community. I just want to put forth the divisional leadership at NINDS. I do believe it's nice to have faces to names. I'm sure you have heard names of all the individuals at NINDS, sometimes it's good to see their faces too. I'm within the Division of Neuroscience and under the leadership of Dr. Patrick Frost-Belgovan. And then you have the Division of Translational Neuroscience, Division of Clinical Neuroscience, and then Division of Extramural Activities. I just want to quickly point out that between 2015 and 2024, NINDS, in addition to many of the other neurological diseases, did support HSP research. And the interesting thing is NINDS is probably one of the only institutes at NIH which is 100% neuroscience driven or with neuroscience commitment. And as you can see, about 62% of the funds during that time period came from NINDS support. And the other word cluster that you see indicates all the different areas, many of the fundamental neurobiology in the space that's been supported by NINDS. I do want to highlight three of the projects that were recently supported from the FY26 budget. Dr. Stefan Zischner from University of Miami, whose R01, which is a research project grant, that's in the 15th year, and he has been studying the exome genomics and exome analysis in HSP in collaboration with Dr. Rebecca Shuley and Dr. Shuei-Jun Lee, who's here. She just got an award for, it's called the Bridge Award for her project that is related to axonal degeneration in SPG 11 and 15. Correct me if I'm stating anything wrong, Shuei. And then we have Peter Bass and Oscar who have been studying SPG4 and spastin biology for a long time, and they have made a lot of inroads from moving from disease biology to therapeutic standpoint. So excited to have that notice of award go out actually last week, I believe, and their study related to testing a new therapeutic strategy for SPG4. They know that science better than I do. I just know the titles. So with the current landscape, as you all know, there's been a lot of different aspects that have moved over the last 15 to 16 months. And I just want to mention that NIH continues to move from opportunity to impact, but through some tweaks and what is called the unified funding strategy. I guess in the federal government at NIH, we love acronyms. We really like all the acronyms. So you'll see a lot of it here. I'll try to make it clear. NOFO is Notice of Funding Opportunity, and UFS is Unified Funding Strategy. So it's pretty much a funnel where we get a lot of number of comparative applications, and we are glad that there is competition because that's one way to move the science forward. That's one way to move knowledge forward. So while the funnel literally takes so many applications and then there are very few awards made, it also means that there is a lot of diligence being done at so many different levels, at the peer review level, and programmatically deciding which projects are getting prioritized over others. There's a lot of diligence that happens behind the scenes before the notice of award goes out to the investigators. And as a program director or as an NIH employee, one of my principal objectives is to reach out to investigators, reach out to trainees, and I'm always open to conversations. Some of you who have interacted with me probably know this. I always say that if you all are not successful, I will not have a job in terms of having to fund all those or having to recommend awards to all that work, good work that's being done. So we want you to be successful in the research projects that you take on. And again, I want to reiterate one point here, which is we want to also develop that next cohort of researchers, the trainees and the fellows. So it is important to move. That's one way to move the field forward. So we have had a few challenges, and one of the challenges over the last few months has been consolidation of notice of funding opportunities. There have been a reduction in the number of funding opportunities, but there are new ones coming up. That's one way to move forward, which is evolve in any landscape that you're in, evolve to make something better. So currently, the total number of notice of funding opportunities that NINDS participates is a total of 94, of which 62 of them are in the research and development space, and 32 of them are in the training and career workforce development space. And of that, 32 are NINDS issued notice of funding opportunities. What the difference is that on the top, of the 94, if NINDS is participating, it means that there are other institutes. Remember, there are about 24 other institutes. Those institutes might be the primary authors on those NOFOs, and NINDS is one of the participating institutes, whereas the bottom number 32 means that NINDS is the primary author, whereas other institutes can participate on those notice of funding opportunities. The other thing that came up recently is highlighted topics. All that means is an institute is interested in that topic. For instance, NINDS participates on 9 highlighted topics, of which 5 of them are NINDS issued, and each as it says here, each highlighted topic pertains to a selected research priority area. What I mean by that is, as you can see here, some of those topics are related to fundamental neuroscience, increase the number of projects and research in the fundamental space. So NINDS is interested in that area. And you have the brain initiatives the other one is the neural exposome function. That is neural exposomes. That's a whole different topic. I don't want to spend time talking about it, but it's related to if there are any pollutants or environmental exposures that can lead to neurological diseases. That's a different science there. So all I want to say is bring to your awareness is NINDS is interested in certain topics. Fundamental neuroscience is one of them. The other important thing that came about is not that NIH was not taking care of the data stewardship and data management sharing plans before, but there is increased increased importance provided to data sharing. For each project, there should be a clear data sharing management plan. And we make sure that there is clear information on what are you sharing, what data are you getting from your project, where do you plan to share the data or deposit the data, when are you planning the timeline of sharing, meaning that you cannot finish all the projects and then share it, or after 10 publications, you cannot put it in a public space. So it has to be timeline driven, and we are here as programmatic staff to guide researchers how to manage or how to talk about it. And so always reach out to program staff at any point in time as researchers. And I just want to bring to your attention some of the other active programs from the Division of Translational Research and Division of Clinical Research. For example, something that's very relevant to this community, which is the ultra-rare gene therapy network. These are active programs, and at the end, I also mentioned the program director, one of my colleagues, who is the lead for this program. And again, for this community, I do not want to, I don't need to spend time talking about the rarity and the ultra-rare scenarios of neurological diseases and how important it is to take it forward. But all I want to say is, there are opportunities within this program, within this network. Similarly, there is it's called the IGNITE. I told you we love acronyms. IGNITE actually stands for Innovation. I never remember this, so I wrote it down there. Innovation Grants to Nurture Initial Translational Efforts, IGNITE. So that is the number that you see there, R61, R33. They are just grant mechanisms by which we know what it is and researchers know what it is, what kind of mechanism it is. It's a phased, milestone-driven research grant for, you get funding for about 3 years. And you can try to, you have different flavors. You can maybe assay development or validation of an assay, validation of an animal model or other models, organoids, for example, that Oscar has been working on in terms of organoids for the SPG4. So these are the important things that I want to bring forth to you. The other program is the Blueprint Neurotherapeutics Network, or BPN, and this is for the biologics, and it includes peptides, antibodies, oligos, gene therapies, cell therapies, all of these. Again, there is support, there is active, these are again active notice of funding opportunities. As you can see there, down there, the next anticipated due date is July 15th, which is 1 month from now. This is from the Division of Clinical Research. This is one of the clinical trial NOFOs that is active. It's called Network for Excellence in Neuroscience, also known as NeuroNext, NeuroNext clinical trial program. I'm zipping through these slides because it's all busy slides. This is the other thing that I learned from Dr. Fink's talk today. He just had catalyst/disruptor, but he spoke about that slide for a few minutes. It's really nice to see not busy slides, unlike mine, and actually have a lot of information out of it. So something I learned today. So the next one, we heard a lot about this from Darius. All of you are aware of the SP CERN that's coming up in the next slide or two. But the RDCRC is Rare Disease Center for Research Consortiums. And they are, again, forget the U54s, U01s. I mean, there's so many of those. So they are just a grant mechanism. But as you can see, the RDCRN that's led by NCATS. NCATS is the other institute, National Center for Advanced Translational Sciences. And NINDS participates in this particular program. And this is primarily to move the rare disease space forward towards a clinical trial readiness or even a therapy stage. So we heard a couple of them yesterday, or a couple of the awardees. We heard Darius. It's a multi-PI a grant that was awarded on September 25th of last year, I think. I was super happy when that notice of award went out just before the October 1st deadline. So that's, again, a excellent team of Darius, Stefan, and Josh from St. Jude's. And you heard from Darius, so I don't need to say much about that. The other consortium that is in the second cycle of funding is CREATE Consortium. That's led by Dr. Benneter, who was on the call yesterday. And that is, again, for looking at ALS and related disorders. Again, Dr. Benneter and Darius will be able to talk more about the details of their studies than I can. So the other thing that I've been involved with is the Undiagnosed Diseases Network, or UDN, and that is in its third phase. When I say phase, that's each phase is about 5 years. So it's in its third phase that is coming up to an end of its 15th year. And now we are in the process of planning for phase 4. So those conversations are ongoing. So I recently, over the last 6 months or so, got associated with this program and leading a couple of those projects. So next one, I'm sure all of you have read in the news about baby KJ that came out of this particular program, the SCGE, or the somatic cell gene editing. There's a lot of focus on genome editing and base editing that's happening in for different diseases. And they have moved from phase 1, which was the technology development, to the therapy development, and again, baby KJ. That's all I have to say. So there are a few other initiatives that are led and supported by NINDS. One of them is the Bespoke Gene Therapy Consortium. I believe Terry from the SPG50, Cure SPG50, has been part of this BGTC over the years. And again, that is a program that's led by NCATS. And the other one I want to bring to attention, especially to the trainees and the fellows, is the High Risk, High Reward program from the NIH Director's Office. And this has been running for several years now. You have different awards. There are 4 different programs. One is called the DP2, or the New Innovator Award, for instance. That's for early stage investigators, only for early stage investigators. And these projects need to be high risk, high reward, which is you can think out of the box, come out with wild ideas, not too wild, somewhat practical too, and you can propose something that is highly innovative and impactful. And impact should really not just be near-term impact, but long-term impact. So there are these other what we call HR, or high-risk, high-reward programs. I'm almost done. I'll zip through the next few, but the next thing is the public-private partnerships. This, again, I've been fortunate, even as I started at NINDS, I was fortunate to be part of the AMP ALS. AMP is Accelerating Medicines Partnership in ALS, and we have wonderful partnerships with Critical Path and FDA, and also other private industry partners, nonprofit organizations, and this is to develop therapies for ALS. And this public-private partnership came out of the Act for ALS that was passed. That bill was passed in 2021. So again, I don't want to go into the details, but I just want to put forth the objectives of this AMP ALS, which is if I want to simplify it, it is for biomarker development, data portals, which I do want to emphasize that it's important to have data repositories, and having a lot of the data sets that Critical Path, for instance, has been involved in over the years in different disease paradigms, or even Sage Bionetworks. It's nice to have federated data, and data sharing becomes very critical if you want to move forward a rare disease space. The third arm is the clinical outcome assessments, and again, Critical Path's expertise in making things regulatory ready, and their interactions with the FDA is very beneficial to this program. And this is the other thing. These AMP programs are managed by foundations for NIH or FNIH. They are the ones, they are again a nonprofit organization that works closely with NIH, and they are the ones who manage this. And again, these are the different things that have happened. For instance, we launched the ALS Knowledge Portal in August of 2025, and we have more than 25 data sets, both molecular data sets and clinical data sets. The clinical data sets are ingested and curated and harmonized by Critical Path, whereas the molecular data sets are done by Sage Bionetworks. The other AMP program that I recently got involved in is the PDRD, which is Parkinson's disease-related disorders. Again, this is the second phase. They already finished the AMP PD, and now it's moved to AMP PDRD, related disorders like Lewy body dementia or Parkinson's related dementia. So anything related to Parkinson's. Again, this is another program that is fast moving in that public-private partnership space. A shout out to NINDS funded biorepositories. This is called BioSend. Any study, I mean, there are plenty of studies that are being deposited, the samples, any biosamples. As you can see on the left, all the different varieties of samples that are deposited in this bank, and this is held by Indiana University. NINDS has funded Indiana University to have these huge freezers to hold these samples. And you can see that all the different studies, for instance, all ALS and the ataxia studies and Lewy body dementia, and so there are plenty of studies that is being banked here in the biorepository. The important thing is these biosamples are available to any researcher. It goes through a request process. You have to outline to us exactly why you want what you want and how much do you want, and then you will get the aliquots to do the research. So it is, as one of the shout outs here is two things, if I want to say, share data and share samples. We are happy to have them banked and then have it distributed to the researchers again. So what comes in is readily available for the 90 seconds. So the next one is the Brain Donor Project, or the NIH Neurobiobank, and this again funded through NINDS. And I don't want to say much about it, but it's an active brain donor bank or program. Additional information and announcements, I just want to do the prop here. There is a nonprofit forum, NINDS. Every year we organize this nonprofit forum wherein any nonprofit organization can come and interact with the NINDS folks. And so It is scheduled for August 14th. So SPF, I hope SPF will be there. It's going to be in Bethesda, Maryland. Whoever is interested, I can talk about it. And some of the other things, there is 2026 Brain Initiative Conference coming up, the NINDS Small Business Program, the NOFO. By now, even if I do a pop quiz here, everybody can tell what NOFO stands for. So the SBIR STTR Notice of Funding Opportunity just went out. It just got published. And then a few other things. There are some requests for information that's, or what we call RFI. And there will be other RFIs that will come out. So I just want to make sure that the community is able to respond to that RFIs. We need responses to such requests for informations. In the near term, I just want to say we have the A to H covered here, and add P to it. So A, B, C, D, E, F, G, H is acceleration, biomarkers, collaboration. We have heard all of that in the last 24 hours. You can use the C, you can either say collaboration or clinical trials. Either ways is fine. Data sharing, endpoints, funding, well, that's funding. And then gene therapies. So with that, I just want to say that we have to work in a collaborative space, everybody coming together, but most importantly, the center of it all is the PWLEs. PWLEs are the people with lived experiences. You all, the caregivers, we really want you to help us through this collaborative process to reach the milestone. Thank you all for listening. If I take a second to have a quick shout out to my colleague who helped me through every process of what I spoke, that's Dr. Tao Sun, who's sitting right back there. She's a health program specialist and scientific and operations manager at NINDS, and thanks to her too. Thank you.
Wow, the room got real quiet. You guys are doing all right. Hey, a couple of quick notes. There's going to be a box lunch served to you at your table at noon, and also bottled water is going to be provided. But there is also some tea and coffee available for you too, if you want to do that. But I want to let you know this is going to be a working lunch. We have a presenter at lunchtime. And another note, raffle tickets are still available, and you have to remember they must be in the little bowls by 6:15. Okay, if you buy a bunch of them, make sure you put them where you want to put them. Okay, by 6:15. That's very important. Okay, moving right along. This morning we explored scientific discovery, therapeutic development, regulatory readiness, and national research priorities. Those efforts are essential, but they represent only part of the journey. While researchers work to build tomorrow's therapies, each of us still faces the challenge of living well today. Health is not only about what happens in laboratories and clinics, it's also about resilience, purpose, motivation, and the daily choices we make to maximize our quality of life. Our next speaker reminds us that hope is not simply something we wait for. It is something we actively build. Javier Tuel has inspired countless individuals through his message of perseverance, personal responsibility, and living life fully despite challenges. Ladies and gentlemen, Javier Tuel. We're going to make work? There we go. Okay, put your notes here. Take this out of here. This, we got this. Okay, and then let me check this here. This is going to be for you. When you're ready for this later, you just flip up this switch. Okay, got it? And we'll put this over here on this chair. 1, 2, 1, 2. You're on. All right, if you can't hear me, just tell me, okay? One of my things is I have a very light voice. Good morning, everyone. I don't believe people are broken. I believe people adapt. I believe human beings are far stronger and smarter and more resilient than they give themselves credit for. And I believe movement is one of the greatest reminders of that, not because movement has to look perfect and not because everyone in this room moves it the same, but because movement means you're still participating in life. Too often, fitness gets presented as something that only belongs to athletes, the people working for the perfect body, or for those without challenges. I completely disagree. Movement belongs to everybody. It belongs to a person lifting weights. It belongs to a person relearning balance. It belongs to a person using mobility aids. It belongs to a person who simply decided not to quit today. This belief became the foundation of Believe Stronger. My name is Javier Tuel. I'm 47 years old. I'm the co-founder and co-owner of Toolshed Training, and before personal training, I worked in human services. Over time, I realized something important. People don't just need information, they need connection. They need encouragement. They need someone to help them see possibility again. About 12 years ago, I was diagnosed with early onsets of Parkinson's. And honestly, that diagnosis changed the direction of my life in a powerful way. Not because I saw myself as limited, not because I stopped believing in movement, but actually the opposite. It made me realize how important movement truly is, not just for fitness, not just for appearance, but for confidence, independence, quality of life, and identity. The truth is, movement is deeply connected to how we experience life itself. The ability to enter a room confidently, to move as independently as possible, to participate with family, to travel, to work, to laugh, and to feel capable. Those things matter. Before I move on, I want to share something important. I am a son, I am a brother, a father, and many other things. My diagnosis did not define me and did not erase the person I was before it. The same is true for each one of you. Long before a diagnosis entered your life, you had hopes, passions, relationships, talents, and dreams. You had roles that mattered to your family, your friends, your community, and to yourself. Those parts of you still exist. And to the loved ones, caregivers, this message is for you too. Before your loved one was diagnosed, you had your own identity, your own goals, your own life story. Caring for someone is an act of love and dedication, but it should not come at a cost of losing yourself. You're more than a caregiver. You're a spouse, a parent, a sibling, a friend, a professional, an artist, a dreamer, whatever makes you uniquely you. Hold on to those parts of yourself. A diagnosis may change many things, but it does not take away who you are. Remember that you're more than your illness, more than your challenges, more than the role you have been asked to carry. Never forget the person you were before and the person you still are today. What I started noticing was this. Too many people with movement disorders, neurological conditions, or cognitive challenges are being left out of mainstream fitness conversations. Not intentionally, but realistically. And too many people started believing maybe fitness just isn't for me anymore. Movement belongs to everyone. This became the core belief of the Believe Stronger philosophy. Believe Stronger is not pretending life is easy. It is about believing stronger than the obstacles, stronger than fear, stronger than frustration, stronger than the voice that says your best days are behind you. One thing I've learned over years of training is this: strength looks different for everybody. For one person, strength may mean deadlifting 400 pounds. For another, it may mean making movements more confidently. For another, it may mean trying again after a difficult week. Every version of strength matters. We know conditions like HSP can change movement patterns over time. Statistically, it tells us that many individuals may eventually need mobility aids or additional support. But needing support does not mean someone stops living, and it definitely does not mean someone stops moving. Inactivity, on the other hand, creates problems: faster physical decline, loss of balance, reduced confidence, isolation, depression, increased health complications. But movement changes everything. Movement improves circulation, cognition, mood, confidence, energy, and quality of life. Movement is medicine, and movement enhances medicine. Please don't ever forget that.
When I say movement, I want to be very clear. I'm not talking about perfection. I am talking about not turning everybody into elite athletes. I'm talking about helping people maintain and improve daily life. Can someone stand more confidently? Can they move their body safer? Can they maintain independence longer? Can they reduce fear while moving? Can they reconnect with confidence again? Those victories deeply matter. At Toolshed Training, one of the biggest things we focus on is adaptation. Movement is adaptable, inclusive, and essential. Sometimes adaptation means changing the range of motion. Sometimes it means seated exercises. Sometimes it means slowing down. And sometimes it means modifying balances and challenges. Sometimes it simply means creating an environment where people stop feeling judged. Because when people feel safe, they participate more, and participation changes lives. What I've noticed in working in the fitness industry was that neurological population is this. People often carry more mentally than physically. Fear of decline, fear of falling, fear of embarrassment, fear of becoming dependent. But movement can help interrupt that fear, not because it removes every challenge, but because progress creates confidence, and confidence changes how people live. One of my favorite things to witness is when somebody begins believing in themselves again. Maybe at first they're hesitant, quiet, unsure. Then little by little, something shifts. Their posture changes. Their attitude changes. Their energy changes. Eventually, they stop focusing on only what they lost and start focusing on what they can still build. And that's powerful. One of the greatest mistakes a society makes consistently is defining people by the limitations instead of possibilities. I refuse to do that. I focus on capability, and I hope you do too. Maybe capability looks different than it did 20 years ago. Maybe progress happens slower. Maybe adaptation is required. That is okay. Different does not mean defeated. I always tell clients, progress over perfection. Perfection is exhausting and unrealistic, but progress builds momentum. Small progress matters, an extra step matters, a hand or arm gesture matters. Improved balance matters, getting stronger matters, slowing symptoms matters, showing up matters, and trying again matters. Over time, small victories create major change. And last year, I presented a goal, outlining a goal and steps I plan to take to achieve it. The goal was to be able to cut my food with my knife. I have a real issue with my left hand. It's hard for me to stick a steak and cut. And one thing I noticed last year was some of my friends wanted to help me cut my food. So I wanted to change that. My goal wasn't just about cutting my own food. It was about building the confidence to dine out in public without limitations or limiting my choices. Over the past year, I've made real progress. While it's not perfect, yet I continue to work intentionally towards it. A big part of my progress came from attending this conference last year. To be honest, I was scared. I wasn't just worried about the trip itself. Over the years, I had greatly limited my travel because I didn't think I could handle it. I was also afraid of something many people take for granted. I was afraid to eat in front of others. And then something happened. As I sat in the restaurant, I looked around and saw people who, like me, were dealing with limitations and challenges. But what stood out to me wasn't what they couldn't do. It was what they were doing. They were living. They were socializing. They were enjoying themselves in a new place. Whether they felt fear or not, they weren't letting it stop them. For the first time in a long time, I realized I didn't want fear to keep deciding what my life would look like. The experience inspired me to work harder, not just toward my health goals, but to reclaiming my life. Don't get me wrong, I still have difficult days. There are still times when things don't go as planned, and there are moments when fear tries to creep back in. But when that happens, I remind myself, you can do this. I refuse to be the reason I don't do things I'm still capable of doing. The lesson changed me, and I will always be grateful for it. I am grateful for this conference. I am grateful for the people in that restaurant who may never know the impact they had on me. I am grateful for every person sitting in these seats today, because your courage, your presence, and your willingness to keep showing up may be changing someone's life in ways that you don't even realize. This year, I had to reboot after dealing with shingles, which set me back temporarily. Even so, I made strides, and I'm proud of progress I've maintained. Shingles is the real deal. But you know what? I got over it. It's a reminder that growth isn't about perfection. It's about persistence, self-compassion, and the willingness to keep moving forward. And my goal in the battle this year is to get my left arm and left leg to move again with the rest of my body. So this morning, I saw everyone at the gym, right? Right. So this morning when I was at the gym, I'm lifting heavy weights because I can still lift some heavy weights. But then I get on the treadmill and I have to go like 0.2 and just learn how to do this again. It's something. And I think about people in the gym for a second. I wonder what they think about me. But what I understand, it doesn't matter what people think about me. It matters what I'm doing and not limiting myself. And I hope you believe that too. So what is the goal you're working towards? What steps do you need to take? This is just not for somebody with HSP, it's for everyone. For example, making your bed, more movement, or just a better outlook on life. Community is also critical. No one should feel like they're fighting alone. Conferences like this allow people to be surrounded by others who understand the frustrating days, the unexpected days, the difficult moments, and the victories that others may overlook. That connection matters. Encouragement matters. Support matters, and belief matters. Sometimes before people believe in themselves fully, they need someone else to believe in themselves. This is why I believe Stronger exists, not simply as a workout philosophy or a business slogan, but a reminder that people still matter, that growth is still possible, and that movement still belongs to them. I truly believe that movement changes lives, not because it solves everything, but because it reconnects peoples and possibility. And possibility changes everything. I also want to be realistic. There will be some difficult days, frustrating moments, setbacks, and moments where motivation disappears. That happens to everybody. But consistency matters more than perfection. You do not have to win every day. You simply cannot stop participating in your own life. This is where resilience is built. And resilience is something I see throughout this community. I see it in the people continuing to show up, continuing to learn, continuing to move, continuing to encourage each other. And that takes real strength. I hope everyone in this place remembers you still have purpose. Please don't forget that. As we go through this conference, my hope is simple. I hope people leave feeling encouraged, understanding that they are capable of more than what they think. Stop underestimating themselves and begin seeing movement differently, not as a comparison or a pressure, but as an opportunity to improve your quality of life, build confidence, maintain independence, and reconnect with purpose. One of my favorite verses is Philippians 4:13. I can do all things through Christ who strengthens me. To me, this verse is not about perfection. It's about perseverance, faith, and continuing forward despite challenges. Believe stronger. Believe stronger than fear, doubt, limitations, and understanding that your diagnosis may influence your journey, but it does not define your worth. There is still strength inside you. There is still purpose inside you. There is still life experience and still growth ahead. And together we'll keep moving forward. In the other room, there's some green folders I want you guys to pick up. And tomorrow I'm going to show you guys a phase. We actually started a free app for everyone last year. And what got me motivated to do it was I knew people needed exercise. And this app that we've created has 3 different kind of exercises in it. It has seated exercises, hardcore workout exercises, workouts for everyone. So you can do it with your friends and with your family. It is absolutely free. We have dedicated it to help people with neurological disorders. So please stop in. And also, there's a bracelet that you can wear that says believe on your hard days. But now I'm going to do what I'm supposed to be doing, get you guys motivated to work out a little bit. So let's work out. All right, 1, 2, can you guys hear me? All right, so everybody sit up in your chairs. If you can stand, please stand. I'm going to get you ready for lunch. All right, the first thing we're going to do is we're going to take our arms and take them up just like this. Let's do 10 of these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now break for a second. Now get your abs tight on this next one. Take a deep breath. Abs tight. Let's do it again. 1, 4, 5, 6, 7, 8, 9, 10. Now shake it out. Now this time I want you to reach as tall as you can. I want you to get your abs tight and be intentional about the motion and movement that you're about to do. Okay, take a deep breath. Abs tight. 10 of them. Here we go. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Now shake it out. Clap for yourself. Good job. Next exercise. You're going to give yourself a hug. You're going to reach way out here. And you go just like this. Let's do 10. Here we go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now break, shake it out. On this next one, I want you to get your abs tight. Now what we call this is controlled motion. I'm telling my brain to tell my abs to get tight. I'm going to do intentional reaching, and I'm going to reach as far as I can. And I'm going to come together. Let's do 10 more. Here we go. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Clap for yourselves. This is a conference, right? Are you guys excited to be here? Okay, because I've been listening to everyone talk, and I'm pretty excited to hear the claps at the end, but I'm kind of wondering how excited you are to be here. Are you excited to be here? Right. So on this next one, let's do 10 more hugs, and really hug yourself, all right, for being here. Here we go. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, and break. Good job. Okay, next exercise. Yeah, clap for yourselves right there. Okay, next exercise. If you need to, stay seated, but we're just going to march. If you need to, grab onto a chair. Do anything you do to keep your motion moving. So this one, you're just going to pick your feet up. You're going to set them down. We're going to march out 10 times. Let's go. 1, count with me. 2, 3, where you at? 4, where you at? 5, where you at? 6, louder. 7, come on. 8, 9, 10, break. Clap for yourselves again. Now we're getting some momentum. Okay, the next one. So to keep our stability, I want you to keep your abs tight. Take a deep breath. Every single time you come up, squeeze your abs. Bring it down. Here we go. Abs tight. 10, 9, 8, 7, come on. 6, 5, 4, 3, 2, 1, break. Clap for yourselves again. There you go. Next exercise, let's work those biceps so you guys all look jacked today while you're eating lunch. All right, you're gonna go just like this. Hammer curl. Straight up. Now here's the thing. If you're seated, you can do these exercises. If you're standing, please do these exercises. Do your best to participate on this one. Okay, you guys ready? Here we go. 1, 2, strong reach. 3, reach for the sky. 4, reach higher. 5, higher. 6, there you go. 7, keep going. 8, keep going. 9, 10. Break. Now I want you to do one thing. I want you to squeeze your abs again. Just go abs tight. Abs tight. So when you say abs tight, this helps you with your stability. So everybody do it really, really close. Ready? Say abs tight. Abs tight. What are you doing? You're keeping your abs what? Tight. Keeping your abs what? Tight. Keep your abs what? Tight. Don't forget that. Every time you stand up, every time you move, keep those abs tight. That helps us with our stability. So let's get our abs tight, and let's do this exercise one more time. Here we go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Break. That was perfect. Clap for yourselves. Okay, we're almost done. I'm going to tell you tomorrow's more intense. Okay, just to let you know. So wear your workout clothes tomorrow. I'm just kidding. Okay, next exercise. I'm going to have you grab onto a chair if you need to. If you need to stay seated, we're going to do a seated or a standing leg extension. You're going to bring your leg up and kick it out. You guys have been sitting for a while. Here we go. 1, 2, 3. Your abs are what? 5. You just did 2 things at one time. 6. You talked and did an exercise. 7, 8, 9, 10. Break. Other side. Get yourself situated. Grab onto, switch sides. Grab onto something you need to. Leg up. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Break. You guys are pretty excited and pretty motivated. So if somebody came in the room today and said, if you did this as hard as you could, extended that leg as far as you could, I'd give you 5 more years of your life, what would you do? Would you be like, no way? Or would you kick that leg out even further? So what I want you to do is I want you to kick that leg out as far as you can. I want you to keep your abs what? Tight. It's going to help with stability. All right, leg up. Here we go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Other side. Here we go. 10, 9. Let's go backwards. Mix it up. 8, 7, 6, 5, 4, 3, 2, 1. Clap for yourselves. Go ahead and have a seat. So I've heard a lot of people talking today, and I've heard a lot of doctors and researchers talk today. And the one thing I want you guys to understand, I'm going to switch. I want you to understand one thing. In order to help them do their research, we need to do our part. And movement is our part. There's still things that we can possibly do. So when I say movement is medicine, but I also want you to remember that movement helps medicine. So in order to make ourselves stronger and to help those researchers out, please do your part. And it doesn't have to be the Believe Stronger app. It has to be something, but we did create something for you guys. So I want you to remember something before I leave and before I talk to you guys tomorrow. Every single day, tell yourself that you believe in yourself. If that's hard for you, find us at Toolshed Training or the Believe Stronger app, and I will tell you personally how much I believe in you. I already responded to an email today, and I promise you, if you send me a message and you have any questions, I will respond to you as immediately and as fast as I possibly can. Thank you guys for this opportunity, and I'll see you tomorrow. Also, don't forget to pick up those folders, okay? Just so you don't get after me.
Yeah, we don't want that to happen, do we? Thank you, sir.
Thank you so much.
Thank you, sir. Ladies and gentlemen, Javier Tuel, thank you very much. All right. How's everybody enjoying their lunch? Doing okay? Good. That quiet room? Okay, that's a good sign. Very good, very good. No big deal. Everything's good. We're actually a little bit ahead of time, so taking it easy. We're taking it easy. Hey, I just did want to give you a couple of notes while you're there. Okay. Let me bring them up. A couple of things we wanted to remind you guys about, that we'll be taking a group picture for the 5K run, walk and roll at 6:20 today, just before dinner, outside of the ballroom. Okay, so any past participants, please wear your 5K shirt from previous walk, roll, and runs that you've done. And if anyone is interested in participating this year, please join us. All is welcome. Also, there are 2 rare bottles of bourbon to auction later tonight. One will be in the live auction. Details will be shared later. The other will be in the silent auction. It is in the room with the raffle baskets. It's a very unique bottle. If you have any questions about it, ask Jim or Melissa Sheehan, okay? Another thing we want to remind you that tonight, make sure you bring your paddles, okay? They're going to be an integral part of this evening. So if you got in your bags, you had paddles with numbers and stuff, and we know who you are and all that stuff. They're going to be a big deal later tonight, okay? And then we also remind you once again to thank our sponsors. Guys, check them out on page 4 of the big brochure and the big book. And we just thank them immensely for all the help that they do in helping put this on today, okay? And now here's another note of encouragement. We encourage everybody to remember that one of the greatest strengths of the SPF community is the relationships that are built here. Scientific progress happens through collaboration. Patient support happens through connections. Many of the conversations that change lives occur not at the podium, but around a lunch table, in a hallway, or over a cup of coffee. So we're glad that you get to enjoy your lunch today and take some time to meet someone new, reconnect with old friends, and continue the conversations that bring this community together. And of course, here's another note. We have a special guest today. Fred Bird from the St. Louis Baseball Cardinals will be arriving and be available for some pictures out in the lobby space between 1 and 1:30. So if you're interested in doing that and being with the Cardinals baseball mascot, please do that. Okay? So we are just a little bit ahead of schedule. We do have a presentation for you that's going to happen while you're having lunch, and we're going to get that fired up here in just a minute or two. Okay. Let's see this. Oh, come on, baby. Work for me, work for me. There we go. There we go. Oh, you're killing me. Oh, come on. God, there we go. There we go. Stay. Nice. Okay, guys. Hey, LJ, we okay? We ready to go? You're the man. Here we go. One of the themes woven throughout this conference has been the convergence of the critical building blocks for therapeutic development is creating opportunities that would have seemed unimaginable a decade ago. As we work to accelerate therapies for HSP and PLS, it is important that we remain open to ideas, technologies, and scientific advances emerging from many disciplines. Innovation often occurs when knowledge from different fields comes together to create new possibilities. Our next speaker brings a unique perspective to that conversation. Dr. Chadwick Padromos is an internationally recognized physician, researcher, educator, and leader in regenerative medicine. Throughout his distinguished career, he has worked at the intersection of critical care, science research, and biological innovation, helping advance our understanding of tissue repair, regenerative therapies, and emerging approaches to improving patient outcomes. He holds degrees from Princeton University and the Johns Hopkins University School of Medicine, and completed advanced training at some of the nation's leading medical institutions, including Harvard Medical School and Massachusetts General Hospital. He serves as medical director of Forum and is a founding member of the American College of Regenerative Medicine. As medicine continues to evolve, researchers and clinicians are increasingly exploring new ways to support healing, protect vulnerable tissues, and improve quality of life for people living with chronic and progressive diseases. While many questions remain, these emerging areas of science are expanding our understanding of what may be possible in the future. Today, Dr. Padromos will share his perspective on innovation, regenerative medicine, and the evolving landscape of biologically based therapies. Please join me in welcoming Dr. Chadwick Padromos. Thank you, sir. And I hope I got that right.
Oh, that was one of my
reserves.
Thank you, sir. Hi. So let me just tell you, there are some wonderful researchers here who are doing awesome research. But let me just tell you something. The point, so you're going to want to listen carefully. We have treatment I'm going to cut to the chase right now. I'll get to this in about 15 minutes. But we have treatment that today is helping people. Proven, published research, getting people better with neurodegenerative disorders, specifically PLS, many other things. So the idea is, when will there be treatments that are available now that can help people today? And I will tell you that day is here now. So let me run through this. I'm going to talk really quickly because I just want to get through some boilerplate things. I was asking Norma, I said, should I just talk about PLS or other things we're doing? And she said, talk about other things. So I'm going to take maybe 10, 12 minutes to talk even faster than I am now. And then I will talk to you specifically about PLS and leave time for questions. So, so we right now perform treatment. The logistics and the regulatory environment of what's going to go on is very important. And I don't have slides about it, but I will just tell you, I treated RFK Jr. for his spasmodic dysphonia with the same treatment that we're doing for people with PLS, ALS, and other things. And in February of last year, he had a remarkable improvement. And if you listen to him talk now, his voice is much stronger than it was. This happened basically overnight. He asked me to advise him on policy matters. So I was the principal speaker at the Stem Cell Roundtable in Washington, D.C. last March. Again, met with Vinny Prasad before he was let go with the FDA. I just met with RFK Jr. and the FDA leadership last week. There is a lot of turnover going on. There's great opportunity. So I am right in the middle of the changing regulatory landscape, which is accruing to our benefit. Without getting this too much, the entire stem cell industry is forced offshore by some wrong-headed regulations until last year. Some states have passed laws that now allow this to be done here. It's a little complicated as to how, and there's certain conditions, but we had been in Antigua for 5 years. We are now in Naples, Florida, treating patients. We also are able to do this work in Texas. We actually have an office in Athens, Greece, where we treated a PLS patient, and they're just opening one in Tokyo. Asia is dramatically ahead of us in regenerative medicine due to regulations, by the way. Cultured stem cells, which are the kind that are useful, are prohibited by the FDA. Now, let me just say something about stem cells. 20 plus years ago, there were all kinds of ethical issues about embryonic stem cells and abortions and all that. Let me just tell you, that is 2 decades old work. These are embryonic stem cells. Nobody uses them. They're dangerous. We don't. So there are no ethical issues. We use stem cells that ladies donate placentas and umbilical cords after term cesarean sections from newborns. The stem cells are cultured. Exosomes, which I'll describe to you, are derived from the stem cells and are used for treatment. These are, in Florida, we have certain regulations where we can do this now, pursuant to a law that they just passed. And I'll talk to you a little more about that. So we have transitioned from Antigua to Florida. So stem cells, and I will tell you, and this is going to get a little confusing, and I know you're not biochemists or cell biologists, but the treatment that I'm going to describe are not stem cells. They are exosomes. Stem cells are ineffective for neurocognitive disorders. They're ineffective for ALS, for Parkinson's, for PLS, for everything. And for that reason, we have not used them. I founded a 501c3 nonprofit. We have our own federally approved institutional review board. We have 4 full-time researchers, 2 biomedical engineers, 2 cell biologists. We publish regularly, and it separates us from pretty much everybody else in the space. We publish regularly in PubMed index peer-reviewed journals. We've had 15 papers in the last 2 years alone. We are the only people in this hemisphere, except for the University of Miami, that does this regularly. If you guys access our website, our papers are up, they're readable, they're downloadable. We're completely transparent of everything we do. But I just want to tell you, stem cells, we will not do them. And I was talking to somebody here who had somebody scammed by them. So if people are telling you, excuse me, that they want to give you intravenous stem cells, don't do it. They don't work. We would not do it. We are talking about exosomes, which is a different thing. They're derived from stem cells. So I just want to get through. So we use exosomes from a lab called Cellularity in New Jersey. These cells are AABB approved, which they have to be used in Florida. They are the best stem cell manufacturers, I think, in the country. So let me talk about our clinical work. So what really matters, it's unclear exactly how these work, although we have some information. Probably they work on the mitochondria from some Chinese research. But we do clinical trials. And you will hear here from people here talking about getting funding so they can start a clinical trial. And it often takes a few years to get $7 or $8 million for a phase 1. And then you have to do a phase 2, which is $100 million. And then you have to do a phase 3. And the total cost of getting a drug approved now is $2 billion, takes 10 to 15 years. We are trying to change that as we speak. I am in active conversations with Washington, D.C. about getting somebody in who will turn back the clock to the way it was in 1960 when it was $2 million and 7 months for a drug, as opposed to today when it's $2 billion and 10 to 15 years. But I have my own research organization, our own institutional review board. So we started a clinical trial in May of 2024 based on 41 animal studies which showed the following. So this is the important stuff. Exosomes are tiny vesicles that exist within stem cells. They're about 1,000th as big. When you inject any of these things intravenously, they cannot get into the brain because of the blood-brain barrier, which you've heard about. But what is that? That is, the capillaries in the brain are coated with neurogenic cells that do not let things escape. That's why these treatments don't work and why we would never do them. But what you can do to bypass that is give things intranasally. At the top of the nose, 2 nerves pierce the cribriform plate into the brain. Pharma for 25 years has been using special syringes to inject things into the nose and get them into the brain. So there were 41 studies that we reviewed 2 years ago where people used intranasal exosomes in mice and rats. Exosomes, again, are vesicles found within stem cells. They're not living cells. They're much smaller, but they have the medicine of stem cells. They have growth factors, cytokines, and microRNA. So 41 studies, and they did studies on models of autism, ALS, Parkinson's, stroke, Alzheimer's. And they found the following. I'm sorry, I'm not going exactly with the slides because I want to get through this a little quicker. They found no adverse events of any kind. And they found efficacy for all of these disorders in these animals. Now, normally, if you see that, you then try to get funding and you do a phase 1 study and a phase 2, and it takes years to get it and years to do it. But because I have my own research institute, because we had licensure in Antigua, I was able to get a phase 1 study, a study started in 3 months. So we started this in May of 25. We treated 3 patients with Parkinson's, intranasal exosomes. What does that mean? That means the patient lies down on a table and you have a little syringe that goes into the nose, a little tiny tube. This is what's used in people to get drugs into the brain. There's no needles, no pain, and it takes about 12 minutes, and you just go spritz back and forth, and these things climb into the brain, and they get distributed everywhere in the brain. We did 3 Parkinson's patients, and I'll never forget this, in May, and those patients that night were better. That night, we were stunned. They were walking better. Their faces were more animated, because I didn't really know at that point if this was going to work, but it clearly did. So we kept treating people. In November, we started treating people with ALS. We published our first 14 ALS patients in a good journal. We also did 2 patients with Lewy body dementia who did well. We did a patient with Kennedy disease, which you all have probably never heard of, but it's a genetic disorder. It is in many ways phenotypically similar to PLS. People get very stiff. We did a guy had horrible pain. He got dramatically better. And we did another thing called congenital myasthenic syndrome. So we published these 18 people. They're on our website. You can read the article from a journal. They got much better. We then published our first 19 Parkinson's patients. Fast forward to today, we just submitted to a journal 2 weeks ago, 70 patients with ALS, and we are going to submit next week, when we get done with it, 50 patients with Parkinson's disease. So I don't want to take too much time with this, but let me just tell you, with ALS, the only drug that does much of anything is Riluzole. Riluzole slows down the disease by about 15%. So instead of losing 1.1 points a month on an ALS-FRS-R scale, which we also use for PLS, they lose about 0.95. So it's a little better, but not much. In our study, people lose about 0.4, dramatically better. And 25% of our people with ALS are a little better or don't get worse at all. So that's ALS. Now, the Parkinson's data is even better. We treated a patient in Athens, Greece last July who flew in from the island of Corfu who has PLS, which I had honestly never heard of before that. So we treated this patient on one day. The next day, the patient came in, and he was able to do, and he was very stiff, did not talk well. He was able to bear weight. Sialoria, PLS people maybe know what that is. It's saliva, you drool, right? It doesn't happen to SPF, but it does with that. So he was able to do a straight leg raise the next day. His balance was better. He then went back. A week later, his wife texted me, and said his sialoria, which he had had for a long time, was gone, completely gone. Now, we had some customs problems with Greece, and we had some manufacturing problems, so we have not treated him again. But we called him. I called and talked to his wife in Corfu a week ago. And here's what she said. This is 10 months later, PLS patient. She said, number one, the sialoria went away for 6 months. But then it came back in January, but not as bad as before. She said he's still a little less stiff than he was. He can open his hand. His gait was good for 6 months, deteriorated some. She said, though, and she was very excited about this, she said before you treated him, he was deteriorating every month. She said since then, he has deteriorated 0. This is 10 months later. So we're very excited by this. We just treated another PLS patient in Florida 3 months ago. He came in the next day. He was a little looser, spoke a little better. This was not dramatic, but definitely better. He's coming back in 2 months to get treated again. So this is what we have to offer for PLS. Now, the good thing about this is that it has shown demonstrated efficacy, and as you probably know, nothing else has. Nothing else has for ALS. For Parkinson's, we have good results. The people are not progressing at all, actually. It's up to 50 people. But for PLS, there just isn't anything, as I think you know. But this does exist. Now, how does this work? We don't know for sure. And unfortunately, because of the regulatory morass, American universities don't do any research on this. And so all the good research comes from China, which is absolutely eating our lunch now, doing tons of research. Japan, too. Almost nothing taking place here, unfortunately. So there's a really good study in China that showed that these exosomes, when they get in the cerebral spinal fluid in the brain, it's like a backdoor to get into neurons. They go through the membrane of the cell into the cytoplasm. Then they go to the mitochondria, and they get to the outer and then the inner border of the mitochondria. Now in ALS, the inner border of the mitochondria folds, and they get things called Christi. They can't tell they unfold, but what they do know, and this is very important, is mitochondrial apoptosis. That means death is dramatically reduced. Mitochondria are made healthier, they live longer. There was one mouse study which seemed to show new neurons being formed. We're actually doing some other things, and I don't want to get into the weeds on this exactly, but we work. We do a lot of NFL football players. We do a lot of other things that have nothing to do with this, actually. And actually, if you look on our website, you'll see household names, Kirk Cousins, Tyreek Hill, a lot of people who come to us. So we have a doctor who works with them, who does EEG studies, who did EEG studies in our autistic patients, which we also have published. And with a heart strap, you could tell what the vagus nerve is doing. And the vagus nerve we found after treatment was much more active. The vagus nerve, if you don't know, is very anti-inflammatory. You can cure rheumatoid arthritis by stimulating the vagus nerve. So we did some patients, this is just happening right now, who came last month. One guy came from the UK, and he was treated on a Friday and a Saturday. Vagus nerve, Sunday was better. Then he came back a month later, and his vagus nerve was still stimulated. So this may be how this is working. I don't know exactly, but I will tell you again, we care a lot about if it works. It's nice to know how it works, but the question is, does it work? And we are in a unique position. I have the freedom to do these studies. I am independent. And by the way, I'm not selling anything. Every other study in the country that you will see about things that are used for treatment, somebody is hawking something. They're all industry driven, and they're selling things, and they hire doctors. I am kind of the last man standing. I'm a 72-year-old board-certified orthopedic surgeon. All my colleagues are all retired, right? But I have the freedom, because I have my own institute. Nobody pays me for anything. We use different things. We use exosomes from one company. We try them from other companies and see what works better. So I have the freedom to do studies if I want to do them. I have the wherewithal to do it. I have the resources to do it. All the resources thing gets kind of tricky. Virtually every other study that's done, Everybody else that you will see are done by wonderful, smart people, but who are constrained by their universities and have to do what the universities tell them. They can petition them and maybe they can get to do them, but it's very, very hard. And it's very unfortunate about our system, and we're kind of trying to change it a little bit. Let's see. So as I said, the studies to figure out how these things work are difficult, and we are interested. But the real thing you need to do is to follow up and see if they work. We do exquisitely detailed research studies. They're published in good journals. We have nearly 100% follow-up. Every treatment that we do is followed up by our research people. We take copious notes. So we have terrific, very detailed data. It takes forever to go through this stuff, but they're very high quality studies. The FDA, by the way, I was talking to someone who said, so our studies are recognized by the FDA. The FDA knows us. They like us. And they can be done outside the confines of traditional research organizations. Lewy body dementia. We've had terrific success doing that. It's terrible disorder. Nothing else has worked for that. Essential tremor, 3 patients so far. This is what we treated. Trigeminal neuralgia, just to give you an idea. We had a patient with a 27-year history of trigeminal neuralgia, very painful, intranasal exosomes. Symptoms were gone. She got off of meds, felt infinitely better. She's out to a year now. Autism. We have a published paper on autism. We've had, and by the way, if you all are interested in this, it's all on our website. We published a paper about it. We've had terrific success. Intranasal exosomes, stem cells actually work for that. We've had some people with hearing loss and tinnitus get better. Okay, so getting to the PLS patient from Greece that I referenced. Let me read you this. This is from what she sent to us at her 2-month follow-up. Good morning, doctor. I would like to give you a report. You guys probably can't read this, so I will read it. Nikos, that's his name. Nikos Koutsopakis. I'm of Greek descent, by the way. So this is broken English, but this is a direct quote. It still does what he was doing with his mouth, meaning this saliva. This was before it went away. And what the physical therapist tells me is that when he does the exercises, his body listens very easily. This is how he says to me that before the exercises, it was too hard to do stretching or the other things he was doing, but now his body goes on and listens very easily. Also, Nikos says that he can lift his foot more easily, more in the beginning, meaning that he has better improvement. I am walking with him, and before, when he was walking, he was shaking his head because his head was going round and round because he didn't have balance. But now he walks more stable, not more fast, but more stable. And he lifts his foot more and better and more easily. His body every week has an improvement. Didn't stop. It is going little by little, but better every time. Whatever started from the first day goes on better, has more improvement, to say better. The way he lifts his foot, his mouth, and his body with the exercises is listening very good. This is not only my opinion as I watch him, but also the physical therapist who always practices with Nikos almost every day. Now, this is from when I talked to her 10 days ago. I don't have a quote from her, but this is what she said. That the sialorrhea I mentioned completely disappeared for 6 months, but then partially recurred. However, even today, it is still less than it was. He can still lift his leg, it's less stiff. More importantly, and she was excited about this just last week, that prior to treatment, he was worsening most parameters, but he's had no decline at all in any way since treatment. This is a patient from Western Pennsylvania who we treated 2 months ago and are going to treat next month. Dr. Padromos. As you know, he wrote this. I had 2 treatments on consecutive days in April of this year. Due to the PLS, the little finger of my left hand has been curling under for about 4 years. The night after the first treatment, I was able to straighten the finger part way, as if this time I can still partially straighten it. Since the onset of PLS, I wake up congested. I have tried many things to relieve the morning congestion, including but not limited to humidification and Claritin D. Nothing worked. A few days after the treatments, I realized that the morning congestion was gone, and I am still clear when I wake. Physical therapy does a blood pressure reading on me here 3 times per week. I tend to have high blood pressure. Since the treatments, my blood pressure is lower, even recording a 60 for the bottom number. So we're going to treat him again. What we have found with patients is the treatment, the problem tends to come back, and it comes back at different rates with different disorders. For ALS, it tends to come back within a month or 2. With Parkinson's, 6 months or a year. I don't know for the PLS, and the only way you can tell is to do more and more people and get longer follow-up. You know, I have 2-year follow-up on the Parkinson's, 18 months on the ALS, 70 patients on the 150 on the other. I have 2 patients with PLS, but I'm encouraged that the results seem to be longer lasting. The future, stronger exosomes, neural exosomes. Now cost. So let me just tell you, this is patient funded. We have no funding, although I will tell you, not for this, but for our stem cell treatment, I treat a lot of NFL players for sports problems, and there's things that you can read in the national media about me and a lot of these people. I won't get into it here. Actually, Kirk Cousins come to us, brought NFL films down, and Netflix did a documentary that I'm in. But anyway, our stem cell treatments are about $22,000. Our exosome treatments for like Parkinson's are about $12,000. But I've kind of committed for people with ALS to treat them at cost, like $1,000 above cost, and I'm going to do the same thing for PLS. So let me just tell you, why am I doing all this? I don't make any money doing this, believe me. I have two goals. I want to help people, and I want to pay my bills. I can't go under doing this, but I want to get data. Because with data, we now have access in the U.S., not in every state, but Florida, Texas, Georgia just passed a bill, Arizona's about to pass one, Montana has passed one, all red states, by the way. So California, New York, Illinois, forget about it. Can't do it, probably never going to be able to do it. Oh well. So we have access in the states, but it needs to get paid for. So, as I might have mentioned, the NFL is already paying. They're paying for this NFL player because they know it works. We're going to the airlines because airline people get ALS more than others. We need data. The data is power. With data, I've already talked to Mehmet Oz about paying for ALS. We're talking to Blue Cross. When you get data and you can show them that it works, and by the way, doctors from all over, people find me from all over the world. We had a guy come from Mongolia for autism, and he came, and the patient, his boy did well. He came back a year later, and it's kind of amazing. People are very grateful. He has a cashmere factory and mill in Mongolia, and so he brought this big thing that was woven from cashmere with my picture on it and things written on it. He carried this thing from Mongolia to Antigua, and so we have testimonials, but what you really need is data from good studies, and we have that. And when you go to the Blue Crosses and you go to the Medicare with good data, eventually we're going to get this paid for. We're working simultaneously with RFK Jr. and the people in government to try to make the studies less expensive so you can get things FDA approved more easily. But data is power. Doctors, the lead doctor for this disease at the Mass General found me. People call me because we publish. So I want to treat people. Cost is an impediment, and I know it, and it bothers me. And people with ALS and people with PLS, they have no money. Their lives are destitute, right? So we are trying to make it as cheap as we can so people can come. We're applying for grants. I don't know how well we're going to do. Actually, I was saying to Norma when I called, and I became aware of you guys, and I knew nothing about SP. I get the initials wrong. Whatever. Thank you. Although we're helping a company that has a drug with the FDA for spinal cerebellar ataxia. But all I really wanted to do was tell people about what we're doing. And then she said, we have money. So I said, Norma, I didn't call you guys to ask you for money. And I didn't. But I will tell you this. Money that goes to us goes directly to treat patients, and it goes to help defray I pay over $1 million a year out of my pocket to pay the researchers from my foundation and do this. And it's difficult to do. So I am committed. So we're doing PLS people. I have to buy these exosomes. So we're doing PLS people. We charge $1,000 over cost because we have shipping charges, rent charges, things like that. So for that, it's about $6,000. So it's expensive. It's not crazy money, but it's expensive. I want to do 3 people with the spinal paralysis, and I'm committed to do 3 people either for free or very close to free because I don't know if it's going to work. I know it works for everything else I've told you. I would say of all the things I told you, all our great results, for Alzheimer's, it doesn't work at all. We did 5 Alzheimer's patients for free, did nothing, so I stopped, right? So I don't know if it's going to work for the spinal paralysis or not. I think it will because I will tell you, every disorder that we've treated that involves motor neurons has responded. So that's ALS, PLS, Parkinson's, Lewy body dementia, MS, Kennedy disorder, congenital myasthenic syndrome, and I'm probably forgetting one. Every single one. So I think it's going to work. but I don't know that for sure. And I don't want to charge people anything. So I'll just eat it. I'll just pay for the cost of these things to do like 3 people and see. So you have to buy a plane ticket to Naples and a hotel room for a night, and we'll try and see. I have one young lady. Are you here today? Yes. Where are you? Right here. Okay, I don't want to say her name, but and one thing we were talking yesterday that I think I might have forgotten to tell you, and I don't know if I did or I didn't. We have to be very careful. So the media watches me very closely because they know that I work with RFK Jr. and they see that we publish. So New York Times wanted to interview me about Kennedy, and I wouldn't talk to him. ProPublica interviewed me, and I hesitated, but they're pretty objective, so I did. The Guardian, you know The Guardian? So The Guardian, Monday night, the night after I met with RFK Jr., sent an email and said, and there's a guy named Knepfler who goes after private stem cell organizations, and everybody's very afraid of him, and I was too. And he finally found me, and he wrote 9 months ago, asked me questions, and I answered them. And he wrote a piece that wasn't bad, I think, because we're doing things right. The Guardian were writing a hit piece about stem cells for autism, and he wrote a lot of very aggressive questions, but we wrote this very long reply with all our evidence, and that article just came out 2 days ago, and it actually was not bad. It had some bad things to say about other people, but not us. But he was quoting the law, asking how we can do what we're doing and stuff, and we are squeaky clean. We follow every regulation there is. So to do exosomes or stem cells in Florida, they have 3 indications. They have, or did I talk to you about this? Orthopedics, wounds, and pain. They have to have one of those 3 things. So for example, he was saying, how can you do autism patients? Because they're not approved, and he's right. But a majority of autism patients have headaches, and all the patients we do have headaches. We had one kid who was banging his head against the wall, and he had head pain all the time. So for PLS or the spinal condition, you have to have pain. And most of our people do. Our Kennedy disorder patient looks like PLS, had horrible leg cramps all the time and got better. So to do anybody in Florida, you have to have pain, and I have to document it in the chart. If you don't, the regulations are a little different in Texas, and we can have our affiliate do it in Texas. It costs a little more because I don't charge to administer it, he does. So it's about $600 more in Texas. But so I have to talk about this because, as I said, people are watching me all the time. We're kind of out, front on this. And by the way, scams abound. I can't tell you the number of people that I've had who have had $20,000 taken from them in Mexico. I had one Parkinson's lady who got treated in her spine in Mexico, and they crippled her, got her sick. There's all kinds of bad stuff out there. So I don't blame people for being skeptical. But I do want to mention that for anybody who's interested in doing this, so I was going to take the first 3 people, first come, first serve. So she's number 1. So if there's a couple other people who are interested in getting treated, come see me now, and we can get that set up. For PLS people, so what the way to get in the pipeline if you want to, I think my contact information is someplace. I hope it is. And I'm going to hang around, and I've got cards. If you call our office, we've got a wonderful young lady who does logistics. She onboards people. I spend my day seeing patients. I spend my nights calling people, and we discuss everything that I'm telling you here. And then if everything's appropriate, we book you there. Our research team deals with people. We're very fastidious of doing about research ratings before and after, like ALS, FRS-R. I was talking to a young lady. I don't know what's the best research form for the spinal paralysis. Actually, I was reading about it last night. We have to figure that out, but we'll do that too. So that's with our research team. So I think I'm just about done here. So this is our contact information. So if any of you are interested, normally it's no place printed, right? My contact information? Probably not. And she was very nice to get me on the docket anyway. But if any of you can see this, there's a phone number up there. If you call that, all you'd have to do is call, and we can we can talk if you're interested. I'm happy to answer any questions. If you look on our website, as I say, our research is all up there. If you press the research link, the papers are up there. You can read them. You can download them. We're completely transparent. I tell everybody that I treat exactly how many people we've done. I am absolutely not trying to sell this. I don't make money on it. I'm trying not to lose so much money that I can keep my doors open. And we tell people, if you have any doubts about this, you shouldn't come. We're kind of overwhelmed with people that want to get treated, to tell you the truth. Anyway, that's that. I have a whole nother talk, which I'm not going to do, and describes what we do with stem cells, our work with football players, autoimmune disease, all kinds of other stuff. But I kind of wanted to, excuse me, just get the main points out about this. So I think that's everything I have to say. I am happy to answer questions now. I'll be hanging around answering questions. I'm leaving in a couple hours to go back, and we're happy to answer inquiries. So let me ask, are there any questions at this time? So can you guys see that number on the screen? So write it down. That's all you really need. And I'm sorry, I have cards. I can give you a card, but so if you just call that and say you're interested in stem cells, exosomes, anything, our team will put you in touch with Zara. Excuse me. She will pre-screen you. She knows, we know what we can do and what we can't, like Alzheimer's. I wish I could help it, and we're working on other things, but right now we just can't. So if somebody calls about Alzheimer's, she'll say, I'm sorry, we can't treat it this time. She knows these other things we can't. Some things she isn't going to know. So the hereditary spinal paraplegia, she's not as familiar with. But anyway, but if you say you're at this meeting and whatnot, if she doesn't know, she'll ask me, and then she'll set up a call. She'll ask you to, and people ask what it costs, and it's kind of variable. And some of this is a moving target, by the way. I make deals with the companies, so they'll give me their stuff cheap, and I do research studies for them. But some of this stuff is like, what are you going to charge me this month, So she knows pretty much for ALS people, and we're treating the PLS people like the ALS people. Anyway, if you just call that number and ask her your questions, she'll get you plugged in, and we can talk. The other thing is our emails, but the one is mine. The other is our website. It's the lower one is our foundation, our 501c3 nonprofit. The top one is the medical corporation that treats. So it's 7 letters, the PSCI. It's PadromosStemCellInstitute.com. So if you're interested in finding, or you can just Google me. If you Google Padromos and stem cells, I think you'll find it. And you can read a lot more what I've talked about here, but a little slower. Are there other questions?
Hey, doctor, we've got one over here on your left side, left-hand side over here. Right over here,
right
look
there.
at me.
Got it, thank you.
What is the legislation that you need approved?
Well, let me tell you, for instance, when I treated Bobby, and I've got a video I could show you, the next morning, his voice was dramatically stronger, and it's been strong since, and we talk. And he said to me, Chad, I would like you to help me. These are kind of his words. He said, I would like you to help me increase stem cell access in the United States with what I can do with the stroke of a pen without it being the wild, wild west. So, because you see, the problem with this is, if you make it too open here, there's scams out there already. I mean, in Florida, you have to be very careful. So here's the thing. Without getting into this in too much granular detail, there were 2 regulations put into place in 2005 with their complete nonsense, made up out of thin air, that says that you can't more than minimally manipulate cells. You basically can't culture them, and you have to culture them. The cells are gotten from these umbilical cords and placentas, but they have to be cultured. We're giving people for stem cells 150 million stem cells. If you don't culture them, the most you can give people is like 50,000. Now, that's hard written into a statute, and there's another regulation too. The only way to change that is to go to Congress, and I mean you guys read the papers, right? I mean, that's not happening. But there are ways around it. And the ways around it are, the states have passed laws that say, the Florida law says, we recognize that cultured stem cells are prohibited by the FDA, but we think people should have access. Therefore, if you do these things, you have to do them only for certain indications. You have to have high-quality cells, a lot of other stuff. We'll let you do it here. Now, Supremacy Clause of the Constitution says that the states cannot trump the feds, right? So the federal government can shut this down. And when I met with Vinay Prasad shortly before he was fired, I said, and Vinay is a purist, Vinay Prasad, I said, Vinay, you going to shut him down? He said, no. And I urged Bobby. I said, Bobby, just leave it alone. I said, let them do it. There'll be guardrails. I don't think people are going to get hurt because it's extraordinarily safe. This is the safest treatment that exists, by the way. We have had no serious adverse events at all with these exosomes. Nothing bad happens. You lie there, it doesn't hurt. The only thing that happens is most people say they sleep a little better. But there's nothing safer if you do it right. If you do it badly, bad things happen. Anyway, so where it stands is they're letting them go. They're letting the states do it. It is exactly analogous to cannabis. Cannabis is federally illegal, but various states have said it's okay for medical, recreational, what have you. And the federal government does not shut them down. And they're not going to shut this down either. As far as changing it with Congress, I think personally, there's no chance. We are working with the FDA, and it's a lot more complicated, but the studies, the $2 billion that it costs now, this can be much closer to the $25 million that drugs cost in 1960. That's in 2026 dollars. You have to make changes as to how they interpret the study requirements. And we're right in the middle of doing that and getting new people in. I don't know how that's going to go. But regardless of that, and that affects pharma more. And there's some horrible pharma things we're dealing. You may have read about the melanoma study that was approved and then turned down. The Wall Street Journal wrote an article about this. It's horrible. Never should have been turned down. There's another one for SCA, ataxia, spinal cerebellar ataxia, yeah. And I've met with the CEOs of these companies. The data is rock solid. They were approved and they were turned down. Horrible. And so we have talk to the government. I was telling Bobby, telling the other people there, you got to approve these. So, but the main thing you need to do is make the studies a lot cheaper, and this can be done. We've got a real opportunity now. My politics are going to be showing a little bit, and forgive me, but both parties have been, let's just say, influenced by pharma. But RFK Jr. is not, and Donald Trump is not. They're willing to buck them. And so we have an opportunity now. If it doesn't happen now, I don't know if it'll ever happen in the future. But anyway, so that's kind of where it stands. And we'll know in the next 3 weeks how much success we're going to get with the new people in there to get the study requirements changed. With our case, with stem cells, we're in pretty good shape with these stem cell laws. Pharma is in worse shape because they have to pay these crazy dollars. That's why somebody was saying it's like $1 trillion to do all this stuff. It is. It's obscene. Shouldn't be. And that's what we're trying to get changed for drugs. Does this answer your question? Okay. No questions?
Any other questions, anybody? Okay.
All right.
I don't see any hands. Here we go. Thank you very much, Dr. Padromos. Thank you very much, sir.
And I'll hang around.
Okay, guys, we're gonna take it easy for a little bit. So make sure you get up, stretch a little bit, take a little break. We're a little bit ahead of schedule, so we're going to give you a little bit of time to relax and talk and warm up a little bit. Use some of Javier's workout techniques to kind of keep that blood pumping, and then we'll get back into the show in a little while. See you soon.
Well, what a morning. A lot of information. My brain can't keep it all and digest it all as quickly. I'm going to have to take notes, go home and think about it and do a little more research. Hope you guys are able to hear all the things, all the different ideas that are talked about. And as you know, we're trying to move the foundation in a direction of being on the cutting edge, helping bring all the parties together collaboratively to find that treatment as soon as possible. So I hope you're finding this information very positive. But we're going to take a short break here. We'll be back about 1:15. Our first speaker this afternoon is Dr. Sachdeva, who this foundation awarded a grant to just this last fall, and he's working and studying with us on neurostimulation techniques. So some new information, what can we do? I'm looking forward to hearing what he's doing in that area as we move forward. So you guys take a break with us, enjoy your lunch at home, and we'll see you again about 1:15. See you then.
Talking and chatting and having a lot of fun. And anyway, now it's time to move on. It's in the afternoon. And we've got a little change to our program because one of our speakers could not be with us. Unfortunately, Dr. Marcondes França Jr. from Brazil was unable to join us this year. However, we wanted to highlight a promising area of research that many of you have heard discussed throughout this conference, neuromodulation. What you're about to see is a short video demonstrating the potential impact that targeted neuromodulation may have on walking and mobility. While this is just one example and not a treatment for HSP or PLS, it offers a glimpse into the kinds of innovative approaches researchers and clinicians around the world are exploring to help improve function and quality of life. As you watch, I encourage you to think about what becomes possible when science, technology, rehabilitation, and patient determination come together. Let's take a look.
Here, I think it's a very, very, compelling example of the benefits, of the potential benefits of neuromodulation. This comes from a publication released, I think, maybe 3 years ago in the New England Journal of Medicine. So this guy here, he was wheelchair bound because he's got pretty severe spinal cord injury after a car accident. And this patient was, treated with invasive neuromodulation. And as you can see here, he was, able to stand still and to walk for some distance using these kind of, device.
Well, that was a very short video. I was expecting 20 minutes, okay? But it got the point across, okay? It showed somebody who was able to move because of the neurostimulation. So now we move on to part two. And we're going to introduce our next speaker. One of the most exciting developments in neurology today is the growing recognition that meaningful improvements in function may come from multiple directions. Future therapies may emerge from drugs, gene therapies, biologics, rehabilitation, neurostimulation technologies, or combinations of these approaches. Our next speakers represent an area of growing interest, how the nervous system itself may be influenced through neurostimulation and functional neurological interventions. As we continue exploring the pathway toward therapies, it is important to remember that innovation is not limited to pharmaceuticals. Researchers around the world are investigating new ways to improve function, quality of life, and neurological performance through a variety of emerging approaches. Please join me in welcoming Dr. Raul Sachdeva, presenting neurostimulation and functional neurology. Dr. Sachdeva, there he is. Do you have the clicker for the thing? Well, guess what? I'm gonna go get that for you.
Okay.
There it is. Oh, there it is. Handle it.
Thank you. Thank you.
Good job, sir.
Thank you.
Breathing's good.
Can everybody hear me okay? Perfect. Thank you for the kind introduction, and thank you to Dr. Franca, who we were able to show some slides, some videos for, from his trial in spinal cord stimulation with people with spinal cord injury. And I wanted to also mention that I also come from the spinal cord injury background with some experience in cerebral palsy as well. I attended the HSP conference last year, the Spastic Paraplegia Foundation conference last year in DC. That was my first one, and I knew I had to come back for this one. And I also wanted to give a shout out to Dr. Bass, who introduced me to this world of spastic paraplegia. And so today, I will be presenting some of the early findings, or rather the feasibility of our trial with Spastic Paraplegia Foundation, the study funded by Spastic Paraplegia Foundation, to look at the effect of neural stimulation in patients with hereditary spastic paraplegia. So my objectives for today's talk is, one, I will very briefly introduce what neuromodulation is. I will talk very small, couple of slides about my findings with cerebral palsy and spinal cord injury and what we saw in the motor improvement in those conditions. I will also show some data from a survey that we've conducted with the help of the foundation last year. And this will be shown by one of my students, Makaira. She's sitting right here in front of the room. She will be talking about her survey data that she used as a part of her master's degree in my lab. Followed by that, we'll show some of the outcomes that we are looking at in our trial. And Dr. Pandey, who's also sitting here in the audience, he will come up and present a couple of slides and he will talk about what are the outcomes that we are measuring right now. We heard the word outcomes many times today and yesterday. So we will talk about what are the outcomes we are doing. And lastly, I will show very, very preliminary findings, what we have achieved so far from this trial that just started a couple of months ago. So we don't have a lot to report, but we have some exciting things to report. Okay, so neuromodulation, you heard this word. What is neuromodulation? The word neuromodulation, as you can guess, is you're altering the activity of a nervous system, of the nerves, of the neurons, whatever you want to call it, of spinal cord, of brain, by targeted delivery of a stimulus. That stimulus can be magnetic field, that stimulus can be chemical, it could be light. What we are focused is on electricity. We are using mild electrical currents delivered towards the spinal cord, and using that current to modulate the activity of spinal cord, and that's what we are calling electrical neuromodulation, spinal cord stimulation, spinal cord neuromodulation. You will see these interchanging words, but that's what it means. Using mild electrical pulses targeted towards the nervous system to modulate the activity of the nervous system. So just a brief history. Let's go 5,000 years back. This is where the first interaction of humans is known for electricity. This is way before the actual man-made electricity came into history. So this is from an Egyptian tomb 5,000 years ago. This is basically a stone plate that shows interaction of humans with electric fish in the Nile River, I believe. And that shows that humans knew about these electrical properties of animals 5,000 years ago. However, the first documented use of electricity in medicine was about 2,000 years ago, when this Roman physician used this torpedo fish, which can generate up to 220 volts of current. He used these fish to treat arthritis pain, to treat headaches, and that is the first documented medicinal use of electricity. This is way before man-made electricity came. Fast forward 2,000 years, this is the modern day neuromodulation. By modern day, I mean 75 years ago. This is one of the first cases of implanted spinal cord stimulators in a human being with chronic pain. He had complaints of chronic pain. And while this patient did not survive more than a week after implant, due to other reasons not related to this implant, within a day of implant, he was able to feel benefit of the stimulation, and he had no pain. Obviously, reporting is not great. This is an 80-year-old study. But this was one of the first cases of spinal cord stimulators being implanted in the spinal cord for treatment of pain. It doesn't look very different today from this one. So there are right now in the present day, there are two main ways we can stimulate spinal cord. There are many other ways to do that, but the two most common ones, one is an implantable device, where a stimulator is placed inside the spine, right over the spinal cord, inside the spine. So this is essentially between the bone and the spinal cord. And then the current generating power pack is placed somewhere under the skin. Do we have a pointer here? There's no laser pointer, no. Okay. What you can see in this image, you can see near the hip bone, there is an electric pack, which is where the current generates. And then this electrode cable that goes towards the spine, that's where the current is delivered. So that is an invasive way of spinal cord stimulation. It's called epidural spinal cord stimulation. Epidural means above the dura. The other way to do this is an image from a participant in our study, is do the same thing through the skin by placing these electrodes over the skin. This is called transcutaneous, meaning through the skin. That's what we are interested in my lab, using non-invasive spinal cord stimulation. And obviously, there's no hard guesswork here. You will have benefits of using a non-invasive versus an invasive treatment. Some of those basic benefits are, there is no need for surgery. You don't need to undergo any procedure to get this stimulation delivered to your spinal cord, because we are placing these electrodes just on the skin. It's obviously low risk. When there is no surgery, there is less risk involved. The cost is lower. These electrodes we just buy off Amazon. We are not really making any fancy stuff in the lab. This is just store-bought, off-the-shelf electrodes that can be placed on the spinal cord, and the spinal cord gets the current through them. This can be used and removed and can be stored for future use, for few uses. It is removable, so you don't have to wear them all the time. And the placement can be customized based on the target. Our spinal cord is segmented. Each segment of the spinal cord goes to a specific target. The lower part of your spinal cord controls lower part of your body. Upper part of the spinal cord controls upper part of the body. So the placement of these electrodes can be changed, whereas if something is implanted surgically, you cannot change that. And it does not make you ineligible for other trials. If you have something permanently implanted, in future if you want to get involved into a study, there is a chance that one of the exclusion criteria for the study is you cannot have something implanted in your body. So at least with something like this, you are eligible to try other things as well. Obviously, so now these need to be replaced every time. So is that a good thing or a bad thing? I think mostly it's a good thing, but I did want to mention that one of the concerns could be, how do we put it on? Do we need a caregiver? Do we need my husband or my wife to put this on me? Am I putting it correctly or not? So those concerns are there, and I just wanted to acknowledge those, that being able to remove them is most of the time a good thing, but it could also be a trouble sometimes. And also, some of you might wonder, especially the scientists in the room might wonder, if you are delivering something through the skin versus something that is placed right on the spinal cord, is it as precise as the surgical placement, or is it, wishy-washy? It's not that good. So these are the questions that scientists have asked in the past as well. So I'm going to give an example from this. So the way this study answered this question is whether spinal cord stimulation, when delivered either invasively by placing the electrode right on the spinal cord, or non-invasively by placing the electrode on the skin, does it have similar effect? So this was a study published in Europe a few years ago. And what they did was they provided spinal cord stimulation and recorded something called as EMG, electromyography, or electromyogram is the activity that is recorded, which is basically the activity from the muscles of the leg in this case. And the way they did it was very, very interesting and very smart. They had 10 participants in the study. 3 out of those 10 participants were eventually going to get an implant. So what they did was, those 3 participants, they first did the surface stimulation. They looked at the responses of the muscles when the spinal cord is activated from the skin. And then those 3 patients, after several weeks or months later, they went for an epidural implant. Now the same patient who got their muscle activity recorded from the surface are now implanted with this stimulator. I don't know what reason was that they chose to go for an implant, but that's what they did. And this is the muscle activity that they found. So the top 3 rows that you're seeing is each individual patient. And each row has different columns. Each column is a muscle that is being recorded. And within each column, for example, let's focus on S8, which is subject number 8. There are 2 traces. One is from epidural stimulation, one is from transcutaneous stimulation. And the effect is same. This is the intensity of firing. Same thing for other muscles, same thing for other patients. Now, if the last column where it says latencies, onset or offset latencies or response durations, all of that are basically characteristics of this muscle firing. And all you can see, I want you to pay attention is to 2 different colors of bars there. They're all very similar. So they found that the muscles are responding in a similar way, whether it's in response to an invasive stimulation or a non-invasive stimulation. The response is very similar. So that's why they concluded that there are common neural structures that are activated by these approaches. So why do we think that spinal cord stimulation, transcutaneous spinal cord stimulation, can improve function in people with spastic paraplegia? This comes from our experience with other conditions. One of them is cerebral palsy. Could I have you play this video? So cerebral palsy is basically a condition that occurs in response to an injury to the developing brain. And that leads to motor impairment and spasticity. It's a non-progressive condition, but it's one of the most common childhood motor disorders. Sorry. Oh, perfect. Thank you. Could we play this video? Thank you. So this is a child with the most severe form of cerebral palsy. It's called GMFCS level 5, and you can see she has no trunk or neck control. I'm going to later show a video of her with the spinal cord stimulation. But what you can see is this child also has a lot of motor impairments and a lot of muscle weakness. Spinal cord injury, if we can play this video, is a different condition which normally occurs in a traumatic manner. There are non-traumatic spinal cord injuries also, but it's normally a traumatic event that leads to disruption of the pathways within the spinal cord, and now it leads to similar motor and sensory deficits. There is spasticity involved, and then we all know about the spastic paraplegia. We've been hearing about this almost every year in many of these great talks here, so I'm not going to explain about HSP here, but the point is that there is a disconnection between brain and spinal cord, and now we have something left in the spinal cord that we need to work with in these cases. So these are very different conditions, but they have some similarities that made us think that spinal cord stimulation could help these populations as well. So this is the real-time effect of spinal cord stimulation in cerebral palsy. By real-time effect, what I mean is it is the effect when the stimulation is turned on. We are not looking at the long-term effect in this video. We are looking at the effect of stimulation being turned on. Let me see if I can play this. There you go. So when it says effect of scone, scone just means spinal cord neuromodulation. So you're looking at a patient for sitting and head control. It's the same person, same girl with level 5, the most severe form of cerebral palsy. And on the left, you are seeing spinal cord stimulation is off. Scone means spinal cord neuromodulation off. On the right-hand side, you are seeing when it's turned on. And I think the video says itself that this is, there is a lot more control and balance. Obviously, it's not perfect, but it's a lot more when the stimulation is turned on. And this study was done in collaboration with Spinex, which is the company that makes these. It's in collaboration with the company that makes these devices, spinal cord stimulators. So this study was performed at a pediatric physical therapy clinic. So the same child now looking at sit to stand, trying for the child to go from sitting position to a standing position. And I want you to pay attention to the leg of the physical therapist also here. See how much assistance she's providing with her leg on the left versus right. So see on the right side, the leg is already off from the therapist. There is no support at the legs. In the first attempt with scone off, the child could not stand. So it's trying again. So this is the most severe form of CP. I wanted to give you one more example of slightly less severe. So this is level 5. Level 4 is slightly less severe. This is a child with level 4 CP. There is head and neck control, there is trunk control, but there is no stepping. And when I play the video, you will see left side, TSCS, transcutaneous spinal cord stimulation off. On the right side, you will see it's on. And this is again the real-time effect of spinal cord stimulation, meaning when the stimulation is actively present. This is the child after 12 weeks of therapy. This was a video sent by their parents. We heard yesterday from wheelchair to bike. So this reminds me of that. So this bike is not motorized. He's doing it by himself. Okay, you get the message. So now I wanted to show one study that we recently published. It came out last month. This is a study in spinal cord injury. It was done by Dr. Raza Malik in Dr. Andrei Krasikov's laboratory. Dr. Krasikov is my postdoc advisor. I spent 10 years in his lab before coming to University of Kentucky and starting my own work. So this work was done in collaboration with Dr. Malik and Dr. Krasikov. And this study was actually focused on improving blood pressure control in people with spinal cord injury. So we were not even trying to improve motor function in these people. People with spinal cord injury have a lot of other autonomic issues, such as blood pressure control. But what we saw in some of these participants was improvement in motor function. So we decided to document that as well. So before I start the video, I wanted to tell you a little bit about the spinal cord injury here. So this is a 29-year-old male. NLI means neurological level of lesion. So unlike HSP, it's a focused injury at C4, meaning their fourth cervical vertebrae, they have a spinal cord injury. So anything below that is disconnected from the brain. AIS-B means American Spinal Injury Association, which is Asia, impairment score B. By B, it means the person is motor complete and sensory incomplete. What that means is the person has absolutely no control of movement below the level of lesion. The person has no control of movement below the lesion, meaning they cannot move their arms, they cannot move their legs, they cannot move their toes. And so in the video, you will see real-time effect of spinal cord stimulation again. After about 10 sessions of stimulation, the patient is asked to raise their legs against gravity. So it's not walking, it's not biking, but for a spinal cord injury patient who has a complete motor impairment, who are completely paralyzed, this is a huge effect if they're able to move their legs. So let's look at the video. Oops. Can we play the video, please? There you go. So when the stim is off, if there is a volume that you can increase, that will be great. So this is with the stim on. You can see the legs are able to move. So when the stimulation was off, there was no movement whatsoever. But when the stimulation is on, while it's on, the legs are able to move against gravity. And what I want to emphasize in all these videos that I've shown so far, this is not what you've seen probably in FES stimulation in physiotherapy clinic, where there is an electrode applied on the muscle and the electricity is triggering the muscle contraction. This is all voluntary, volitional movement. We are asking the patient to move, and they are moving it at their will. While the stimulation is on, they can choose not to move it. So it's not the stimulation that is causing the movement. It's the stimulation that is enabling the movement. And also the intensity of stimulation is not that severe that we are causing these triggers in this movement. Okay, so this brings me to the transcutaneous spinal cord stimulation for the treatment of spastic paraplegia. This is a study that is funded by the foundation last, I think, December, and we just started recruiting about three months ago. So what we are trying to do here is, the goal is to recruit 15 patients. This is a pilot study. All 15 patients will go in a treatment arm. There is no sham in this because we just wanted to see whether it works or not. And the stimulation paradigm is twice a week stimulation for 1 hour for 8 weeks. And we will do one last assessment after 8 weeks of no stimulation, meaning when the stimulation ends, the therapy ends, we will wait for 8 weeks and see if there is any persistent effect. So there is a baseline assessment, there is mid-intervention, there is post-intervention, which is where the trial ends, meaning the therapy ends, and then patient will come for one more visit for a reassessment to see if there was an improvement at the end of 8 weeks, will this last for another 8 weeks or not? This is the inclusion criteria. It's fairly straightforward. You need to be an adult to participate. Diagnosis of HSP for now. The hope is to include PLS in future. Need to have stable medications, and rest is just standard. You need to be able to participate. You need to be able to follow instructions. What I don't have listed here, which is an exclusion criteria, is the intrathecal baclofen pump implants right now. There are some studies that have shown that stimulation is safe with the baclofen pump, but we, for the early study, decided to not go with that because we did not have enough evidence to safely say that it is okay for baclofen pump as well. So we have right now 3 participants enrolled. The first participant who gave me his permission to say this, that he's sitting in the audience right there. So if you ever want to, know what it feels like, he's happy to talk to you guys. Thank you for doing that. Second participant, so he has already done half the study. We have done his baseline and mid-intervention. He's almost getting to the end. Second participant is, so he's 56-year-old male with SPG7. Second one is a 40-year-old female with SPG4. And the third one we just consented is a 24-year-old male with SPG11. He will start his first visit in first week or second week of July. So we talked about a lot of outcomes, and Dr. Fink mentioned about priorities of these individuals. Yesterday, there was a very good discussion on that. And I just wanted to highlight that, yes, there is obvious motor impairment in spastic paraplegia, but there are also non-motor impairments, which is chronic pain, urinary urgency, bowel dysfunction, sexual dysfunction, and even cognitive impairment. So yes, gait problems, balance, spasticity, fatigue, reflexes are impaired, all of that is there, but there are also some of the non-motor symptoms that may not be obvious to the outer world, but it may dictate the quality of life of the individual suffering from these. So why are we talking about this? I wanted to bring this slide from the spinal cord injury field. So this is Dr. Kim Anderson. She is herself a person with spinal cord injury. She's been living with spinal cord injury for almost 30 years. In 2004, she published this paper. So this is a snapshot from her Google Scholar. She published this paper called Targeting Recovery, Priorities of Spinal Cord Injured Population. And what I've circled there is the citation number. This paper has been cited almost 3,000 times. What she found as a person herself with spinal cord injury is that if you ask people with spinal cord injury, what is the number one thing you want to be improved? Look where walking is on that graph. The walking is, can I point it? Right there in the middle. The number one priority for these individuals with paraplegia was sexual function, then bowel and bladder dysfunction. AD means autonomic dysreflexia, which is blood pressure control. Trunk stability. Walking is not the number one priority. So me as a new scientist to this field of HSP, I was curious to know this as well, and we did not find any good studies published on priorities of people with spastic paraplegia. So we decided to do our own survey. And to tell you about the survey, I wanted to invite Mikaira on the stage. She can tell us about this survey because she did this as a part of her master's thesis. This goes forward, this goes backward.
Hi, everybody. I'm really excited to talk to you about what we did. And so thank you, Dr. Sachdeva. And like he said, we really used Dr. Anderson's paper as inspiration because we wanted to know what was important to patients with HSP. And so we titled our survey, What Matters Most, to get patient perspectives. And so we distributed this survey in December of 2025. So it's been active for about 6 months with the help of the SPF. And so their collaboration really allowed us to reach out to you all so that we could hear what you all had to say. And we really do appreciate those of you that were able to participate. The information that I'm going to be describing today is from 136 responses. We just got about 40 more that we're excited to do some data analysis on. So it'll be interesting to see how and if that changes any of the things that I talk about today. And one of the first things we asked in our survey was, what symptoms do you currently experience as a form of your HSP? And so what we found was that the majority of patients reported that they had trouble walking and with gait problems, which was to be expected for us. We also saw that about 85 and 87% of respondents said that they were dealing with muscle stiffness and spasticity and ataxia. But what I also found to be really interesting was that more than half also reported physical fatigue, muscle weakness, and 70% also reported that they were dealing with urinary urgency. So that kind of goes back to what we were saying about some of those non-motor symptoms that people deal with and how we can start to incorporate those in the research and the clinical trials that we are designing. We also asked, what is your first priority of recovery, kind of like Dr. Anderson did in hers, and we found that 44% said that it was really important for them to regain leg function and movement, and about a quarter also said that they wanted to have better coordination and balance, which goes back to that 87% that was dealing with ataxia. And so in addition to understanding the priorities of recovery for patients, we wanted to know what barriers that they faced in clinical trial participation, since that was our ultimate goal. And so we found that this was split between the two. So 34% were saying that they were having transportation or travel difficulties, as well as not knowing enough information about current clinical trials and how to participate in things like that. And finally, we wanted to know what their biggest concern about clinical trial participation was. And we really saw that patients were kind of weighing the risk and benefits between clinical trial participation. And so some things about that their condition may get worse, that they might have new side effects or pain. We saw that most of them were concerned that the trial would not help them at all, as well as some worried about the time commitment. And so all of this was really understandable. I really want to emphasize yesterday that during our roundtable discussion, Dr. Fink mentioned that some of these priorities that we saw may differ between the type of mutation that people have. And so we are really excited to go back and, with that in mind, look to see how that differs between the mutation. But with all of the information that we were able to collect, we wanted to try our best to design a clinical trial that incorporated some of these aspects that we found. And so Dr. Pende will actually talk about some of the trial outcomes that we found from the clinical trial that we're currently doing.
Thank you, Makaida. As she was saying, I'm going to bring your attention to our HSP trial that Dr. Sachdeva was talking about. So we are measuring several outcomes, which is motor and non-motor. So just to begin with, we are measuring one of the tests that is time up and go test to measure the mobility, balance, and fall risk. We time the participants when he gets up from the chair, walks 3 meters towards the mark, and comes back, turns around, and sits back to the chair. Next, we measure 6-minute walk test to see aerobic capacity and endurance. The participants are asked to walk in a hallway, and we record the total distance that they walk during the 6 minutes. They can take rest if needed. We also measure knee strength, where we ask participants to kick out or kick in with their full force. And we measure the strength that they apply. You can see this in the video. So our team actually encourages the participants to kick with the full force. We also measure 3D gait. We analyze the 3D gait. We actually put 14 reflective markers on the joints of the participant, and we ask them to walk. High-speed infrared cameras capture these reflective markers during the real-time movement, and they build a precise digital model of movement, as you can see in this video. And we measure the force that they apply to the ground and the muscles that get activated during walking. We also do sit-to-stand test to measure the lower body strength, balance, and mobility with these markers on. During the 3D gait analysis, we ask participants to fold the hand, get up from the chair, and sit down without trying to look down. As we are saying, so some other outcomes we measure in our lab, where a clinician comes in and they ask the participants to lie down on the table. The clinician actually applies some specific movements and measure the hip, knee, and ankle muscle spasticity and score them according to the modified Ashworth scale. We also analyze disease severity and quality of life using questionnaires, SPRS and HSP SNAP. We measure bladder and bowel dysfunctions by using the certified and validated questionnaires. We are also collecting blood at baseline, mid-intervention, post-intervention, and during follow-up to measure the metabolomics and biomarker analysis. This work is done in collaboration with Dr. Bass and Dr. Pierre Marini at Drexel University. With this, I'll leave you guys with Dr. Sachdeva. Back to Dr. Sachdeva about our initial findings. Thank you.
Thank you, guys. So this is one of my last few slides. I just wanted to give you very, very preliminary findings from only one participant that we have collected 2 time points so far. And I'm really stressing on the word preliminary here because it's really preliminary. We are not trying to make any efficacy claims here. We're just trying to show that it's doable. We are trying to do this, and it's safe so far. So something happened to the text here. I don't know what happened there, but you've seen this diagram before. So based on the 6-minute walk test, as you can see, only 2 of the 4 time points are filled so far. the distance traveled by participant 1 in 6 minutes actually increased by tens of meters. The spastic paraplegia rating scale, SPRS, in this scale, the lower means improvement, higher means more dysfunction. In 4 weeks of stimulation, the SPRS score was also lower. The time up and go test was also the time taken to do that 3 meter walk and come back and sit on the chair again. That is the time that is recorded from the time up and go test. That was also progressively lower and lower, meaning the patient is faster and has more control and balance in getting up, going, taking a turn, and then coming back and sitting down. So that is also increased. What's interesting about this test is also it predicts fall risk for the patients with any neurological or motor impairments. And usually the line for cutoff for increased fall risk is around 12 to 14 seconds, depending on which literature you cite. So at the time of enrollment, he was above that cutoff line for increased fall risk, but now he's below the cutoff line for that. And this is the data from the spasticity assessment on the modified Ashworth score. And so the participant did not have a lot of spasticity to begin with, but at least in these 3 joints, this is again the effect of real-time stimulation. When the stimulation is present, the effect of spasticity, there was an effect on spasticity in this patient, and you can see the score went from 1 to 0, 0 meaning no spasticity, 1 meaning a little bit of spasticity based on that score. But he did not have a lot of spasticity to begin with. So this is my last slide. I just wanted to conclude by a few important points here. Hopefully I convinced you that spinal cord stimulation can promote recovery and maybe some long-lasting effects in various neurological conditions. That includes spinal cord injury and cerebral palsy, and that is all done non-invasively. Our hope is that it can potentially improve function in people with HSP, like you guys, and PLS in future. Right now, our early recruitment, we have N of 3 recruited so far. It shows feasibility for us. And as Makaira mentioned this, understanding consumer needs is important. So we are trying to be as inclusive as possible in terms of the outcome measures that we can measure in this early pilot study. So we are trying to measure not just motor outcomes, but also some of the non-motor outcomes. And lastly, we need much bigger, much larger study that we are trying to do right now, which will eventually lead us to have some claim on efficacy of this treatment in patients with spastic paraplegia. With that, I wanted to thank my lab. Vivek and Makaira are here. We have Jennifer, who's also a summer student in the lab, our research admin, Diana Sama. And all the fancy mechanics work that you saw was done in Dr. Michael Saman's lab, who's listed here. And Dr. Patel's lab is helping us with the blood draws and metabolomic studies. And the PM&R clinicians who are helping us do the spasticity assessments, Dr. Bass and Dr. Piermarini at Drexel, who are going to help us with the biomarker analysis. We'll be sending blood samples to them. And of course, to all the study participants and funding sources, and most importantly, Spastic Paraplegia Foundation for helping us being able to do this study in the pilot format. Thank you. I'm happy to take any questions if we have time.
I think I see my...
Yes, please.
Hang on a second. Okay, I saw your hand first. I'm coming. It's a big room, as you all well know. Here we go. Here, Doctor, our first question.
Is it possible to simulate this process at a physical therapist's office?
Absolutely. Yes.
May I ask what the specifics are?
You mean the specifics of stimulation?
Correct, yes.
So most of the specifics, the parameters are kind of already factory set in the device. I believe the stimulation frequency is 30 hertz. I can give you the details if you want in a writing.
Wonderful.
But in terms of, are you asking for levels?
Yes.
Level of stimulation?
Yes.
So we are trying to target the lumbar spinal cord, which is by bones, we go for about T11, T12 bone, and also the second electrode at L1, L2 bone. This is to cover the lumbar enlargement of the spinal cord, which is the area where the lower motor neurons are controlling the legs. So that's where we are targeting.
Wonderful. Thank you very much.
Of course.
Appreciate that.
Thank you. Any other questions? Okay. I see another one, doctor, right in front of you over here.
There's two here.
I'm working my way. I saw a hand here. I saw you first. That's how we do it.
How is fall risk measured again?
Sorry?
How is fall risk measured?
Good question. I would have to go back and look.
Okay.
We just started these studies, but I think it's based on a lot of statistics. That's all I can say right now. But what they have done is basically they have seen the cutoff for this TUG test, where if the patient is taking much longer to complete the test, what it means is they're taking much longer to get up, they're taking much longer to turn around and then come back and sit on the chair. So for context, this participant's video that you saw, he was a lot more independent. So his TUG test score, the time taken to complete that test, was about 16 seconds. It is above the cutoff, which is a higher risk for fall, but it's not a lot compared to other population. We have done one study in a almost same-aged woman with CP. Her initial time to complete this same test was 40 plus seconds. So she was taking 40 seconds to go and walk 3 meters and come back. And by the end of 8 weeks of trial, her reduced time was about 20 seconds. She was still above that fall risk, increased fall risk, compared to a normal able-bodied individual, which would be 5-6 seconds, who can quickly get up and go and come back. Right. But if I'm not saying you bring it under 12 and you've completely, got rid of the fall risk. It's just that the longer time you take, the more impaired you are, and you're more likely to fall in your daily activities. That's what it means, really.
Okay, thank you.
Mm-hmm.
Doctor, you got a question over here to your left?
Are you currently looking for more people for your pilot study?
100% yes.
Okay.
Any other questions? Any other hands? Okay, that's great. Great work, doctor. We appreciate your presentation. Really great stuff. Our next presentation is going to be all virtual, so it's going to be a couple of seconds for us to make sure that we get that all set up and the timing and everything, okay? Because, hey, it's not supposed to be like this, but we're ahead of schedule. But I do have one thing that you're going to be interested about tonight. We've got that big banquet in this room, okay? Remember, and we're going to have prime rib for dinner. So I hope you're a prime rib enjoying person. Not really sure what the verbiage is on that, but we're going to have that tonight. So be looking forward to that. Make sure you get your tickets. You get them in all the bowls that you need to do. You guys have over-delivered a lot of way. Those bowls are getting filled up. And you need to bring your paddles. You got to have your paddles tonight. So we're going to do that cool auctioning paddling thing, which is going to be an experience unto itself. So, well, we'll see how that one goes, all right? So we're going to have a really interesting, enjoyable night tonight. So everybody take just a quick couple of second stretch like Javier told you to, okay? Every time you get a chance, stretch like he taught you. And then we'll get on in just a few more minutes when we get the thumbs up from the satellite production crew. And probably a couple of more minutes, okay? So just hang loose. And then I'm getting mixed signals. It's great fun. So anyway, so when we get the word, we'll go. So just hang in there. We're almost there. We're going to take a 10-minute break, and we're shooting for about 2 o'clock, right? Fantastic. 2 o'clock is go time. Okay? So everybody take a break, and we'll be right back. Dun, 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 dun. Okay, you guys, we're getting close here. Okay, just trying to. It's okay. Don't worry about it. You'll see it when it pops up. But just reminder, we've got the big banquet tonight. We've got the auctions and all the raffle tickets. And remember, we're going to have some breakouts later. The men's, women's. Men in one room, women in another, and the youth group in another space. There's still time to do what you need to do in the other room. And there's a health and wellness going on over there. Has anybody seen Ray, the massage guy, doing the little hand massages? I encourage you. He's a deep tissue massage guy, and he's really good. He'll make your hands feel great. He actually does my wife Tina's deep tissue massages for her. And I'll tell you, this guy's got a thumb that can make you cry. Right across the room. And so, I mean, that's why I don't let him touch me. So he's too good at his job. Okay, everybody, we're going to do our introduction and we're going to get this show on the road. Throughout this conference, we have discussed infrastructure, biomarkers, trial readiness, and therapeutic development. Ultimately, however, every one of those efforts exists for a single purpose: to create better futures for patients and families. Our next presentation reminds us why scientific progress matters. For individuals and families affected by SPG 83 and other rare genetic conditions, every research advance represents another step towards understanding disease biology and identifying potential therapeutic opportunities. Research often moves forward one discovery at a time. What may seem like a small advance today can become the foundation for tomorrow's treatment. Please join me in welcoming Michael Pacold and Claire Miller presenting HOPE, SPG 83 Research Progress and Emerging Possibilities.
Thank you very much. Very greatly appreciate the introduction and the opportunity to present. We are more than happy to talk about HOPE, and particularly for SPG 83 and related diseases. But the work that we're doing actually starts much, much earlier, probably about 5-6 years ago at least, with a very unexpected neurodevelopmental journey tracing molecular oxygen that came out of my lab, and that has since been translated into the clinic through the efforts of Claire Miller and Juliette Garibaldi, whom we will who will be presenting. Dr. Miller will be presenting the second part of this talk. So these are my disclosures. The story that we'd like to present begins actually with a postdoctoral fellow, Robert Bann, who was in my lab, jointly mentored by Alec Kimmelman, who is now the dean and CEO of our institution, New York University Langone Medical Center. And the question that Robert asked actually is a much older question than that. It's about 300 years old, and it was first asked by this gentleman and his wife. So this is Antoine Laurent de Lavoisier and his wife, Marie-Anne. They were chemists. Antoine in particular was the chemist who really turned chemistry from alchemy into the modern quantitative discipline that we know of today. And his wife, Marie-Anne, designed many of the experiments, designed much of the apparatus, kept all the records, and is the reason why we know of his work. Antoine Lavoisier discovered oxygen, as one of the co-discoverers of oxygen. He also named it. And he was the first person to realize that oxygen was required for cellular respiration. He basically showed that a guinea pig consuming a certain amount of oxygen and a fire consuming the same amount of oxygen actually produced the same amount of heat. And from this, he concluded that respiration or metabolism was slow combustion. And this was in the late 1700s. And 300 years later, we know that he was right. Now, Lavoisier was also interested in figuring out where oxygen went in cells, but he didn't have the technology to discover this. And so when Robert came to my lab, again, about 300 years later, he asked the same question that Lavoisier did, which is, how do cells use molecular oxygen? Now, we all breathe oxygen. We know we need it to keep us alive. In fact, the most oxygen is used by the mitochondria, which are no strangers to this audience. These are used to produce energy in cells. This process also results in the production of reactive oxygen species that are important for signal transduction and oxidative stress. But importantly, there are a whole set of these oxygen-dependent enzymes, the dioxygenases, monooxygenases, and cyclooxygenases, that use oxygen directly as a substrate. And in order for these enzymes to work, they actually need molecular oxygen. And these are important for a whole host of cellular processes. And what Robert wanted to do was to move the study of these enzymes out of the test tube where they've been characterized extremely well and into living cells and organisms. So what Robert did was developed a technique for gaseous labeling, for tracing oxygen into cells. And suffice it to say that this was previously done, but not regularly. It's extremely expensive. Each cylinder of heavy oxygen is about $20,000. And prior to Robert's work, you needed 2 to 3 cylinders of oxygen for an experiment. So even the most generously funded investigator can't spend $60,000 on a single experiment. After we take the cells and expose them to heavy oxygen, we then extract the metabolites and look for shifts. The heavy oxygen is 2 daltons, 2 neutrons heavier. We can actually measure that difference using a device called a liquid chromatograph mass spectrometer, or LCMS. And that actually allowed us to find metabolites that are labeled by oxygen. We call this gaseous labeling in a sealed space. Now, using this technique, we found a whole bunch of oxygen-labeled metabolites, and we were very pleased to see some of these that are known to be labeled by oxygen, like hydroxyproline, and things that are made by reactive oxygen species, like methionine sulfoxide. But what really excited us was that the majority, in some cases, in some cell lines, of these metabolites that were labeled by oxygen were completely unknown in mammalian metabolism. In fact, the most highly labeled metabolite, or feature as we called it at the time, since we didn't know what it was, is extremely highly labeled, over 80% labeled by oxygen at physiological hypoxia, and even in cells growing in the extreme hypoxia of the tumor environment, where oxygen is 100-fold lower than what we're breathing, it's still labeled. And so Robert said, well, I need to follow this up. And I said, you're probably never going to find out what this metabolite is. And even when you figure out what it is, we still aren't going to know what makes it, and we don't know whether it's important. And Robert promptly ignored my advice and went straight for it. And I'm very glad he did. In about 2 weeks, he brought back data showing that he'd identified the metabolite. And after that, I had really no recourse but to say, look, you should go and work on this. What Robert figured out was that this metabolite was unknown in mammalian metabolism. We at first thought it was this metabolite called homogentisate because it has the same mass to charge ratio. But homogentisate and other metabolites are structural isomers. They have the same molecular weight, same mass to charge ratio. They just have different structures. And it turned out that this metabolite was 4-hydroxymandylate. You can actually see when you fragment this metabolite that it fragments like this. This is the unknown metabolite. The standard for 4-HMA looks like this. And now we can play the game of, does this look like this? Or does this look like this? This looks like this, and so the unknown metabolite is 4-hydroxymandylate. Now, to summarize a whole lot of work, Robert was able to show that this metabolite, 4-hydroxymandylate, is made by an enzyme called HPDL, 4-hydroxyphenyl pyruvate dioxygenase-like. This enzyme is found in the mitochondria. It makes 4-HMA. 4-HMA turns out to be the first committed intermediate in the mammalian coenzyme Q10 head group synthesis pathway. And again, CoQ10 is no stranger to this audience, but we'll get into that in more detail in a moment. Now, what's interesting is that we thought that the mammalian CoQ10 head group synthesis pathway had been figured out in the 60s and 70s. After all, that's how most metabolic pathways were figured out at that time. It turns out that it hadn't, and so this actually is the first committed step in the mammalian CoQ10 head group synthesis pathway. So a lot of questions arise from this. When do we need HPDL? Can we tolerate the loss of it? What organs require HPDL? When do they need it? Why do they need it? Now, in order to discuss this quickly, we need to understand what CoQ10 actually is, because if this is the first step in CoQ10, we can answer these questions by asking when and where do cells need CoQ10? So what is CoQ10? CoQ10 is actually a lipophilic antioxidant. It's got a long hydrophobic tail made out of 50 carbons, 10 isoprenoid units, so 5 carbons each. And it has a quinone head group, which is the reason why it's called Q. The Q is the quinone. The 10 refers to the 10 isoprenoid units in the tail. This head group is capable of accepting and donating single electrons. And in fact, this is the only endogenously produced lipophilic single electron carrier in our cells. Now, some form of quinone that's capable of handling single electrons in a lipophilic environment like a membrane is present in pretty much every kingdom of life. Hence the other name for these metabolites for coenzyme Q, ubiquinone, the ubiquitous quinone. You can find this in bacteria, you can find these in plants, you can find some variant of CoQ10 in pretty much every living organism. It turns out that CoQ10 is really important for mitochondrial function. You can't run the electron transport chain without it. And it's also very important for quenching reactive oxygen species that get into the membrane. So if it really is important for cellular and organismal function, then you should be able to figure out what the consequences are by studying patients and cells and mice that don't have the ability to make CoQ10. And as it turns out, there are a whole host of these rare genetic diseases called primary CoQ10 deficiencies that are caused by loss of the enzymes of CoQ10 head group and tail group and decorations of the head group of the quinone, of ubiquinone. So we know now that CoQ10 head group synthesis starts with TAT and then HPDL. That goes to this molecule called 4-hydroxybenzoate, and you'll hear more about 4-HB later in the talk. 4-HB is the immediate precursor of the quinone head group. It's conjugated to this tail, and then the head group gets modified to form the final bioactive CoQ10. And there are variants in every single one of these enzymes in the CoQ10 synthesis pathway. Now, how do these patients present? Well, you can imagine that if you don't have the ability to run your mitochondrial function very efficiently, that you would have loss of function of tissues that require mitochondrial function. So you would expect, for instance, loss of kidney function, loss of muscle function, loss of cardiac function, liver failure, and all these things happen. But what's really striking about the CoQ10, primary CoQ10 deficiencies is that almost all of these diseases are associated with neurological symptoms: encephalopathy, seizures, ataxia, and spastic paraplegia. Now, you would expect that, given that these are primary CoQ10 deficiencies, that you should just be able to treat these by treating with CoQ10. Now, the problem with that and after all, you can buy CoQ10 in pretty much any drugstore that you want, that when you walk in, you can buy it on. If you go into Costco, you'll see bottles of CoQ10 for sale on the shelf. The problem is that CoQ10 is actually pretty poorly bioavailable. And the way that we can conceptualize this is to look at these rules, the Lipinski's rules of 5, which are essentially criteria by which chemists describe what a drug looks like. The ideal drug, according to Lipinski rules, has 5 or fewer H bond acceptors, 10 or fewer hydrogen bond acceptors. Not yet, Michael, I'm presenting. If you want to watch me present, come, Michael, come here, Michael. Michael, please join. Michael, please. I was going to say my son is here and he wants to watch me present, so he may very well join us. So ideally, these molecules have a mass of less than 500 Daltons. Michael, shh. And a log P, which is a solubility in octanol and water, of less than 5. The CoQ10 passes the first 2 criteria, but it utterly fails the second 2. You can see that it's very large, and you can see that it has a log P of almost 20. Michael? Yeah, yes. And so, not surprisingly, CoQ10, when you try to give CoQ10, it doesn't get into the brain. You can give it even if you give it parenterally, it doesn't get into the brain. It doesn't, it can get into some of the other tissues, but its bioavailability is pretty abysmal. So about the time that we discovered that HPDL was responsible for primary CoQ10, the newest primary CoQ10 deficiency, we found 4 groups independently published that HPDL variants were associated with an ultra-rare pediatric neurodevelopmental disease. The first is called NEDSWAMA, which is severe and lethal. It's associated with seizures, spastic paralysis, brain white matter abnormalities, and it's progressive. And most of these kids don't live past the age of 2. There's an intermediary form that's progressive. And finally, hereditary spastic paraplegia 83, or spastic paraplegia group 83, which is associated with variants in HPDL, and which, while not lethal, is severely debilitating. Many of these children end up in wheelchairs. And so one of the groups that published these asked the question, could you recapitulate this disease by knocking this gene out in mice? And the answer is yes. You actually can knock out HPDL in mice. And what you can see is that HPDL null pups have lower body weight, they have smaller brains, and by day 13 to day 15, they die. So that led our lab to ask the question, could we restore CoQ10 head group synthesis in these mice? Specific in living organisms, first in mice and ideally in humans. And this is the work of Guangbin Shi and Sota Kuno in my lab. So the first thing that we needed was we needed a model of that recapitulated the symptoms of patients with HPDL deficiencies. And so what you can see here are a set of mice. These are HPDL null mice that we got from the MMRRC, which is the NIH-funded consortium that has pretty much all models of knockout mice. I'm going to run a video of these animals, and you can immediately see which animals are HPDL null. These two. These three are heterozygous litter mates, one wild-type gene missing a second copy of the gene. And you can see that these animals are lying on their sides. They're spastic. They have these repetitive movements. They actually have some degree of clonus and spasticity. And you can immediately tell what these mice, which mice lack the gene, and all of these mice don't survive past 15 days after birth. Now we can ask the question, if you lose HPDL, could you treat these mice with CoQ10? Or could you treat with the head groups? Could you treat with 4-HMA, or could you treat with 4-HB? In theory, you should be able to get the same result by giving any of these three. Now, the first thing that we wanted to do was to see whether or not we could get carbon from 4-HMA or 4-HB into these mice behind the blood-brain barrier. As I mentioned previously, CoQ10 doesn't really get through the blood-brain barrier and it doesn't get into the brain. But 4-HMA and 4-HB can be labeled. You can put carbon-13, which is heavy carbon, on the ring carbons, and in theory, all of that should end up in CoQ10 or in CoQ9, which is a dominant mouse form of coenzyme Q. And what you have here are wild-type, heterozygous, and null mice that were all treated with 4-HMA. You can see that in the wild type and in the heterozygous mice, not much of this makes it into the CoQ9, which is the mouse form of CoQ in the brain. But in the null mice that don't have HPDL, half of the CoQ comes from 4-HMA or from 4-HB, depending upon what you're feeding. And if you don't treat these mice with either 4-HMA or 4-HB, you don't get any labeling at all. And so that tells us that these head groups are capable of making it into the brains of these animals, making it through the blood-brain barrier, and being incorporated into coenzyme Q in the brains of HPDL null mice. Now we can ask the question, can we actually treat these animals with 4-HMA or with head groups? And so this is actually one of the first mice that we treated with 4-HMA. We treated this animal, not this animal. And you can see that both of these mice were small and spastic and had repetitive movements very early on at day 7. Now, 2 days later, treated, untreated, and heterozygotes. Now, let's fast forward 2 weeks. Can you spot the treated mouse? If you guess this one, you're right. Yeah, absolutely. So this is the treated mouse. So this is not a complete rescue. If you look at the gait of this animal, you'll see that its gait is not quite normal. I'll go back a little bit so you can see that the gait's not totally normal. And you'll also notice that this animal is small. It's runted relative to the other animals. So I need to make it very clear that at least in this model, under these conditions, this is not a complete rescue. You can actually see that the animal's size is smaller, and you can see that its gait is not completely normal. But it's alive. And the other animal died at day 14. All right, that's great. That's one mouse. But can we do this repetitively? The answer is yes. So these are HPDL null mice that we treated either with CoQ9 or CoQ10. We also treated them with idebenone, which is a soluble form of CoQ, and mitotempo, which is a mitochondrially targeted antioxidant. And you can see that all of these animals are dead by day 15. But if you treat with the head group intermediates, 4-HMA or 4-HB, you manage to get these animals to live to a year. At least 85% of them survive, and the rescue is very durable. We actually don't have to treat these animals after day 30, and it's not clear to us why that is the case. All right, great, we can do this, but does this actually have any effect on the mitochondria in these mice? So these are wild-type mitochondria from the cerebella of these mice. And what you can see is that when you knock out one copy, the mitochondria are a bit smaller, but when you knock out both copies of HPDL, the mitochondria are tiny and fragmented, and they really have no internal structures. And when you treat with 4-HMA, you get Christi back, you get the size of these mitochondria mostly back. And you can actually see a nice decrease of these in the size of the mitochondria in the cerebella of these mice. And again, it comes back up with treatment, not to the full wild type, but most of the way back there. Now, does this actually have an effect on the histology of the cerebellum? Well, again, this is a crowd that's very familiar with cerebellar histology, but what you can what I'll run over quickly is that the developing cerebellum has very specific architecture with a layer called the external granular layer and an internal granular layer made out of granule cell neurons or granule cell precursors, which ultimately become granule neurons. And these are the single most abundant class of neurons in the brain. At this point in development, they're dividing almost as fast as activated T cells. Anywhere they're dividing at basically between every 6 to 12 hours. And what you actually end up seeing is the migration of these cells from here to here, following a gradient of the sonic hedgehog made by the Purkinje or Purkinje cells here. Now, in wild type and HPDL null heterozygous animals, the cerebellum looks pretty normal, but in HPDL null animals, the cerebellum's tiny, it's fragmented, the granule cells are vacuolated, they're shattered, and all of the granule cells pile up in the EGL and don't make it into the final, into the internal granular layer. And when you treat with 4-HMA, you get most of the architecture of the cerebellum back. So this is all well and good in mice, but it still doesn't tell us when do we need HPDL and what cellular processes are completely reliant on CoQ10 head group synthesis. And it doesn't address the question of whether or not we can treat patients with HPDL deficiency with CoQ10 head group precursors. So I'd like to thank the people that funded this work. I'm going to and the people who did the work, particularly Sota and Guangbin. I'm actually going to hand it over now to Claire, who is going to talk about our experience treating a patient with CoQ10 head group intermediates. Claire?
Hello, and thank you so much for having me. And I want to also give a big thank you to my colleague, Giulietta Riboldi. And this is a once-in-a-lifetime story for me. So on the Friday, the week before Thanksgiving of 2023, Giulietta called me and she said, I have a patient with HPDL deficiency, and we think we may have a treatment for him. And I was able to learn about Michael Pacold's really strong and fascinating data, and was able to make the decision to go ahead and treat a patient, a first-in-human treatment trial with one of these head group intermediates. And so, are you still there, Michael?
Yep, just let me know when you want the next slide.
You can go on to the next slide. Michael has done a wonderful job sort of giving you what the role of oxygen is in the coenzyme Q synthesis pathway and what happens in mice. And I want to remind you before we go further what the phenotype in humans can look like. And so, first of all, HPDL deficiency was first described in around 2020, and likely it had been sort of captured under the umbrella of diagnoses of cerebral palsy before then. It has a phenotypic variability that correlates with age, and the presentations range from severe and life-threatening in infants to milder but profound in terms of their severity in adolescents and young adults. And so the mouse model recapitulates the infant presentation where you have profound disorder, spasticity, encephalopathy, likely seizures, and children and infants with this tend to die within the first few years of life, although with support, they can certainly live longer. There's a childhood presentation in early childhood years where children will have spasticity, seizures, cognitive decline, and neurodevelopmental delays, and this can be fatal as well. And then there's the later adult presentation or adolescent and young adult presentation where the primary feature is spasticity, and there aren't necessarily seizures or cognitive disability, although there also can be. But the spasticity can be debilitating, and many of these people end up requiring assistance for ambulation or wheelchairs. And you can go on to the next slide. So I want to tell you about our patient. And again, this sometimes the world aligns and you're able to do something really wonderful. And I think if Michael Pacold and his coworkers had not been making their discoveries in the few years before this boy's life would have been very different. And so our patient at the time that we met him was an 8-year-old boy. He had been born at full term and he had developed normally. He was starting 3rd grade. His parents were incredibly proud of him. He was great in school and he was on the soccer team, and they described him as one of the fastest players on the soccer team. And at that point, he had been really healthy. He had been, there had been no major illnesses, no hospitalizations or medications needed before this time, although he had had a growth spurt in the past year. The family history was notable for 2 younger siblings who were both deceased in infancy and had similar features. They had global delays, they had refractory seizures, and the 3rd sibling in the 3-sib set underwent a genetic workup and was shown to have pathogenic variants in HPDL. They never tested the 2nd sibling. So our patient was normal until the summer of his 8th year, when he was about 8.5. And his mom noticed that he was slightly off balance while playing soccer. He had trouble kicking the ball straight and his balance was a little bit off. And then late September 2023, his mother noted that his ankle had these strange jerks. It had clonus, which is a sign of upper motor neuron injury that correlates with spasticity. And they presented to the emergency room for workup at that time. He had an MRI of his brain and spine that were normal. He had blood work including creatinine kinase, which we check when people are having neuromuscular problems, and that was normal. And then they started a genetic workup, which, as you know, can take 4 to 6 weeks to result. And about 6 weeks later, in November of 2023, he was found to have the same heterozygous pathogenic variants in HPDL as his youngest sibling. His course progressed over that time. In October of that year, his soccer playing was severely impaired and he couldn't participate anymore. He was falling multiple times daily by the end of the month, and they tried to go trick-or-treating for Halloween and he wasn't able to walk those distances without falling. And then by November of 2023, he was still falling multiple times daily, and parents were noticing worsening of his condition on a weekly or biweekly basis. And by the end of November, he needed support to be able to stand. You can go on to the next slide. And so that's when we met him. And although sometimes animal models don't translate into human models, we felt that we needed to try treating him, and his parents very much wanted to try treating him. And so we designed a trial for hydroxybenzoate, which is our molecule of choice. It was more readily available to us than for HMA. And we didn't know what dose to use. We knew that it had a good safety profile because of some studies in mammals. And so we gave him a big dose. We rapidly up titrated to a dose of 100 milligrams per kilogram. We decided to treat him for 30 days to see what would happen. Remember that Michael's mice did not need a long course of treatment. We chose outcome measures. We decided to see if he would be able to walk 10 meters and at what pace. We did the 9-hole peg test, which is a measure of hand dexterity or ataxia. We used the spastic paraplegia rating scale, which is a measure of sort of quality of life depending on your degree of spasticity. And then we used the modified Ashworth score or scale, which is a measure of spasticity, which is somewhat subjective on the user or me as the physician judging his spasticity. Then we kept track of his neurological exam. We got another MRI of his brain, and we did videography, which I think is actually the most fun thing to look at. Next slide, please. So he did really well in this treatment because no human, except off-label for Michael Pacold, had tried 4-HB before this trial. We had to give it to him in a monitored setting. And so we gave it to him at our NYU research facility on weekdays, not weekends. And so the first 30 days of treatment were non-consecutive, Monday through Friday. And he got 100 milligrams per kilogram per day on the majority of those days. He did have emesis on 4 occasions, immediately after the rapid drinking of the full dose, which was 600 milliliters, but he's an 8-year-old boy and he wasn't used to drinking so much. And so we did what you do in pediatrics. We loosened his pants and we burped him a little bit, and he did better after that. Because of the success, which I will talk a little bit about later, we extended that treatment further through 60 days. And then because of that success, we ended up extending through 2 years. And this is actually out of date now, where we've extended through 5 years of treatment. And that leads us to another question, which is how long do we need to treat him anyway? And nobody knows the answer to that quite yet. So currently he's on a dose of 25 milligrams per kilogram per day. And again, I will show you how he's doing in the next slides, if you want to go on. So one of the best ways to understand the results is to kind of go through and describe how he was doing. So in November, before we began treatment, he could stand only with support. He was falling multiple times daily, and he would go up and down stairs on all fours. In January of 2024, he was able to walk for 20 minutes without falling, and he was only falling weekly, not necessarily every day. In February of 2024, he was starting to run, and remember, he was on the soccer team. He loves running. He loves going fast. In April of 2024, he was starting to ride a bike and falling rarely at that point. In August of 2024, he's stepping laterally to catch a ball. And if you have spastic hips, I challenge you to step laterally. It's not easy. He was able to balance briefly on each foot, which he was unable to do when treatment started. And he was able to walk up and down stairs without support, without holding onto the railing. In October of 2024, he was able to work the gas and brake pedal on a go-kart, which is, again, hard to do if you have spastic or spasticity at your ankles. And he was able to play flag football with his friends. In June of 2025, he was hiking up to 4 miles. In August of 2025, he ran 200 feet in 17 seconds. And in October of 2025, and continuing, he's very close to tandem walking with his feet just a few inches off midline. And he is able to jump around he can hop on each single leg. He can jump with both legs. He can dance. He can do all the things a kid wants to do at this point. And go on. In terms of our outcome measures, they did capture this described improvement, but there were some caveats. And so the Ashworth score, which is a measure of his spasticity, improved from 10 before treatment to 6. And specifically, he had decreased spasticity across his knees and hips. And that's where that improvement comes from. The spastic paraplegia scale, rating scale, which is, again, a measure both of quality of life, so strength, ability to go up and down stairs, endurance, as well as spasticity, with also a lower score being better, decreased from 13 before treatment to 5 with treatment, which is over a 50% improvement. And again, I think the description of his improvement mirrors that. Our other tests had some flaws ultimately. So the 9-hole peg test, which is a measure of dexterity and ataxia in your hands, he started off very slow for his age and he improved. But this is also a learned task and he is very motivated to go quickly. And so I think some of this maybe reflects some improvement in his hand dexterity, and some of it also reflects his personality and motivation. And then the 10-meter walk test was also it doesn't really give us a true understanding of his gait and its improvement, because in the beginning, the way he could stay upright was by walking really, really fast and letting his sort of propulsion keep him upright until he would run into a wall or fall over. And as time has gone on, he can actually walk more slowly and with a better gait with heel contact instead of toe contact. And so although we use walking speed, typically when we're thinking about the 10-meter walk, he was able to show that he could walk more slowly, and that was actually reflective of improvement for him. And next slide. This was an image of his brain, and he has a great looking brain. We have one from before treatment and from about 30 days of from 30 days of treatment, which is about 6 weeks because we weren't treating on weekends. There wasn't a substantial difference between the two. And next slide. And this is a video of him walking. And so we start before treatment, and you can see his hips, his knees, and his ankles. This is the classic scissoring gait with some compensatory upper arm movements. This is 6 weeks of treatment. And his heels are nearly touching the ground. His knees look more straight. He still has some compensatory and dyskinetic truncal movements and arm movements. This is a little over 2 months of treatment. And again, his gait compared to the first video looks much better. Those heels are getting close to the ground. This is about 4 months of treatment. And this is 8 months of treatment. And so his gait is not perfect. It's not normal. There are still features of spasticity, but it's much better. And this is him running, which is what he loves to do. And maybe just play that one more time. And so from the beginning, again, note how flexed his hips are, how his knees are turning in over each other like scissors. His ankles are can't touch the ground, or his heels can't touch the ground. And here this was a rapid improvement. It's harder to see the difference between the later videos than it is to see the difference in the beginning. It's easy to see the difference in the beginning. So this was a child who went from therapists telling him that he needed a wheelchair to not needing a wheelchair. And he's in 5th grade and school's going really well. Actually, he just finished, but. Okay, next slide. And then I think you added this in, Michael. So since we were able to publish our case, there's been another publication where a child with a primary CoQ2 variant was treated with actually a much higher dose of 4-HB with good safety margins. And so again, this seems like overall a promising treatment, both in terms of safety and efficacy. So thank you so much for listening. And I research and even medicine can be very frustrating, but this is such a wonderful example of how bench research, just in the nick of time, found something that could help a patient with a rapidly progressive course and make a huge difference in his life and his family's lives. So thank you for listening, and I hope that this gives you hope.
Are you going to take any questions?
More than happy to.
Okay. Anybody, Tim?
Yes, I'm right here. This is going to get interesting feedback sounding because we're listening to a Zoom in a big room. Anybody have a question for our presenters? Okay, nobody. Well, you guys learned a lot of great information, and you must have all taken wonderful notes because nobody raised a hand to ask a question. So that must mean that was a perfect presentation.
Well,
there we go. Now we got one. Here we go. Mm-hmm. It's on.
would we think it could work on other SPGs than just the 83?
I think that's one of
We couldn't hear it.
All right, let's try it again. Let's give it some heart.
There we go. All right, go deep. We think this was used for SPG 83. Do you think it could work with any of the other ones that have been identified that cause HSP?
So the question is, can this treatment be used for other hereditary spastic paraplegias? And the problem is they have different molecular mechanisms. And so the biochemistry of this hereditary spastic plegia, aka number 83, depends on the breaking of HPDL. And because we know that it's broken, we can restore the downstream signaling. The other hereditary spastic paraplegias have different genes involved and don't necessarily even reflect mitochondrial dysfunction. And so, unfortunately, no, this is very specific to HSP83, but it does bear relevance for other primary CoQ10 deficiencies. So other diseases that have the same pathway involved rather than other hereditary spastic paraplegias.
Yep, I would definitely follow up with agreeing with that. We have the strongest evidence for this being effective in HPDL deficiency and in primary CoQ10 deficiencies, specifically CoQ2 deficiency, where there are defects in the transport of the head group into the mitochondria, and where you can give high doses to overcome that. The question about the other hereditary spastic paraplegias is there evidence that there is loss of CoQ10 in those as part of the pathology? If so, does that contribute to the pathology and the symptoms of the disease? And then the third question is, if that loss of CoQ10 contributes to the symptoms of the disease, could head group supplementation improve that? The problem right now is that we don't have evidence for any of those three hypotheses, although we're actively working on this. Not so much in the hereditary spastic paraplegias, but in other secondary CoQ10 deficiencies. And that's, the jury's still very much out on that. If we're able to show that this is effective in other secondary CoQ10 deficiencies, like statin use, for example, then I think that we would want to revisit that question, because then we could ask in other hereditary spastic paraplegias, are people taking CoQ10? Is there evidence that there's even slight symptomatic benefit? Is there evidence that CoQ10 is down in these diseases? Does that contribute to the symptoms? And if so, then we can go after it.
Thanks, doctors. Any other questions? Sorry? Any other questions? Okay. No more questions from the room here. Norma, do you have anything to say?
No, you answered that question, Mike, and I appreciate that very much. So we are going to be trying to get back with you and see what other mitochondria dysfunctional SPG numbers that this might benefit. Is that a possibility?
Absolutely. If you bring those to me, I'm more than happy to take a look at them, try to put those models in our lab, and see whether or not our head groups can do anything for them.
Hang on, we can't hear you. Something happened to our audio, so hang on just a minute. Okay, did you hear my question, Michael?
Yes.
Okay,
is that broadcast? Should I answer it again? Should I answer it again?
Yes, please answer it again.
Yes. So the question was, are there any other hereditary spastic paraplegias that this might benefit, specifically those that have mitochondrial dysfunction? And the answer is yes. If there are hereditary spastic paraplegias that are associated with mitochondrial dysfunction, we'd be very happy to take fibroblasts, any other models that there are of these diseases, try to see whether or not we can get those into the lab, treat those with the head groups, and see if there's any improvement. Again, there we would not be treating the underlying pathology, but even if we can modify the trajectory of the disease or attenuate the symptoms, I think we would consider that a win.
Well, thank you so much for answering that, because in the paper, in the welcome bag that we shared with all the patients here, that there is a report that was done by Dr. Giovanni Stavani.
Yep.
And on the back of that, it talks about the SPG numbers that may have mitochondria dysfunction.
Oh my goodness. I'd love to see that paper. If you have a
Yes, sir. So
if you have a
just
copy of it,
for the
happy to take a
record,
look at it.
just for the record, it talks about mitochondria dysfunction. It's got SPG 83 at the bottom, but at the top, it's got SPG 9 and 7 and 13 and 31 and 20 and 28, 43, 55, 74, 77, and 78 and 83. So this could be something that might be beneficial to those people that have this mitochondria dysfunction. We might want to talk more about that.
And we would be very happy to follow up on that. And should this work, we would benefit from your foundation's expertise in mobilizing patients, particularly if we ever run a clinical trial on this, which we're working very hard to get off the ground.
Well, have you guys thought about or considering maybe a basket trial?
Not yet, but send us the paper, and we're always more than happy to look at that.
I will do that. Thank you so very much for your time. I greatly appreciate both of you.
Our pleasure.
Tim, any other questions?
No more in the room, and it was a great presentation, and we're looking forward to the future.
Absolutely.
Mike.
Please give us feedback on this. Anything that wasn't clear, anything that wasn't understandable, more than happy to try to explain that.
That sounds great. I'll get back to
So guys,
back and forth.
You okay, Norma? All good. All right, we're going to get set for our next speaker. We just, want to remind you, we've got a really big, busy afternoon. Remember, we got breakout rooms a little bit later for y'all to be a part of. We got the big banquet in here, so I'm going to tell you now that when we do finish about 5 o'clock, we have to empty everybody and everything out of the room. If you brought it, take it with you, because we're going to have the staff is going to prepare the room for the dinner tonight, okay? And we'll give you some more notes a little bit later, but just to remind you, when we do finally finish and go to breakouts, you got to take your stuff with you and stuff. People, you leave stuff on tables and stuff. It's okay. You're welcome to do that. Everybody lands in the same spot, but clear all the stuff you want to take with you, and because they're going to clean it all off, and they have to put down plates and napkins and water cups and a whole bunch of other kind of stuff that they do to make a dinner. So just want to remind you about that. I think our next presenter is ready to go. We are right on. We're actually a little bit ahead of schedule, which is really good, and I like that. So I am going to introduce our next speaker. And by the way, his bio is in your program, okay? That program that you got is full of a ton of information on all our speakers that we've had today. So take some time and really read it and get to know these individuals and where they come from and what they've done in order to be here for you today. They have worked a lifetime to produce a lot of great stuff for you guys, and their bios are really great, and they take better pictures than I do. So that's why I'm not, mine's in a corner down way in the bottom. Don't look at me. Anyway, here we go. One of the goals of this conference is to recognize that patients cannot place their lives on hold while waiting for therapies. While the scientific community works to build future treatments, individuals and families continue searching for ways to optimize health, function, energy, resilience, and overall well-being. Our next speaker encourages us to think broadly about health and the many factors that may influence it. Whether you agree with every perspective presented or not, SPF believes patients deserve access to information that helps them make informed decisions about their own health journeys. Please join me in welcoming Eric Nepute, presenting The 7 Stages of Disconnection, the truth about health no one ever explained to you. Yay. Dr. Eric.
Thank you. Thank you for that.
You're the best.
Yeah,
Thank you,
appreciate
sir.
you guys.
There you go.
Good afternoon. How's the conference going so far? Yeah, good stuff? Okay. So first of all, like they mentioned, I think it's amazing. I was listening to some of the previous conversations about mitochondrial health and mitochondrial dysfunction. That's actually my wheelhouse. That and genetics is what we do the most. But I'm a little odd because I'm actually still practicing physician. I literally left one of my practices, came here with my scrubs on, threw a jacket on to come present to you, and I've got another 100, 125 patients I see when I get done today. So I love the fact that we get to exchange some information of knowledge that you guys are getting with some of these amazing PhDs and et cetera. And by the way, a huge shout out to the men and women who put this together because this is a very unique and quite honestly a rare opportunity to be able to come and gather and seek knowledge where you may not be able to get it in your own backyard. Would you agree? This is a powerful tool. So let me give you, yes, it is. So let me give you a little background on myself before we get into this. So just like the gentleman who introduced me said, my name is Dr. Eric Nepute. I practice here in St. Louis, Missouri. Have a couple practices in a couple areas of the country as well, Texas and Florida. We do a lot of work. I've got some clinics in Japan, Dubai, and Saudi Arabia as well. We do a lot of work way outside the box. And I'm gonna encourage you to listen to me today, not just with an open mind, but with an open set of ears and an open heart. Listen, the information I'm going to share with you today can and will change your life if you'll let it. And it's not some amazing magic cure or some magic peptide, some of the amazing research you're seeing. Sometimes we just got to get down to the basics and fundamentals of what health really is. And how many of you would agree that most of us don't really understand what health is? And listen, in my offices this afternoon, in the last 18 months, we've got a paper that we just published on this. We've had 127 patients that have had neurocognitive decline, nerve damage, functional nerve issues that we've able to show complete reversal of in the last 18 months. And I don't tell you that to impress you, but to impress upon you that we have some amazing advancements that are happening in clinical practices all across this amazing country. And it's great to be able to come to a venue like this to share that. Now, what I want to tell you is this, is what we're going to discuss today. What I'm going to discuss today is I want to talk to you about a concept that our group came up with about 2 decades ago. And it's helping people get to the root cause of their problems. We're hearing presenters were speaking about mitochondrial dysfunction. How many of you have heard mitochondrial dysfunction before, that term? How many of you know that that's something that we've been looking at and talking about for a long time? But the reality of it is that if you don't have certain balances in your body, which we're going to discuss today, you cannot get the mitochondria to work and repair and regenerate like it's supposed to. Everything is mitochondrial regeneration. All healing is mitochondrial regeneration. Your mitochondria needs to have certain nutrients, certain nerve inputs, and etc. And if I do a good job today, I'm going to help you get a better understanding of that. And I'm also going to give all of you access to this presentation so you can have that, because I'm going to share a lot of info today with you. And then tomorrow, I'm coming back and presenting on a topic that we're very, very, very dear to my heart, and it's about toxicity and toxemia. And that may not sound exciting, but that's so important. I don't know if you know this, our government just invested $144 million into what's known as the war on microplastics. Have you heard about this? You know, these microplastics are some major causes of mitochondrial dysfunction. Well, if your mitochondria are not functioning, your cells are not functioning. If your cells are not functioning, your organs are not functioning. Your nervous system in your brain runs off of this. And if you've got things that are gumming them up and causing problems, you need to know about that and what you can do with it. So tomorrow I'm going to share that with you as well. So let's talk about what we discussed. We call this the biggest missing link in healthcare. We talk about the 7 stages of disconnection, which many of the physicians in this room all studied out of a textbook called Guyton's Physiology book. And Guyton's Physiology textbook talks about the 7 stages of disease. Well, the 7 stages of disease, many of you have heard of diseases. It takes a process for your body to get there. And therefore, it takes a process to reverse it. What we do in our practices across the globe, and people come from all over the planet to see us do this, is we measure the physiology of the body to find what stage of disconnection you're in. And then once you find that out, then you can actually find a portal of entry to help yourself or your patients get their bodies to heal. Now, I'm going to tell you one of the common sense conversations you're going to hear me talking about during all my talks is, listen, self-care is the real healthcare. The more you learn how to take care of yourself, the less you're going to be reliant on others to take care of you. So the more you know how your body works, genetic testing, all these things that are available, that is a very powerful tool. And the world that we're living in with AI and all this ability to have our access to all of our data on our phones, there's literally conversations happening now. I'm part of a group of doctors and scientists across the globe that have started a project called The Helm, which gives you back, we're putting you back in the pilot seat, where you can literally take and download all of your test results, your scans, your images, run this through our supercomputers, and it helps you narrow down where you're at in these 7 stages and how to reverse it. So let's talk a little bit about health. We're going to talk about health today a little bit. I'm going to share some interesting things with you, but before we do that, we have to get a definition of what health is. Now, how many of you know that health is more than just looking good and feeling good? How many of you know people that look good and felt good and dropped dead of a heart attack? We all know people like that, okay? So health is not just looking good, it's all about functional health. And that's what our primary practices are in functional medicine and functional neuro, which is what I primarily specialize in. It's all about understanding that health is all about a balance. It's a balance of your body's systems, and not just the nervous system, the integumentary system, the GI system. It's about the cellular systems in the body. It's about your body's ability to adapt and overcome and to be able to repair from the damage that it has. The last speaker was talking about mitochondrial damage. You know, one of the issues with mitochondria, your body is not healthy if it cannot repair from damage to the mitochondria. And the reality of it is that most people look at health from a whole different aspect. We talk about health the way Guyton's textbook of physiology talks about it. When I learned about it, when I was going through my osteopathic training, was health is a balance of 3 things. It's a balance of your physical well-being. You know as well as I do, if the body's not moving like it's supposed to, the brain and the organs can't move like it's supposed to. There's been research that's proven that the number 1 nutrient for the brain is movement of the body. Well, if the body physically is out of balance, we have problems with that. If the body is neurologically out of balance, we have problems with that. And emotions, how many of you have ever been emotionally stressed out before and you feel it in your body? How many of you ever experienced that before? All of us have. By the way, if you don't raise your hand, I'm assuming you've got frozen shoulder. We can probably help you fix that after the seminar, okay? I like to try to have fun when I do these talks because I think it's important to engage people because there are opportunities out there. And if you're just sitting here reading your slides and just telling you about the data and the research and not making it humanized, it's not okay. So nutrition, how many of you have seen the advanced therapeutics that are out there with peptides and other nutraceuticals that are out there that are showing major changes in mitochondria and nerve health? Well, there's a lot of that out there that I want to discuss, and we're going to discuss a lot of that tomorrow. Today, I'm going to kind of tee up the idea of what health is, because how can you possibly get healthy if you don't know what health is? You see, I just did this workshop very similar to this a couple months ago. We had a couple thousand people in a stadium I was speaking at, and I asked the question, what does it mean to be healthy? And I got all kinds of answers. Some people said, well, I look good, pain-free, no medicine. I had one lady stand up and say, my husband, my kids, my life, just the stress. I'm like, hold on, I'm Dr. Eric, not Dr. Phil. But she was talking about the emotional stresses in her life, because we know that if you have an imbalance in your body emotionally, that can downregulate your nervous system and increase your symptoms. You've all experienced that before. So health is not just about the absence of symptoms. It's about your body moving to ease from disease. And this is what I want you to understand. Years ago, I'm and just like a lot of the other physicians in here, we're young enough but also old enough to remember when they told us that certain conditions like degenerative disc disease couldn't be reversed, or that once the brain is set a certain way, it'll never change. Well, neuroplasticity completely blew that out of the water. I want you to be aware that we have so many advancements in science, in longevity, in nutrition, in peptides, in brain health, in nerve regeneration, that you need to be paying attention to these things, because a lot of times, unfortunately, your physicians aren't. And I'm going to tell you, most of you in this room, I know how hard it is to find qualified physicians to deal with a lot of the conditions that you're dealing with. But most physicians get stuck in what they learned in medical school. Most of them don't keep up with the new data. Most of them don't keep up with what's going on. We now know, I don't know if you've seen this, but some of the latest data and research has clearly said if a human being can live to the year 2030, which is only 4 years away if you think about that. It's pretty wild if you think about that. If you can live to the year 2030, 120, 130 years of life or more is not out of the question. And many of you are thinking, I don't know if I want to live that long. But the reality is that we now have an ability to get into the system, into the body, and help reverse a lot of the damage on a cellular and even a genetic standpoint. So let's talk a little bit about this. How many of you have ever heard of the word ease before? You ever heard someone say, I'm kind of at ease? You know what that means? That's the Greek word for balance. You see, when your body's in balance physically, chemically, and neuroemotionally, you're at a state of ease. Well, what happens is when you get out of balance physically, chemically, or emotionally, which, by the way, you can get out of balance physically by having a car accident, by sitting too long, by picking something up and hurting yourself, by not moving enough. There's a lot of things that can cause physical imbalances, causing your body to be in a state of now dis-ease. You ever heard of that term, dis-ease, before? You see, dis-ease comes before disease does. But see, in my conventional training, I was only taught to work on disease. If you don't have a disease, how can I help you fix your problem? When now the reality is in the functional world of functional medicine, functional neurology, and longevity, it's all about why did you develop the disease? And so we now have the ability to do that. So what this whole conversation I want to introduce you to today is helping you go from a state of disease back to a state of ease. So just keep that in mind, because I'm telling you, this conversation you've probably not been exposed to. I presented the first time on this topic in front of my colleagues at the American Academy of Anti-Aging a year and a half ago, and it was received with huge, huge accolades, but it was also received as, what do we do now? Because it's a complete paradigm shift. But how many of you would agree the definition of an insanity is doing the same thing again and again and expecting a different result? How many of you are kind of sick and tired of that whole deal? There's so many options out there, and coming to conferences like this gives you an opportunity to have exposure to that so that you can be your own advocate. That's what you need to make sure that you understand. You need to be your advocate. Go check out the vendors that are in the vending booths. You need to speak with the physicians that are here. Speak with other advocates that are doing things that might be different than you're doing, and all those things are helping them go from a state of disease to a state of ease again. So let's talk a little bit about what most people get wrong when they're treating their problems. You see, a lot of times you'll go to your physician because you have a symptom. You guys all know what symptoms are: fatigue, anxiety, upset stomach, muscle spasms, inflammation, pain. These are all symptoms. Well, you guys know what we talk about symptoms now. Symptoms are just like your check engine light going off in your car telling you there's a problem. And most people have two choices. Well, I guess really three. If your check engine light's going off in your car, you can either, number one, ignore it like a lot of folks do and just kind of deal with it. Or what other people do, they put a piece of tape over it or a picture of their kids or grandkids or dog or something like that. You're giggling when I say that, but a lot of you are covering up your symptoms of digestive issues, of chronic pain, of depression, anxiety, insomnia by taking certain narcotics that aren't helping you get to the root. Now, narcotics are important, and we use and recommend those as a crutch if we can help get to the root of the problems and reverse that, because a lot of the symptoms people deal with are pain, fatigue, brain fog, poor sleep, hormone imbalances, and inflammation. How many of you know anybody that's dealing with any of those issues? Well, if you'll pay attention, I'm going to share with you some cool things that you can do to help with that. And at the end of this, I'm going to share with you what we like to call, we refer to it in our world, the ultimate feel great system. It's basically the day in the life of a human being that can optimize and maximize what we call metabolic flexibility. And I don't know if you know this, but we now in the functional neuro world have collided with the functional medicine world, finding out that a lot of the chronic issues that people have, neurocognitive issues, inflammatory nerve diseases, demyelinating and damaging diseases, are because mitochondria in the metabolism isn't working. So when we talk about the 7 stages of disconnection, we also refer to the 7 stages of disease. And this is the hierarchy that most of my colleagues, most of the physicians we work with are used to seeing and hearing this. And I'm going to explain to you what happens at every step along the way. And if you understand what's happening at every step along the way, then you might be able to do some things to figure out what stage of disconnection or disease you're in. And then some action steps you can take to hopefully reverse that. And there are 7 of those stages. So what we talk about, this is a poster of the 7 stages of disconnection, and it starts with stage 1. Now, let me ask you a question. Many of you probably know this. You know that you have about 47 trillion cells in your body. Did you know that? Around 40, how'd you like to be the guy that had to count those? 47 trillion cells that we know of. Now, what's interesting is every one of those cells acts like a battery. You see, all those cells make up your tissues and your organs, and they make up you. They make up your brain, your nervous tissue, your gut tissue. Everything is made from that. And if those 47 cells have a problem to them, for example, if the voltage was turned down on those cells, like if the dimmer switch turned down in this room, those cells wouldn't function as well as they should. Would you guys agree with that? That's mitochondrial dysfunction in a nutshell. What if I told you that there were ways that you could not only turn up the mitochondria, but you could increase the mitochondria? You can increase mitochondrial production in different cells, in different ways, with different modalities. But see what happens, the loss of the cells, we're gonna go through all 7 of these, but when you lose the voltage, what happens is your cells lose their communication. Did you know that every cell in your body communicates with every cell in your body? Your brain tells your legs to move. Your leg tells your brain, I moved. Your bowels tell your brain it's time to go to the bathroom. If there's disconnection there, what happens? Your brain and body doesn't get the input. That happens on a cellular basis. Does that make sense? And the only way that that can happen is if the voltage of your cells is turned in the way that it's supposed to. Now, did you know, you've probably heard this as well, that your body regenerates itself? You cut yourself, the Band-Aid that you put on it isn't what fixes the cut. Your body does that. You know, you get a new liver every 12 to 14 months. Your GI tract replicates itself every 7 to 14 days. Your nerves regenerate themselves every 6 to 8 months. Our body's in a constant state of replication. But if your cell voltage is too low, and we can measure this now. Up until around 5 years ago, to measure something like this would cost someone thousands of dollars, and you could only do it at university research hospitals. Now we have the ability as physicians to do these in our practices. We can literally measure the voltage of the cells in your body. And if they're low, there are things that can be done to turn those up. So could you imagine if the voltage was low, messing up the communication between your brain and your feet? It'd be kind of hard to make that work like it was supposed to, wouldn't it? So just be thinking about that as we go through this process. So let me go through step by step of these 7 stages, and then keep any questions in mind that you've got, and we'll do our best to get through those. So the first thing that happens when your body gets out of balance, when you start to get in a state of stress that your body has trouble adapting to, the first stage of disconnection or disease is loss of cell voltage. This looks like and feels like occasional fatigue, occasional brain fog. Every once in a while, I feel like, Every once in a while, my GI tract bothers me. Every once in a while, I might get some numbness and tingling. Every once in a while, it's not a constant state. But this is a very huge component. This is the first domino that falls. So if you lose your voltage, think about those 47 trillion cells in your body. They all work like a pump. They all have a pump. That pump is powered by the battery. It's called cellular respiration. And those cells are like sponges. When they contract, they squeeze out the toxins. And when they relax, they open up and allow nutrients to come in. Now, there's an electrical component to that, but how many of you in here know people that like take supplements or medications or eat food? Is that anybody in here? Okay, good. Just wanted to make sure you guys are still with me. When you eat food, that food breaks down into very small, microscopic components, and those little nutrients, vitamins, minerals, herbs, fats, proteins, and carbohydrates have to get inside the engine somehow. The engine is the cellular mitochondria. If the respiration doesn't pump, guess what? It can't get in, and the toxins can't get out. We're going to really lay heavy into this because I'm going to tell you, in my experience, 25 years of doing this kind of work with folks with all kinds of neurological and chronic internal health issues, unless we get the voltage turned back up, a lot of the advanced therapies that we may do, stem cell therapies, brain stimulation, peptide therapies, will not work because we don't have the roots where they need to be. You guys tracking me on that? This is a very important step because if you've not ever had your cells measured or voltage looked at, this is something that you need to speak to your providers about. And I'm going to give you this presentation so that you can share this with your providers. So when we get low voltage, we get poor cell communication. We get chronic low energy. We get impaired cellular respiration. We get slow healing in the body. Does anybody know anybody that heals real slow? It's because the voltage is turned down. We start to get issues with impaired respiration of the cells. We get declined function across multiple systems in the system, and it's all because the cells need to be charged back up. I was explaining this to a group of physicians at Wash U about a year ago, and one of the doctors came up afterwards and said, your conversation reminded me of my Tesla, his Tesla car. He goes, all we need is human Tesla charging stations to charge up these voltages and we're good. Well, there are therapies out there like post-electromagnetic therapy. There are therapies like certain peptides that we know they can stimulate mitochondria recharging. There are certain deficiencies in certain types of B vitamins, and there are genetic imbalances in the methylation detoxification pathways that many human beings deal with that causes them to be more prone to loss of cell voltage. And so when you turn the voltage up, the body heals. But if you don't turn on the voltage and the cells do not continue to respirate, we go to stage 2. The second domino falls. And the second domino is loss of communication. This is when the pumps that transport the system's toxins out, nutrients in, it's when they become toxic and deficient. I'm going to show you tomorrow how this happens. I'm going to show you also tomorrow how to get this out of the body. I'm going to give you some action steps tomorrow of things that you can be doing. In fact, one of the studies that we published, actually we published this, and I was in DC about a month ago, and we were talking about it in front of the Maha group, and we were able to show them that we can actually reduce some of the microplastics in the body by using some of these techniques that are very simple. Some of them are completely free to do by up to 50% in 7 days. There's a lot of things you can do to clear the clutter to help get that communication to come back. So if the cells aren't communicating, then the brain and the body's not talking like it's supposed to, and we can't control either one. And we see a lot of that in our practices these days, especially with a lot of the conditions that many of you are dealing with on a regular basis. So nutrients, how many of you right here know people that you're get up and go, got up and went? You're just exhausted all the time. You could sleep 14 hours a day, go on a 3-day weekend, and wake up Monday like, nope, I got nothing left. That's a phase 1, phase 2 problem. And I'm telling you, this is treatable, but it's a lifestyle change that must be done. Because if you keep doing what you've always done, you're going to keep getting what you've always gotten, period. So if we don't fix stage 2, this is when the nutrient delivery system fails. This is when we start wasting muscles a little bit. And then the next thing that happens is our microbial system, our terrain in our body gets messed up. Now, how many of you How many of you know that you're supposed to have healthy microbes in and on your body? Did you know that? Did you know that, there's 47 trillion human cells in and on the body? 47 trillion. You know there's over 100 trillion bacteria cells? 100 trillion. And some of those cells in your GI tract, I'm telling you, over the next 3 years, you're going to be seeing a lot of this because there's a tremendous amount of work that's coming out about microbiome and microbial health. We've now identified bacteria that if you don't have these bacteria in your mouth, you got a higher risk of Parkinson's and dementia. We've identified if you don't have certain bacteria in your GI tract, that you have a higher risk of neurodegenerative diseases, ALS, Lou Gehrig's disease, and other problems. These bacteria tell our cells what to do, and these bacteria turn the voltage up on the cells, and they wake up certain types of genetics. Your DNA is not your destiny, but your DNA, if you don't change the lifestyle, we always say your DNA loads the gun, but your lifestyle pulls the trigger. And if you lose your cell voltage, your pumps stop pumping, and things can't flow like they're supposed to. Well, we're not too far from the Mississippi River. And what would happen if I took if I dammed up that river? What would happen upstream from that river? It would turn into a swamp, wouldn't it? Because things can't flow. Well, when your gut microbiome gets messed up, this is when we start to get not just poor digestion, but we start to get imbalances in a nerve in the body called the vagus nerve. How many of you have ever heard of that before? The vagus nerve is a very, very important nerve. It's called the gut-brain connecting nerve. 80% of your vagus nerve lives in this region of the body, 80%. 20% lives up in this region of the body. And if you have imbalances in the gut microbiome, not only can that cause bloatiness, distension, gas, poor absorption, but it causes the rest of your cells not to function like it's supposed to. How many of you have ever heard physicians or anyone make the statement that inflammation is the root of all illnesses? You've ever heard of that before? A lot of people will say that. It's actually the fourth root of the problems, and we're going to talk about that in a minute. But did you know inflammation goes up because inflammation is a protective mechanism that the human body actually uses to try to reverse the problems that was caused by low voltage and lack of respiration. It's trying to burn off the toxicities in the body. It's trying to kill off some of the bad bugs. Did you know that if you get an overgrowth of bad bacteria, candida yeast, and even some parasitic issues, that can mimic a lot of the signs and symptoms of neurological decline, neurological demyelinization, all kinds of other symptoms can happen from that. So the gut microbiome is one of the most important things. How many of you have ever had your gut microbiome checked and measured to see what's going on with it? Not many of you have. If this is one of the most important parts of real health, why have you not had this looked at, and why have you not had it managed or treated? You see, most of us are looking for the home run. We're looking for the magic cure, the magic peptide, the magic, genetic change to fix whatever diseases we have. But we forget that our body has different stages of disconnection, that if we don't deal with those, the therapy that we're doing to fix the problem, the big therapy, won't work. You guys tracking me on that? Does that make sense a little bit? Because this is what we're finding in clinical practice. People are spending tens of thousands of dollars, and some people even more than that, going different places around the world promising cures, when the reality of it is why do those things work for some? Why do those things work for some, but not for everyone? It's because the ones that it worked for had more balance than they had disconnection. So the gut microbiome is a very fascinating and rapidly emerging conversation in the functional medicine, in the paralysis, in the neurological, and in the functional neuro world, because we know that if the bacteria is off, the body cannot heal like it's supposed to. And an imbalanced microbiome leads to multi-system failure. The liver gets congested. We start getting problems with insulin and insulin resistance, which is a very big issue with neurological demyelinization. If you don't have a balance in insulin, and we're going to talk a lot about that tomorrow, you cannot heal nerves quickly. Anybody in here know anybody who's a diabetic? Somebody who's a diabetic cuts their leg, it takes them weeks to heal instead of days to heal. That's on the outside.
and you're trying to get nerves to function, nerves to remyelinate, muscles to work again, it's all about the gut microbiome and the terrain. Well, then what happens if you don't fix stage number 3 is you move into stage number 4. This is inflammation. Now, please hear me on this. Inflammation is our friend in short term. The problem is, and this is a very interesting conversation. I'm speaking at an event in Norway on this topic, on the topic of inflammation. And it's not the inflammation that's the problem. It's the chronic inflammation that's the problem. Now, I'll show you this tomorrow, but just visualize a cell. You guys have all seen a picture of what your cells look like in your body. Like, just think about like a little circle. Well, the outside circle, the cell wall, you know what that's made of? Fat. You know, we've been told for a long time since Fabio came out and said, I can't believe it's not butter. We've been told that fat is bad for us, right? We now know that we need fat, but we need the right kind of fat and we need the right ratio. How many of you have ever heard of something called omega-3s before? Yeah. How many of you have heard of something called omega-6s? Omega-6s, our body actually makes a little bit of omega-6s on its own. It is the inflammatory marker to turn the body on. The problem is if we overconsume too much omega-6, which is about 70% of the American diet, we get an imbalance of our omega-6s to our omega-3s. Now, let me explain that, then I'm going to come back to the importance of omega-3s in the system. Omega-6s are pro-inflammatory, and if they're too high, our inflammation stays on. And if the inflammation stays on, it actually causes more harm than good. You never get repairs of the nerves or the tissues. It's just the fire's too hot. So in medicine, we measure your omega-6 to omega-3 ratio. Write this down or remember this. This is probably one of the most important conversations that you could have with your provider. What is my omega-6 to my omega-3 ratio? If you want your brain and your nerves to work proper, you've got to have a good omega-6 to omega-3 ratio. If you want to fix neuropathy, if you want to get muscles to work better, nerves to work better, your brain to work better, get cognitive decline to get better, you have to have a good balance of omega-6 to omega-3s. Our group has done over 7 million tests of omega-6 to omega-3 ratios across the globe. A healthy omega-6 to omega-3 ratio is typically a 4 to 1, a 4 to 1. A 4 to 1, okay? When it's a 4 to 1, we have neurological plasticity, our body can regenerate, our cells can regenerate, our mitochondria can repair, our cells can repair, and when we're pretty healthy individuals. The farther we go, the bigger that omega-6 to 3 ratio goes, the sicker we are, the sicker our cells are. That 47 trillion cells has to have that fat in it to regenerate and repair. Did you know that the average European, listen to this, the average European, their omega-6 to omega-3 ratio is about an 18 to 1, about an 18 to 1. The average Japanese, and I know this because we have several clinics in Japan that we work with all the time, the average Japanese omega-6 to omega-3 is about a 10 to 1. Do you know what the average American's omega-6 to omega-3 is? It's about a 30 to 1. And you know what the average Midwesterners is? And you want to know why I know that? Because I've tested hundreds of thousands of Midwesterners. It's like a 40 to 1. And so this is when we have patients that come see us and they've got nerve demyelinization, brain cognitive dysfunction, chronic pain, chronic inflammation, and you're taking Advil, Tylenol, ibuprofen, all the things that are out there that's not getting to the root. Yes, it can knock down some of the symptoms, but it's not getting to the root. Getting that omega-3 level up, getting that ratio up is one of the most important things that you can do to get your body truly healthy. And omega-3s, you've heard of omega-3s before. Most people hear the term omega-3 and they think what? Fish oil, right? Well, omega-3s, listen to this. This is wild. Most people don't know this. Omega-3s are EPA and DHA. Just this EPA and DHA. Those are what omega-3s really are. Yes, some can come from fish oils. Yes, some come from olive oils. But omega-3s that make EPA and DHA, when the scientists of the world, I would have loved to have been in the room looking under the microscope whenever the scientists were loading up cells with EPA and DHA and seeing what the cells were doing when they were putting that EPA and DHA in there. The higher the EPA and DHA in there, the cells started to do two things. They started to repair and regenerate, which is wild. And so you know what the scientists are like, well, what are those things? What are we going to call those things, those chemical processes that are repairing and regenerating the tissues? So you know what they call them? Repairins and regenerons. They're little chemicals that repair. And you know what they do? They turn down the root cause of the inflammation. Because when omega-3s are up, it turns off the fire. Your body cannot heal if you're stuck in stage 4. It cannot and will not heal. And stage 4 is the critical point, because if stage 4 stays on, then the brain goes, OMG, we got a big problem. We better call them the big dogs. And the big dog is the brain. Because if you don't fix the inflammatory component, you can get pain, you can get swelling, especially in your lower legs. Anybody in here ever wake up in the morning and they've got massive puffiness under the eyes? They wake up and they've got really a lot of swelling in their legs, or their fingers are really swell. That is a sign of chronic inflammation. And then when inflammation is high, the immune system goes on auto drive. And then we get things like dysautonomia and autoimmune issues. Autoimmune issues, by the way, most people, if you have one, you typically have a total of 3, because autoimmune, the process of it happening is the same process. It's just how your body responds to it based off its deficiencies, toxicities, and its genetic predispositions. So when you're in stage 3, when your omega-3 ratio, when you start taking the right amount of EPA and DHA, and by the way, if any of you take fish oils, make sure your EPA and DHA, when you look at the number, that it's above 400, okay? Above 400. We typically use in our clinical settings some that are around 1,300 for EPA and 700 for DHA, but you want to have those levels high. I'm telling you, if you have not had that test done, the next time you go and get blood work done, ask your provider, I want to do an omega-6 to omega-3 ratio. And it should be higher than, it shouldn't be higher than a 4-to-1. It should not be higher than a 4-to-1. So please, please, please get that test done. That is one of the most powerful tests that you can have when it comes to root cause issues. Now, if you don't fix stage 4, the next domino falls. This is where most of my people come into my world. We do a lot of functional neurology with something called dysautonomia. That's when the brain and the body lose its communication. How many of you have ever had your body and your brain not talking to each other like it's supposed to, right? It's a lot more common than you think. Yeah. And the disconnection is typically on an autonomic nervous system level. I would highly encourage you, whatever providers or practitioners that you're working with, have them evaluate the functionality and the tone. It's called the tone of your vagus nerve. There's 2 big components of the nervous system, okay? How many of you guys have ever heard of the fight or flight response? That's called the sympathetic nervous system. When you're stuck in sympathetic, think about that like the gas pedal of your car. If your gas pedal's stuck on go, if you hit the brakes and the gas at the same time, nothing happens, right? Nothing happens. Well, in the body, we know that you cannot be in survival and healing at the same time. The survival part of the brain is the sympathetic nervous system. That's when the fight or flight response is high. Well, the opposite part of your body is the vagus nerve system. It's the parasympathetic. It's the rest, relax, grow, develop, and heal. The more we get this part to stimulate, the better our body and brain and our body and our organs connect and communicate like they're supposed to. This has been one of the most emerging aspects of all healthcare, really ever since 2016. I remember walking through an airport. I just was presenting at the American Academy of Anti-Aging down in Dallas, and I was coming back to St. Louis, and there was a magazine. I love how God always puts stuff right in the way, and you're like, oh, okay. There's a magazine. It was Scientific America, and it said Electrical Cures, March 2016. It was in Scientific America before it was really anywhere else. Why? Because most of our colleagues, because if there's no drug to treat something, we're not really interested in it, unfortunately. We now know there are ways you can stimulate the vagus nerve without doing brain stimulators and etc. I mean, my gosh, putting cold water on your face is one of like 50 different things you can do to help stimulate the vagus nerve. You don't need brain surgery all the time, okay? Let's try some simple stuff first. But it showed that if in this article, go look it up, March of 2016, Scientific America, electrical cures. It said science has now found the cure for Parkinson's, dementia, Alzheimer's, neurocognitive decline, paralysis, nerve degeneration, obesity, type 2 diabetes. I mean, the list goes on and on and on and on because they found if you can positively stimulate your vagus nerve, the body reconnects itself. You see, most people are eating food, they're doing their supplements, they're taking their medications, but that's like fuel getting into the tank of the car, but it's not getting into the engine because the vagus nerve isn't stimulating like it's supposed to. And if we can get the vagus nerve to stimulate, then our body goes from a state of stress response to relaxation. How many of you, maybe you don't want to raise your hands on this one, but how many of you guys would agree that most of us wake up, 90% of the time, we feel like we're running from the bear first thing in the morning? We're stressed out. We're go, go, go. When you're going that hard, when you can't sleep, when you're wired and tired, that's vagus nerve under stimulation. Like, did you know that, so insomnia is such a big issue with people. Most insomnia, most sleeping issues is vagus nerve not working, sympathetics too high, and your insulin's a little too high. You know what you can do? Try this. Try this tonight. Just try this tonight. And then tomorrow, tell me I'm either a genius or, not. Whenever you have a response. If it doesn't work, then don't tell me anything, okay? But try this tomorrow, or try this tonight. It takes about 3 hours for your insulin to balance out before it goes to baseline. So tonight, try to determine what time you're going to go to bed. Let's say if I want to go to bed at 10 o'clock, be done eating by 7. Give your body 3 hours to let your insulin get back to baseline. No midnight snacks, no handful of M&Ms, no cashews, no grapes, no nuts. Just your last meal's at 7 and be done. And see how much better you sleep because your insulin won't be all over the place. Your cortisol won't be all over the place. And then before you go to bed, right behind the base of your here, right here on each side is part of where your vagus nerve comes out. Massage that area of your vagus nerve on both sides. Literally just take your little hands, go in here and massage this area, kind of, a little vigorously for like 20 to 30 seconds. Pull on your ears. Yep, don't do it around your friends because you'll massage your ears a little bit. And then you can also massage where that vagus nerve comes down and then see how you sleep. Because a lot of people will notice just by a little vagus nerve stimulation that now they sleep better and they wake up rested. Because I'm telling you, so many of you are working so hard during the day to help yourself get healthy, but you're messing it up because you have the wrong nighttime routine. I'm going to show you a little bit about that routine. So the autonomic nervous system, and we're going to get more into this tomorrow, but most people are stuck in survival mode. And so when you're stuck in survival mode, your body won't heal like it's supposed to. And if you don't fix stage 5, this is when the brain gets put into the whole thing. Then what happens is now we get stage 6. Stage 6 is known as adaptive physiology. This is when the body starts robbing Peter to pay Paul. This is when we get long-term hormonal issues, especially insulin. And I'm telling you, insulin resistance is a major plague of real health of humans. Folks, please understand this. Insulin resistance is a blocker. If your body is insulin resistant, and the data shows that some 90% of American adults have some level of insulin resistance in their system because we eat too frequent, we're too stressed out, we don't sleep properly. When insulin goes high, every hormone goes low for the most part. Testosterone, estrogen, progesterone. When insulin's high, cortisol can go high. When insulin's high, your thyroid dysfunctions. When insulin is high, your body stores fat and doesn't burn it. And when insulin goes high, you don't take stored fat and burn it as energy. And your body's reliant primarily on carbohydrates as its fuel source. So you crave the bread, you crave the pasta, you crave the sweets. I mean, how many of you are like I used to be? I'd eat a good healthy meal, but I gotta have a little bit of ice cream before I go to bed so I can feel satisfied. She's like, yep, preach it. Okay, we gotta fix your insulin resistance. Because the problem is when you've got insulin resistance and adaptive physiology hits in, you get blood sugar instability, okay? You get adrenal fatigue and adrenal insufficiency. You start to get things like a congested, impaired liver, which can lead to fatty liver issues. And then we don't fix that. That's when our body says, I can't adapt anymore. And whammo, we've got stage 7, which is the disease stage. And unfortunately, that's where most of my colleagues, that's when you see most of our colleagues. Most of the physicians that you guys are working with work in a system that we used to work a lot in called managed care. You ever heard of that term before? Managed care means I'm interested in managing your diabetes, managing your blood pressure, managing your insulin, not helping you get to the root of it through lifestyle, diet, exercise, and a routine that actually can help you reverse these problems. Does that make sense? You guys track me on that? How many of you would rather have an opportunity to help get to the root and reverse versus getting managed? There's a big difference. And so I just want you to understand that there's so many things that you can do, so many things that can be done, so many things, thank you, mic check. Perfect. Now, I've been silenced before, but not like this. Just teasing. There are so many things that you can do to help get to the root, find out what stage, the 7 stages of this connection that you're in. And so, thank you. So when we get to stage 7, that's when you have the diagnosis. That's when you have the disease. That's the diabetes, the heart disease, the cancer, the liver disease, the whatever disease you've been diagnosed with. By the way, that's also not a death sentence. You are where you are, so now you know what stage you're in, and you just need to work it backwards. You need to fix the root cause of the problem that led to this issue. And again, I keep going back to the idea that the definition of insanity is doing the same thing again and again and expecting a different result. So, try again? Try again. There you go. Okay. So, I know this concept is a little different, and it's not, I promise tomorrow it's going to be a really exciting conversation, but I gotta put the baseline out here now, because this is some things that you've probably never heard before. How many of you have ever had any of your practitioners talk to you like this? How many of you have ever had your practitioners go, all right, let's check your cellular voltage. Let's measure toxemia in your body. Let's look at your terrain, your microbiome in your body. Let's measure your vagus nerve and check your brain functionality. Let's measure your physiology and see, do you have insulin resistance? Are you absorbing the nutrients or even medications that you're taking? I'm telling you, if you are not doing those things, you're not doing all the things that you could potentially be doing to help yourself. So I always tell my patients, and I came up with this term years ago, probably over a decade ago, and it really resonates with people. I always tell them, listen, you're not broken, you're blocked. And you're not diseased, you're disconnected. And so your job as the patient, the person dealing with the issues, and our job as the provider is to identify the blockages, physical, chemical, and neuroemotional, and not just the blockages in your heart, because that's the thing that most people think about first. And identify the disconnections and help the body with a lifestyle that can help them get reconnected. And we've identified, by looking at over 7 million patients all across the globe, we've identified these are the primary, what we call blockages in modern society. And insulin resistance is right at the top. Your body is trying to burn sugar as fuel instead of fat. Do you know when you burn fat as fuel, do you know what the byproduct of that is? It's called a ketone. Have you ever heard of that? You've heard of ketones and ketosis? Do you know what ketones and ketosis really is? It's a state of physiology. It's a healing state of physiology. Do you know the ketogenic diet? Do you know what that was originally made for? I'm sure some of the physicians in here know that. It was made to control seizures. Because when you get someone in a state of ketosis and you feed the body, especially neurological cells, neurological mitochondria, you feed that mitochondria those ketones, it just eats that up, loves it, gets stronger, multiplies, and makes the organs that are fed by those mitochondria work better. That's why it's important to kind of look at your lifestyle. Because if your daily lifestyle is feeding insulin resistance with you, which most people are, it's causing a blockages in the rest of your hormone production. And so many of you are coming to practitioners like me saying, hey, doc, give me some testosterone because I saw on TV that it, fixes all kinds of problems. Well, it's all about the insulin first, and insulin resistance is a big deal. So having that checked is huge. Chronic inflammation is another big block. That's a massive block in the body. The body never gets a window to really recover, and the fire is constantly on go. And when that happens, it depletes everything downstream. That inflammatory marker, which is where most people are really stuck and really blocked, if you can get that omega-6 to omega-3 ratio balance, oh man, that could be a game changer. And some of you, it might take 90 days to do it. It took some of you 3 to 6 decades to get there. You can do this for 90 days. And I'll show you some of the things that you can do with it. Gut dysfunction is a big one. You guys, I don't know if you've heard of the brain. How many of you have heard of the brain-gut connection? Now, we now know that there's a brain-gut connection, there's a liver-gut connection, there's a pancreas-gut connection. The whole body is always in a constant state of communication unless the cell voltage goes down, like I told you. And then what happens is not only do we get upset stomach, that Pepto-Bismol commercial, diarrhea, constipation, upset stomach, blah, blah, blah. You get all kinds of other problems. The neurotransmitters, do you know what a neurotransmitter? Dopamine, serotonin, all these neurotransmitters, they're primarily made in the GI tract from amino acids, from the gut microbiome. And when those aren't working like it's supposed to because we have gut dysfunction, that's a big issue. Now, I just finished, we did a 7.5-year study of the blue zones of the world. How many of you have ever heard of the blue zones of the world? The blue zones are the areas of the world where humans typically live longer. They typically live to be over 100. They're typically healthier. They have a lot of, it's very fascinating. If you want to be successful at something, you should probably look at people who are successful at it. If you want to live a long, happy, healthy, balanced life, you should look at people that have done that. You can emulate some of those things. Now, there are a lot of commonalities that we have found in individuals that live in the blue zones. Like my poor kids, God bless them. I've taken them all over the world. We took them to Costa Rica 2 years ago. They were so excited. They wanted to see the howler monkeys and all the great, they wanted to swim in the ocean. And dad just wanted to study the poop of all the people that live there because I wanted to see their microbiome. I did the same thing in Okinawa. We did the same thing in Sardinia. We did the same thing in Lima, Loma, California, the areas of the world where people live longer. And guess what we found? We found that there is a blueprint, what we consider an ideal blueprint for the human gut microbiome. And when you have that microbiome, you live longer, you look younger because your mitochondria turns over faster. There is no major breakthrough in science. That there are things we can do now. And one of the big reasons why science is telling us that, hey, you make it to 2030, you're going to be able to live 120, 130 years of life. It's not just the new breakthroughs in science. It's about the fact that we now can communicate about things that we've been seeing and doing in our clinical practices. So we took all the data from the longevity studies. How many of you have heard of those? Those are the studies that have been going on now for several decades of seeing how people live long. And then we looked at the blue zones, and we were able to come up with the gut microbiome dysfunction protocol that helps people get their gut microbes right. Because when the microbes aren't good, your brain and your vagus nerve cannot work like it's supposed to. How many of you have maybe ever, I don't want you to tell on yourself, but let's say maybe we went out one night and had a little too much fun, had too much good food, maybe we had some friends and a little bit too much vino, and the next day you didn't feel so good, right? You physically didn't feel good. But how did you emotionally feel? You didn't feel good then either, did you? Most depression we know is a depression of the vagus nerve, primarily from the inflammatory issues of an abnormal gut microbiome. So that's a huge block in the system. Toxic load is a big one. I don't know if you know this, but we now today have a higher toxic burden than any other time in recorded human history. There's over 80,000 toxins that are allowed in our water and food supply that unfortunately are illegal in certain countries. I mean, one of the reasons why I love going to Europe and Japan, I'm leaving in a week to go over and do a 14 city tour over there in Europe, is because their food's different. They don't have as many chemical toxins. I don't eat a lot of bread and pasta here in the good old US of A, but when I go to France and Italy, I'm eating that stuff up because you don't feel so sick and bloated and tired and gross because it's different. We have a massive toxic load in our world. And listen, every one of us across the globe has major issues. You know, I'm one of the physicians here in St. Louis that's working on the Coldwater Creek project. I don't know if you're familiar with this. Okay, we also have, we have a lot of patients that live out in St. Charles, which is not too far from where we are right now, out in Weldon Springs, where the largest uranium dump is in the Midwest. I mean, it's crazy. There's so many toxins in the food we eat, the water we drink, the air we breathe, the clothes we wear. These things are causing stresses on our body. And if we can remove some of these blockages and some of these disconnections, that's when the therapies that you're doing are going to work better. I mean, that makes sense, doesn't it? You know, if I put brand new tires on my car that the engine doesn't work and I put diesel fuel in the gas tank where we're supposed to have gas, that's not going to run very well, is it? It's amazing how well our bodies do. And my colleagues, God bless all of us, we all have specialties that we specialize in. And I always, when people ask me what's my specialty, I tell them two things. Number one, I'm a generalist. I specialize in the generalization of how the human body works. And I lovingly say this, and let's see if any of you can get this, and I mean this lovingly. I'm also a fantastic craniorectologist. I like to pull craniums out of rectums because there's a lot of heads up our heinies in healthcare, and sometimes we just need to have a common sense conversation because you can take all the metformin and all the Ozempic and all the pills and all the stuff on the planet, but if you don't tell your patient stop eating that crap, they're going to keep getting sick. And you as an individual are trying to remyelinate nerves and trying to get your muscles to work like it's supposed to, but you don't have the right macros in your body. You're not taking the right nutrients. You don't have the right minerals. You're not doing the right breathing activities. How is that possible? Explain it to me. You're putting brand new Ferrari tires on a Ferrari with a broken engine. Let's have some common sense and yes, get after it and understand that it is your responsibility as a human being to understand these things. And you cannot just rely on your practitioners to tell you what you should do. If you want to be healthy, you've got to be the one in charge. You've got to know what questions to ask. That's why coming to events like this, engaging with individuals like me to help you think outside the box is something that you should be doing. Because again, the definition of insanity is doing the same thing again and again and expecting a different result. Sympathetic overload is a major problem. I'll tell you, we measure brains. We do SPECT scans in our practices and vagus nerve measurements on every single patient. They're doing some right now for patients that I'm going back to South County when I get done in 20 minutes to go take care of. And we find most of them have very high sympathetic overload. If your brain is stuck in fight or flight response, not only can you not communicate well, how many of you have ever tried to communicate when you're overstressed? Or how many of you have tried to communicate with someone when they're having a bit of a tantrum? Okay? It's hard, isn't it? Adults and kids have these issues. But you know what else is hard? Trying to convince your body to heal itself when you're super stressed out. It's impossible. You have to understand the vagus nerve and how to deal with it. And there's so many things you can do. And then sleep deprivation is a major block. Did you know that if you get less than 5 hours of sleep per night, and most of you are going to go, well, that's me all the time for the last, that takes years off your life and life off your years. Did you know that? If you're not getting at least 6 to 7 hours of sleep, most of you are waking up insulin resistant from that. Most of you are waking up in sympathetic overload. Your cortisol is too high. Your body is not in a state of healing, and you're trying to take all these supplements, medications, and other things to help you, but your nighttime routine is messing you up big time. Tomorrow, I'm going to show you the ultimate nighttime routine. I'm going to talk a little bit about it now. And then one of the last things, this is really wild, but we found this, and this is why it's so good for you to be here. People who are, folks in the longevity studies that live the longest, and people in the blue zones, you know what they were a part of? A community. We are community creatures. We gain health by being together, gaining knowledge, having fun, having fellowship. That's why coming to the dinners and the events, things that you've got planned are so important for you to be doing. These are the major blockages that we find with our patients, and removing these blockages, changing the lifestyle, gives a better opportunity for real healing. So what we do is we identify the phase of degeneration or disconnecting you're in. You identify that dominant stage, which I'm going to, in a moment, give you an opportunity to click on a QR code. You can get this presentation, and my team will send you a quiz that we developed. It's a quiz that you can take that will help you find out what stage of disconnection that you're in. And then you're going to get a fun little protocol to follow. Completely free, no strings attached, just some things for you to start learning, because doctor is Latin for teacher. Our job is to teach, reach, inspire, and hopefully motivate people to do things, not just prescribe medications, supplements, or etc. We should be educating our community. And that's why I'm here right now. So you find the blockage, you remove the interference, you start some therapies, you test, retest, adapt, and you build resilience to the body. For more complex cases, you should be asking questions like this. You shouldn't be asking questions like what should I be taking, doctor, to get rid of my inflammation and pain? You should be asking, why do I have inflammation? How do I control my inflammation? Is my digestion working? Is my blood sugar balanced? Is my stress being managed? Am I handling it myself? Is my mitochondrial function being supported? Listen, there's certain foods that feed mitochondria. Like I just found out my wife and kids are on vacation this week. See, dad's at home working still, right? But my wife and kids are on vacay, and I'm at home feeding the animals. And we have a little a bearded dragon. And I just found out what bearded dragons eat and how they are supported. Well, now I know how to make that bearded dragon grow big and grow strong. If you want to improve your mitochondria, you just have to know how to grow it. You got to have the right B vitamins, the methylated B vitamins. You got to have the right minerals. You got to have the right amount of protein. You got to have the right ratio of omega-6 to omega-3s. This is not anything that's difficult. This is common sense, but unfortunately, common sense is not that common in our world today. So we talk about something all across the globe when I get a chance to present. We call it the Ultimate Feel Great System. And it's a simple daily action step process that we walk people through, and we educate people on this so that you can live a happy, healthy, balanced life. And we talk about certain things that need to be balanced. We talk about discipline and nutrition and recovery and movement and mindset, things like prayer and gratitude and purpose and honoring commitments and honoring your community. There's so many pieces to health other than just taking drugs and taking supplements, in addition to those things that can benefit you. But it's all about your daily rhythm. I always say that your rhythm is your medicine. What you consistently do is what you're going to get. So let's talk about just the daily life of most healthy individuals. And whenever you look at this and you go, I'm not doing that, that might be why you're not as healthy as you should be. So number one, most people, I would agree, most people wake up between 6 a.m. and 8 a.m. Write this down. Please remember this. You want to start your day off right? Hydrate before you caffeinate. Hydrate before you caffeinate. Most of you are waking up insulin resistant. You're waking up with cortisol levels that are overstressed. If the first thing you do is throw a Red Bull or espresso or a coffee at it, this is going to mess you up big time. So one of the things we teach people first thing in the morning is we say, get up, get about 4 to 6 ounces of water. Get yourself a lemon or lime, put a squeeze of that in there, get about a half a teaspoon of organic sea salt. Most salt has about 75 to 80 different minerals in it. Now, doctors, listen, those of you that aren't aware of this, salt doesn't kill people. Sodium is a problem, okay? So I'm not talking about table salt, NaCl that's on the table. We're talking about minerals. Your body needs 92 essential nutrients every day. You must have 92 essential nutrients every day. 84 of them are minerals. If you don't get these minerals in your system, your body doesn't start its day off right. You can drink a gallon of water every day and still be dehydrated. So hydrate before you caffeinate. Our bodies are made to get up with the sun and go down with the sun. Did you know that the healthiest people on the planet, when the sun gets up, they get up, and you know where they go? Outside. Our body needs minerals and it needs light. We have photon receptors in every cell of our body. Those receptors charge our system. I've got patients that come in my practices and spend lots of money to get in our red light therapy beds and all these infrareds that we have that charges mitochondria. And you know what I say to them? Why don't you just get up in the morning, drink some good water, and go outside? Save yourself a little time. Well, I don't do that. Start doing that. Get outside and get some fresh air and some sunshine in the morning. You know what I like to do every morning? I like to get up every morning and go, okay, thanks for giving me another day to do what I'm supposed to do. I kick my shoes off and go outside and stand a little barefooted in the grass, drinking my little morning drink and just being grateful for the opportunity to serve humanity with the abilities that I've got. That's a powerful thing to do. I learned that years ago, and guess what went away? Anxiety, depression, insomnia. And guess what came? Energy, purpose, and health. It's a simple little tool. Here's another thing. Gosh, I'll tell you what. Starting to learn about fasting and intermittent fasting is a powerful thing. I'm telling you right now, if you're over the age of 30 and you want to control your metabolic dysfunctions, and when I talk metabolic, I'm talking about brain, nerves, everything is metabolic dysfunction. Everything is. If you don't have proper metabolic flexibility, if you can't go 2 or 3 hours without having something to eat, you got a problem. If you get exhausted after you eat a meal, you got a problem. We tell most of our people, try to have your first meal of the day around 10 a.m. or 11 a.m. in the morning. And protein heavy, protein first, about 30 to 50 grams of protein with each meal. The fasting window, we also use certain nutrients in the morning. I'll send you this protocol so you can have this, but we like fasting. We find some major changes in the body with fasting because of how well it controls insulin and the hormone cascade. So let's talk a little bit about our feeding window. When I talk about this, it sounds like we're farm animals. You have a feeding time and you have a fasting time, okay? Well, most people should feed between 10 a.m. and 6 p.m. or 11 and 7. You get about a 7 to 8 hour window in there. And I'm telling you, most people, if they'll follow this, will completely change their life. And then when they eat, they should eat in a sequence. Write this down. Remember, they should take a picture of this. Fiber first. When you have fiber first in your meals, it lowers your glucose absorption and insulin. Some studies show by 37%. That's huge. That's huge. Huge. Second, proteins and fats. Proteins and fats. Again, between 30 grams to 50 grams per meal, per feeding. Most people are going to feed twice a day. Okay, some will have another meal, and that's fine, but this is going to help with satiety. This is going to help with muscle. You cannot heal the body if you don't have enough protein. And by the way, as we get over the age of 30, we lose our ability to make enzymes called protease, digestive enzymes. We can't break down the protein we eat to make our skin look good, our hairs look good, to make our own peptides, and to make our own antibodies. Our immune system runs off of antibodies. You don't have enough protein, you can't do it, nor can you make enough hormones. Number 3, healthy fats. Fats are good. Man, extra virgin olive oil, coconut oil, MCT oil, eggs. Isn't it funny how every other year it's like, eggs are good, eggs are bad? No, they've always been good for you, okay? Eggs are like little multivitamins, okay? Just another thing that unfortunately a lot of my colleagues got wrong, and they never really looked at the true data. And now we're showing having eggs, great form of choline. Choline and those good fats in there are so important for brain regeneration, adrenal health, liver health, etc. Good fats, avocados, all this stuff is amazing. And then get your vegetables, number 4, okay? Your veggies, okay? All things in moderation, even your vegetables, moderate those. And then last but not least is when you can have your carbs. When I talk about carbs, that's bread, pastas, grains, and fruits. All those are good, but all in moderation. You want another body biohack? One of the biggest things that we've learned in longevity is if you can start going for 10 minutes of movement, whether it's a walk, standing on a vibration plate, doing something for about 10 minutes after you consume food can lower your insulin and glucose again by about 20 to 30 points. And it can also raise what's called your GLP-1 production. Well, everybody's taking all these shots and doing all these things to do that. You can do it through lifestyle. And then whether you like it or not, we're supposed to move it. If we don't move it, we lose it. You got to get your body moving. We got to activate and hydrate and sweat. You should be doing 2 to 3 weeks of resistance, 2 to 3 times a week of resistance training. Now, I'm not saying you have to go to the gym. Literally putting on a weighted vest or putting some ankle weights on, going for a walk is resistance training. Doing some push-ups, some pull-ups, doing some air squats. Whatever you can do it. It's one of the most important things for longevity because muscle is metabolism. The more muscle mass we have, the longer we live. I don't know if you know that, but one of the biggest indicators, one of the most important indicators of longevity, a test that you can do is grip strength. Because the stronger your grip is, the stronger your body is. It's one of the VO2 max and grip strength are some of the most important biomarkers for the body. Sweating daily. I know sweating is a 4-letter word for a lot of people. I get it. It's 5 letters, but you know what I mean, okay? We're supposed to sweat. Tomorrow I'm going to talk about that a lot. We can get more microplastics out of our body through sweating than any other way that's out there. It helps the lymphatics, helps with all this other stuff. Hydration. This is so important. You could drink a gallon of water every day and still be dehydrated. You need to have minerals in your water. So put a little sea salt in there. Get some minerals to put in there. Put a squeeze of lemon, squeeze of lime. Most people need about 2 liters of fluids, healthy fluids. That's not just water. That could be teas, herbal teas, things like that. And every cup of coffee or caffeine you have, just have a glass of water to cancel it out. Most caffeine, by the way, caffeine is a very good, healthy stimulant in moderation, but it's a diuretic. And you want to replenish those with minerals. And then afternoon movement. If you can go for a 10-minute walk or a 10-minute exercise after your 2 to 3 meals a day, do you know how much years to your life and life to years that can add? It's immeasurable. Simple little changes. Let's talk about the recovery. This is a big one. This is where most people mess themselves up. From 7 o'clock, remember I said no food after 7. Close that kitchen. Shut it down, baby. Okay? Nothing over 20 calories after 7 p.m. because it'll break your fast. Okay? Now, around 7 to 8 o'clock, you start dimming the lights. You know why? You know, our brain runs off of circadian rhythms. If your light's real bright or if you're on your phone late in the evening, those blue lights stimulate your brain to tell your cortisol to wake up. Guess what? It's daytime. We got work to do. And now you're trying to go to bed? So you can wear blue blockers or turn the volume down or the voltage down. That can help you. Hot shower, hot bath, because as you cool down from that hot bath, that mimics the body's ability to start going to sleep. You could try to get in bed by 10 o'clock. Magnesium glycinate, oh my gosh, the studies on 300 to 600 milligrams of magnesium glycinate at bedtime to help lower insulin and help lower cortisol are amazing. It'll help you stay asleep and fall asleep. Okay, cortisol and insulin are the biggest reasons you keep waking up at night. Most people wake up and then go to the bathroom. Okay, they don't wake up to have to go to the bathroom. If you wake up and you don't have to go potty, lay there for a minute and try to go back to sleep because your body gets into patterns and habits. And then between 10 p.m. and 3 a.m., I mean, there's so many studies and data that shows we refer to that as the Genesis window. Why? Because that's when the body repairs and regenerates for the next day. If you're not in bed and resting between that 10 p.m. and 3 a.m., you're reducing your opportunities to heal. And I know a lot of you are going, well, that's my TV time. No, man. That's your time for your body to regenerate and repair. So sleeping in a cool, dark, quiet room, no TVs, no lights, it has to be dark. The darker, the better. You can have some ambient noise playing or some frequencies like 528 frequencies been proven to help with relaxation and deep sleep. This helps your body heal, growth hormone repair, recovery, cellular regeneration, and brain detoxification. When you put it all together, you want to test, not guess. You want to find which stage of the 7 stage of disconnection that your body's in, and then you want to develop a plan to reverse it. And it can be done. And that plan needs to be done by you. Your providers are going to help you hold your hand along the way, but we're there just to show you the path. You're the one that has to go down it. So if you guys want to learn more, you want this presentation, go here. If you guys don't know how to do this, take a picture of this, or you can go to the QR and click on the QR. The presentation I gave is there. It'll give you a link that you can get to go take the quiz. It'll tell you where on the 7th stage of disconnection you are. And that way we can connect, reconnect, and stay in touch with each other. How many of you guys learned something here today? Yeah. How many of you knew people that probably should be here to check this out? Tomorrow's going to be even better. Okay. So I just want to say thank you for your time today. I appreciate what you're doing here. I appreciate the community that you've got. I appreciate the leadership that you've got. I think it's important to take everybody's information and take what resonates the most with you. Get that discernment, start putting it into work, and understand that self-care is the real healthcare. And the more you take care of yourself, the less you have to be relying on others to take care of you as well. So thank you guys so much for your time today. Have an amazing rest of your conference, and I look forward to seeing you tomorrow to talk about detoxification. We'll have some fun then. Thanks, everybody. Appreciate you.
I'm going to fix that tomorrow. God darn it. Come on, baby. Hold up there. Damn it. All right. We'll do the under tilt. Okay. Sorry, guys. Had to wrestle the microphone a little bit to make it work the way I wanted it to go. All right, for our last presenter of this morning's session, as we near the end of our first day together, it is important to recognize that neurological disease affects more than nerves and muscles. It affects relationships. It affects families. It affects caregivers. It affects emotional well-being. The journey toward therapies is important, but so is the journey of living with uncertainty, adapting to change, and finding ways to care for ourselves and one another. Our next speaker reminds us that compassion is not a luxury, it is a necessity. And that includes compassion for ourselves. Please join me in welcoming Christi Lero, presenting Practical Self-Compassion, Sustainable Caregiving, and Emotional Resilience. Ladies and gentlemen, Christi. And you got them? Good.
I got the clicker.
Good. And you got a little screen.
Where can I
here.
put this without?
You set that right there.
Nice.
And you're good to go.
This is so fancy. Thank you.
That thing has its own a mind of its own. So
Okay.
Be careful. It's heavy and weighted.
Can you hear me?
Yeah, you just
Yes?
pump it up. We'll get you.
Better or worse or the same?
It's okay.
Better? Okay, good. Stay. She's being obstinate.
There you go.
Is that okay?
Okay, everybody, don't breathe.
Well, that's barely.
There you
I
go.
heard a barely. That's not good. Does she have a name? Thank you. My kids are going to be so excited because this looks like a rock star thing. I have a new 5-year-old, and she would listen to music and rock out all day if she could. Better? Oh, I think it has to be close. I think she's got to be really close. There we go. Good? All right. Teamwork. Okay. Thank you all so much for having me here today. Let me know if it makes me nervous. It's wobbly. Okay, we're going to try not touching anything. Okay, thank you all so much for having me. It's an absolute honor to be here, and I really appreciate your time and energy and effort in listening to me while I stand between you and your dinner. So I promise not to take up too much of your time. But again, thanks so much for letting me come talk today. I don't have any conflicts of interest, but I do want to acknowledge that my time and my work is partially funded by the NIH. And this work, all the work that has been discussed and demonstrated today is really only possible with willingness and engagement from people like you and from researchers and healthcare providers. It takes a village, right? Okay. So I would like to start today just kind of giving us a reference point of, I talk mostly about caregiving, but really the concepts, the ideas that we're going to talk about with self-compassion today are really applicable to everyone. There's no reason why we need to just focus on caregiving, although caregiving is also not necessarily included in a lot of the research that we have and a lot of the policies that we make. And so in order to make caregivers more visible, we need to talk about it. So I think this is updated. We are now at 63 million family caregivers, do mostly unpaid work that is equivalent to about almost 24 million full-time paid caregivers. About 44% of the caregiving that is done is in high-intensity caregiving situations, just like in HSP and PLS. And then the unpaid estimated value is near 2 trillion. And I don't know about you, but I have a really hard time conceptualizing, like really wrapping my head around how much that is. And so if we think about it in seconds, 1 million seconds is about 11 and a half days. 1 billion seconds is about over 30 years. So 1 billion seconds ago was in the early 1990s. And then 1 trillion seconds ago was the Ice Age. So we're talking about almost 2 trillion ice ages ago of value. which is incredible. And that reinforces this idea that caregivers are the backbone of our society, not just in the face of illness, but really everywhere. We use caregivers in mentorship and guidance and physical health and support and in emotional well-being as well. So we might be looking at a rare illness today, but really caregiving in and of itself is not rare at all. So I'd like to try an exercise together if you all are willing. We're not going to meditate together and we're not going to hold hands and sing a song, I promise. But I do want to kind of test something out. So if you wouldn't mind thinking about a friend or a family member, somebody that you know, not an imaginary person, but really somebody in your life that you know. And I'm going to read off the prompts on the screen, the statements on the screen, and I want you to think about how often these statements might be true and maybe even the intensity of the trueness. Is it really, really true? Not really that true? Somewhere kind of in the middle. So bring that person to mind. Think about the last time you saw them, the last time you spoke with them, maybe how you feel when you're with them. Really just kind of bring them to mind. When they're experiencing suffering, I can be a bit cold-hearted toward them. I'm intolerant and impatient about those aspects of their personality that I don't like. When they're feeling down, I try to approach their feelings with curiosity and openness. And I try to see their failings as part of the human condition. We're all people. So kind of think about how often those things are true or not true and how intense those things could be. And now I want you to shift a little bit and think about yourself. Bring yourself to mind. Maybe look down at your knees, at your body, at your hands, kind of touch your fingers, get a sense of yourself. Think about who you are as a person, your own identity. And we're going to do the same thing. We're going to consider how often the following statements are true and maybe even how intense those feelings can be sometimes, or not intense. When I'm experiencing suffering, I can be a bit cold-hearted toward myself. I'm intolerant and impatient about the aspects of my personality that I don't like. When I'm feeling down, I try to approach my feelings with curiosity and openness. And lastly, I try to see my failings and my mistakes as a part of just being a human. So I'm not going to ask you to share, but I do want you to consider and just think about what you noticed. What was your general approach toward the person that you know? And then what was the general approach toward yourself? Was it the same or different? And why might that be? So I've used the word suffering a couple of times, and I do want to just clarify. So when I talk about suffering, I mean kind of anything that's uncomfortable, anything that we don't like, anything that we don't want. That can be things like mood. That can be things like physical discomfort, financial distress, all kinds of things. These are all forms of suffering. And I'm a licensed clinical social worker, which means I can do mental health therapy with folks, and I did for a while. I was in private practice, and I helped support patients and caregivers and healthcare providers dealing with chronic and terminal illness. And the thing that I heard a lot from the caregivers specifically were these things. I could have done better. Why am I focusing on myself when they're going through so much? I wish I could have trusted myself. How can I forgive myself? And these are people that gave their all and that still continue to. So what's the gap here? Why are we being so hard on ourselves? So I went to the literature, as one does, and the literature showed me that there are two different survival systems that humans have developed over time. The first one we know pretty well. We hear about it often. It's the defense system: fight, flight, or freeze. When we feel threatened, we use these three mechanisms to stay safe. The second system we don't hear about as often, it's the care system. And when someone around us is suffering, we use 3 other mechanisms: compassion, support, and kindness. If somebody that we know falls ill or has something hard happen to them, we maybe make them dinner, we say kind things, we start a meal train, we do all kinds of things, right? But when we ourselves experience suffering, we engage the defense system instead of the care system. And that can lead to isolation, depression, anxiety, burnout, all kinds of consequences. So the question then is, if we have these 2 systems, how can we move from defense to care? How can we activate that care system for ourselves? The literature also, and I'm like, well, why do we want to do this? What is why self-compassion? Does it really have that big of an impact? Absolutely. So in general, I know there's a lot of, in general, the patterns that you'll notice are that when self-compassion is available to people and we're able to engage in it, it diminishes things that we consider to be negative: depression, anxiety, burnout, fatigue, secondary traumatic stress, which happens for healthcare providers quite often, and then also enhances the things that we consider good, that we like. Things like being able to cope, our social capital, which is like strengthening our social network. Self-soothing, we're able to think more clearly and see, have clearer problem-solving skills. Quality of life, and then our capacity for self-care actually also is enhanced when we have access to and when we can engage in self-compassion. So it's important to talk about what self-compassion is not. I think a lot of self-compassion's kind of been popular and kind of a bit of a buzzword the last couple of years maybe. And so I think it's important, some really nice things are said about it, but it can seem kind of fluffy, right? It can seem kind of woo-woo, maybe. But I think it's also important to talk about what self-compassion is not. And self-compassion is not just seeing the silver lining. It's not just about toxic positivity or only thinking about the positive aspects of life. It's not avoiding things. It's not a weakness. And it's not the same thing as self-care, although things like bubble baths, spa days, pedicures, going to get your hair done, those things can be acts of self-compassion. But those alone are not, and the underlying issue, the underlying reason for doing those things is not necessarily self-compassion. And most importantly, self-compassion is not selfish. Are you all familiar with the put your own oxygen mask on first when you're flying? Yeah. So the intention behind that is that you can put your mask on so that you can then help and support others. There's nothing selfish about that. That's the bigger picture. That's seeing the rest of the forest, right? So that's not selfish. That's really thinking about the bigger picture and long game. It's thinking about the long game. Okay, what is self-compassion? Self-compassion has 3 components that we'll talk about individually. So the first one is mindfulness. Again, this has kind of been a buzzword lately, but it's also really buzzworthy. It's pretty good. So mindfulness is acknowledging these aspects of the present moment, what's happening right now in this minute, with clarity and non-judgment, not labeling it, just observing and noticing. And it feels like noticing. It feels calm, right? Contrast that with something called over-identification. So how do you know if you're being mindful versus over-identifying? So over-identification is kind of becoming lost or becoming ruminating. You're just kind of hooked or stuck on this one idea or thought. Or you're getting attached to this idea of what's a good caregiver, or what's a good nurse, or what's a good spouse. And so that's really over-identifying. You're really kind of hooked onto this idea or this identity rather than this observing. So over-identification can feel confusing. You can feel stuck. Or sometimes you're so over-identified or so strongly attached to that thing, you need a break from it. And so you want to distract yourself or avoid, understandably. The next component is kindness. So kindness is being gentle with ourselves. Even when we make a mistake, when we have a failure, or when we feel like for whatever reason we're lacking in some way. And kindness generally feels like support. It feels like warmth. Versus, in contrast, judgment. So being cold or harsh, sort of labeling or lashing out, devaluing the importance. $2 trillion, y'all. So devaluing the importance of that work and the importance of being the backbone of society. And then it can feel like criticism. It feels like judgment. And last is common humanity. So I can't remember his name, but the speaker before us talked about community and common humanity, right? Like he talked about this just in a different way, which was great. So it's this recognition that everyone is human. We all go through hard times. We all make mistakes. We all have failures. It doesn't mean that those failures or those mistakes are going to happen in the same way. They're all different because we're all unique humans. But the experience of making a mistake is universal. The experience of going through a hard time, having losses, is universal. So it feels more like connection, and it kind of takes the pressure off of, I have to do everything myself. I'm all alone. I'm the only one that knows. So it kind of feels a little bit lighter, more neutral, versus this idea of isolation. So isolation is that sense that you're the only person that knows what this is like. You're all alone. And where it can certainly feel that way sometimes, remembering that everyone's got setbacks, everyone has hard experiences, we're all human, can again really tap into that connection piece. So isolation can feel like anger, it can feel like loneliness, and isolation. All right, so the big question is, how do we do this? We know what self-compassion is, so how do we shift from that defense system to the care system? And really it lies in a seemingly simple question. It's not simple, but it's not easy, but it's a simple question. What do I need right now? We don't often ask ourselves that. So if we break it apart into its pieces, if we look at mindfulness and we're asking ourselves, what do I need right now? Okay, I'm going to take a deep breath. how many of you have heard of grounding exercises? Oh, okay. One. I got one. Okay. Well, if we have time later, remind me and we will practice some grounding exercises. But grounding exercises are things like really just touch your fingers and notice what that feels like. Put your feet on the ground if you can, and really, or like feel what your seat looks like, or feel the breeze, using all of your senses to really engage where you are and how you are in the present moment. So taking deep breaths, and then again, noticing, non-judgmental. We don't need to label it as good or bad. It just is, right? So for example, I feel a little breeze somewhere. I don't know where it's coming from. There's bright lights. There's lovely people. There's no funny smells, which is great. And I have this taste of stale gum in my mouth because I had the last piece of some berry blend stale gum that I found in my car and then had to get rid of it. So those are reminding me what's happening in the present moment. And then thinking about what is the next step? What do I need to do to be kind to myself in this moment? I notice the breeze. It's very comfortable. It's not too chilly. So I think I'm going to keep my jacket on. I'm pretty comfortable. But if I was too hot, I always have that option. The most supportive or kind thing for me in this moment might be to take this jacket off. And then remembering common humanity. Who is in your community? Who's in your network that you can rely on and ask for help? I don't know that I necessarily need to ask anyone for help, but I'm sure you are all incredibly kind. And if I did, I asked for help earlier, actually. I didn't know if you all could hear me. And I said, can you hear me? There we go. Nice. All right. So sometimes it's hard to really focus in on yourself. So another option might be imagining what your friend might tell you, what a loving family member might tell you. Sometimes for me, I think about what my kids might tell me, because my kids are just the kindest humans I think I've ever met. So that's always a nice, just imagine what a kid would tell you. So let's practice. The examples that I've got are really thinking about kind of our internal dialogue. That's where a lot of this starts, is our internal dialogue. How do we talk to ourselves? How do we relate to ourselves? So let's practice with this over-identification piece. So if you'll notice, each one of these examples is I am. And then the underlying kind of, this last one is I should have caught that. And that's a bit of a tricky one, because the underlying message there is, if I were a good, insert label here, insert role here, if I were a good spouse, if I were a good partner, if I were a good caregiver, I would have caught that. And so that's over-identifying. That's really getting hooked into what is a good caregiver. So let's start with, I'm the worst. How can we kind of transition that into something mindful? I'm feeling pretty down right now, and this feeling is not who I am. So a little more neutral, feels a little warmer. I'm a bad caregiver. Caregiving is hard sometimes, and I'm feeling it intensely in this hard moment. So again, being mindful, putting yourself in the present moment. It's this moment that's really hard, and I'm feeling it. I should have caught that. Health and illness can be complicated, and hard feelings are a natural reaction to this situation. So there's a little bit more breath there. There's a little bit more space between I should have caught that and really blaming yourself, as opposed to recognizing that this is hard, this is complicated, and I have a right to feel this way. So shifting from judgment to kindness, this could be identification as well. It's really judging how you're doing in the role of a partner. So I'm failing as a role of a partner. Let's be kind to ourselves. I'm doing my best in a really hard situation. I do not have things together, and I am so disorganized. That was me. That's mine. That's from me. And instead, I'm managing a lot right now, and it's easy to get things mixed up. Mistakes happen. Mistakes happen is from my kid. I'm not good enough at this job. I work hard and I care a lot, and I'm doing the best that I can, and that is enough. How many of you can tell when your care providers are doing the best that they can? And they're working hard, and that's all we can ask of them, right? So we're not going to hold them accountable for things as long as we know that they're doing the best that they can. And we should do that, offer that same to ourselves. And lastly, isolation. No one knows what this is like. Dealing with this problem is a burden. I don't want to bother anyone with my issues. I don't know what to do or how to change this. So let's shift over to connecting and embracing this common humanity piece. We are all humans. Everyone goes through hard times, and I'm doing my best. We're all human, and we struggle in different ways. I know people who care about me and who want to help. Everyone feels helpless sometimes, and it's okay to feel this way and not have the perfect answer. So up until this point, we've been talking about the first side of self-compassion, which is tender self-compassion. It's this kind, caring mama bear taking care of her baby bear, right? Snuggling and being sweet and being gentle. Then there's another side of self-compassion. Same principles apply, but this one is called fierce self-compassion. And this one doesn't get as much attention either, as I think it's more, I think they're equally challenging, but I think this one we just don't think about it as often as a way of being compassionate towards ourselves. And so this mama bear obviously is daring someone or something to try to mess with her baby. And I hope they don't, because I think that she would win. So it's really this like change in posture, right? So the tender self-compassion is really loving and supportive and kind. And then this fierce self-compassion is a change of posture. It's really, this is my stand. I have boundaries and I have rules, and you're not messing with my kid or my loved one or me. So fierce self-compassion, again, requires this change in posture. And it's really the attitude that we take, the approach that we take when we're creating boundaries and changing that posture. And it really relies on this, we care about ourselves, right? It's because we care about ourselves, not because we think that we're somehow lacking or we somehow are insufficient or not able to do things. It's really about taking care of ourselves. And this can look like a lot of different things. It can be protecting our time and energy and resources. It can be learning new skills or new tools, finding new tools to be able to show up in a way that makes us feel like ourselves. And then creating boundaries. Most of the time when we think about creating boundaries, we think about saying no to things. But boundaries can also be saying yes. I'm going to create a boundary and I'm going to say yes to taking care of myself. I'm going to say yes to 5 minutes with a quiet cup of coffee in the morning, or I'm going to take, I'm going to get outside and breathe fresh air for a bit. So all of those things are saying yes to yourself. And that's also a way of creating boundaries. Changing behaviors or harmful habits. So if there are habits that we have that are not helpful and don't allow us to really kind of show up as in a way that makes us feel like ourselves, that makes us feel good about ourselves, then those things are self-compassion behavior changes. That's also fierce self-compassion. And then advocacy and standing up to injustices. And we do a lot of these things for other people. I mean, again, remember, we're doing a lot of these things for other people more often than we would think, than we notice. But really, it's about looking inward. How often are we really doing these things for ourselves? So I added the next couple of slides this morning because I ran across this really great analogy and I just couldn't not share. So here. If you're all familiar with the Golden Gate Bridge, it's in San Francisco, California. It was constructed in I learned a lot about the Golden Gate Bridge this morning. It was constructed in 1933, and construction was completed in 1937. It is 1.7 miles long and has a live load capacity of 4,000 pounds per lineal foot. I'm a social worker. I'm a licensed clinical social worker and a researcher. I'm not a engineer. So I don't know what a live load is, but to me, that means traveling, like moving load. Cars, yes, I'm getting a nod. Thank you. So that's the live load. And I don't know what a lineal foot is, but 4,000 pounds per foot of it feels like a lot. So she's working. The Golden Gate Bridge is working. She's supporting millions of people a year. She's incredible. And of course, she gets corrosion and rust and things like that. So to prevent the corrosion, she's painted. And she's painted continuously. Caretakers start on one end of the bridge. They continue to paint and paint and paint until they get to the other side. And then they just start again. The work is not done. It's continuous maintenance. She's tended to constantly, which I just think is a wonderful analogy, to just tend to ourselves constantly, right? So I ran across this photo. Oh, wait. Yeah, caretakers. I would love to take credit for that word, but I cannot. So actually on the Department of Transportation in California, on their website, the folks that take care of the Golden Gate Bridge call themselves caretakers. It's not a crew. They're not engineers. I mean, some of them probably are, but it's a group called caretakers. So I can't take credit for that. So here's one of the caretakers. So just looking at this photo, I just want to take a minute to really, we've been talking about kind of concepts and ideas, but what does that actually look like in practice? And what's a really simple way that we can identify these things? It's not just about theory, it's about how we can use these things in practice and how we can notice these things. So if we're looking at this picture, can anyone spot some things that look like they might be self-compassion? You're welcome to shout out, or you don't have to raise your hand, you can just shout. Sorry? Helmet. Perfect. And the helmet is what? Do you think that's tender self-compassion or more of like the fierce protection? I think so too. I think it's more of this protection, right? He's protecting his head. Anything else? Wedding ring. He's got community. He's got support. And there were... Say it again, sorry. Touch. That's the first thing I noticed. Look at his hands. One is gloved and one is not. And the one that's not is connected. It is just the most gentle touch. He's connected to his work. And the gloved hand it's pointing at something, and he could just be talking to the photographer or who knows. But it seems like he's noticing something. He's observing something or saying, hey, there's something right there. But his face isn't showing judgment to me. Sorry? He's satisfied. Could be, yeah. Yeah. What else? I also noticed he's got a walkie-talkie. So there's more common humanity. There's more connection there. He can ask for help when he needs to. He's got keys. He's got a boatload of keys. So he can use all kinds of tools at his disposal. He's got, sorry? A jacket and layers of clothing to help protect him from whatever the weather is going to do in San Francisco. Goggles. So it's both. It's this tender self-compassion. Look how just amazingly kind he looks. And satisfied. That's such a good word. And then also in combination with fierce self-compassion and really protecting himself. He's doing some hard work. That feels very high up. It's like 200 some odd feet, but apparently it can move and get higher and lower. I didn't pull those stats for you, but it's very high up. And so he's doing some hard, scary things. And he's able to protect himself and show up to the bridge and show up to himself in a way that is compassionate. Okay. So another exercise, we're not going to sing, I promise, or hold hands or meditate. But just think for a minute, if you're willing, to imagine that you are a caretaker on the Golden Gate Bridge. And you make steady progress each day. You show up, you do the work, you put the paint on, you have a rhythm, right? You have a schedule. You've got a rhythm. And you reach the other side of the bridge, and then it's instead of looking behind you and judging your work and saying, oh, I missed that spot over there, or, oh my gosh, a bird already pooped on that. Instead of doing that, you simply begin again. That's the job. And you do it in a way that allows you to show up to do it again. So I want you to think about your life and your rhythms right now. What in your life needs this sort of rhythm? And if the goal is instead care and not completing and perfection, if the goal is to care, then what would that change for you? What would that change about how you show up to other people, how you show up to yourself? What protective gear do you need? This is mine. This is one of mine. I always have a beverage. Before noon, it's coffee, and after noon, it's water. So, what protective gear do you need? And then can you offer yourself the same compassion that you offer others that are maintaining something so essential? So here are some practical components, practical pieces of self-compassion. These are things that you can do, you can try. This is by no means an exhaustive list. There are things every day and every moment offers a new opportunity to be self-compassionate. I would like to point out, I think this fierce self-compassion piece and this boundary, creating good boundaries for ourselves, I think that really doesn't get enough attention. And we're not well practiced at it because we are so helping and so kind, and that is how we survived for so long. So I do want to point out a quote that I think is just really helpful to keep boundaries in mind, and that is that boundaries are the distance at which I can love both you and myself simultaneously. So there's got to be some sort of balance there.
Just
She's mad. So sorry. That's fine. She needed a break. Okay, so other things, incorporate caregiver needs in care planning discussions. This is, if anyone's familiar with palliative care, not hospice, not end of life necessarily, but just palliative care, mitigating pain and suffering and finding ways to live your life within the limitations that you have, and then also being able to lead a fulfilling life. Palliative care teams are really great at assessing that, and part of their job is to make sure that caregivers are also included. So I would highly, and medical care teams in general, I would highly encourage you to, if you're a caregiver yourself, advocate and say, hey, how can we incorporate my needs into this care plan as well? And then if you're a healthcare provider, I would also encourage you to kind of plant that seed in the back of your mind, and the next time you're in a care plan meeting or a discussion, ask the caregiver, what needs would you like incorporated into this, and how can we best support you? And I would also encourage folks to make plans for respite. Don't just wait for respite to come. Figure out a way to have breaks. Map out your caregiver supports. Who can help you? What is their skill? What's their talent? What's their strength? Can they be there on a dime's notice, or is it more planful than that? So that you can just have a list prepared and ready to go. Making compassionate statements to yourself and to others. There are a couple of acronyms that are popular in other areas of mental health. One of them is HALT. Stands for hungry, angry, lonely, and tired. And that's just a way to really check in, again, with that mindfulness piece. How am I feeling right now, and what do I need to do? I would also enter thirsty in there, too. We don't drink enough. And then the other one is RAIN. So another mindfulness tool. You're recognizing how you're feeling. You're allowing that feeling to be there without judging it or trying to suppress it or push it away. You're curious about it, and then you're going to pop in that kindness piece of self-compassion, and you're going to nurture that. What is it that feeling or that thing needs? And many, many, many more. I also encourage people to start small. This isn't any sort of race, and it's not big, massive changes. We don't need that. We often just need to start with small changes that accumulate over time. So in terms of sustainability, and what this shift from the defense system to the care system might look like over time, I don't have data on that. So these are my guesses. And this is kind of just an idea, a conceptualization. So sometimes when we think about time, we think about it as linear, a line, right? Like a timeline. But really, I think that when we have significant events in our lives, I think we really grow around them. I think it's more circular. And as our lives continue, we kind of come back and revisit hard moments in our lives. So sometimes that's, in a kind of a more extreme example, that could be something like trauma. We kind of revisit a trauma. Something comes up every time we go camping. Something happened, or something like that. So you never want to ride a bike again. So the next time you get on a bike, you get nervous. Something like that. But really, I think that our experiences are integrated into who we are. We are new people today than we were yesterday. And we're going to be all new people again tomorrow because we have more information now. We have more experiences now. There's more network and more connections now. You've met many people. So we're going to be a little bit new. And there's also the phrase, or the saying, you never step in the same river twice. It might be the same technical geographic body of water, but really the things change as the water flows, right? It's not all the same water molecules, it's all new. And these things are kind of the spirit of self-compassion, being really gentle with ourselves and allowing these things to be present in the moment. So here's an example. We think that time is linear. We think that events happen one by one, and that sometimes something happens that reminds us of something that's happened in the past. And we kind of deal with it and then move on. But really, I think that time is, this is a crude drawing, and I'm sorry, I couldn't find something cool. So sorry. But yeah, so I think it's more circular. And then something has happened, an event happened, a diagnosis, a fall, a phone call with some bad news. Something has happened that has really disrupted our growth, or that's kind of stopped us in our tracks. And I think we, as resilient humans, figure out ways to grow around it. And then as time moves forward and we continue to grow out, we meet up with that thing again. And then we meet up with that thing again. And it kind of looks like a tree, right? It kind of looks like a tree trunk, like the inside of a tree trunk. And we create these little knots, right? So if we apply these self-compassion principles to a tree knot, when a tree is wounded, it actually softens and then figures out how to grow around the injury. So I think that that's kind of similar to what we do. We figure out a way to grow around it and take it with us and revisit it, but in a different way, because we've been there before. And everyone has scars. Let's talk about common humanity. Everybody has scars. Everyone has tough experiences and is imperfect. Everybody's going to have their own pattern. And then, of course, mindfulness, right? The present moment is not, we're not replaying the past. We're revisiting it. It's a new iteration. It's a new version. We have new skills and new information. Same body of water, different, same river, different body of water. I'm working on that analogy, sorry. So I'd like for you to consider, in terms of, we talked about two different kinds of self-compassion today, the tender kind and the fierce kind. So just consider, what are the things that you've already done today that demonstrate your tender self-compassion and your fierce self-compassion? What else can you do today? Today is not done. What else can you do today? And then, is there something that you can plan for tomorrow? And again, start small. It doesn't have to be big. It doesn't have to be, I'm being so self-compassionate, whatever. You don't have to start meditating right away. But maybe an extra glass of water. Maybe taking that bathroom break between sessions. For Pete's sake, please take care of yourselves. Conferences can be a lot. That's it. That's all I've got for you. Thank you so much for your time and your attention. I really appreciate it. And we've got some time left. So if anyone has questions or comments, I'm happy to be here for that. Thank you.
Sorry.
So
Here we go. No, that was me.
I was told that you were going to be the runner, right?
I am.
So if we can somehow plan it so that we've got a question over here, and then a question over here, and then a question over here.
You don't have to do it that way.
Oh, okay.
I need to get my 80,000 steps today. So anyway, anybody have a question for Christi? Anybody? I know. Okay. I know you guys. I know it's been a long day. It's okay. It's okay. But she gave you a lot of great information that I'm going to start to use that around my house. Okay. I really, guys, I really am going to work on that. And I got a question here in the center coming up. Hang on one second.
I just missed it, and I'm sorry. Would you define fierce compassion and tender compassion again?
Yeah. So fierce self-compassion is really that protective mama bear, right? So I'm going to protect my time and my space. I'm going to protect my energy and my resources. That's really about laying boundaries. And then tender self-compassion is that caring mama bear. She's going to be kind. She's going to be gentle. She's going to be mindful, and really take things. I almost think of it as a little bit more slow-paced, and like sit in the moment in mindfulness. And then the fierce self-compassion sometimes is more action-oriented, not always.
Any other questions before we go into our breakout sessions coming up? Anything, anybody? Okay, now she's here. You can walk up and talk to her. She'll answer your questions in person too.
I will. And my email's on here, so if something comes up later, please feel free to email me.
So, okay, guys. Big applause for Christi, please. Thank you very much. Thank you. Okay, everybody, we're coming to that part of the day. You're not quite done yet, okay? Almost done, not quite ready yet. We just wanted to just say, first of all, throughout the day, we have learned from experts, and we now have an opportunity to learn from one another. The patient-lived experiences represented in this room are every bit as valuable as the scientific expertise we have heard from the podium. Whether you are attending the women's sessions or the men's sessions or the young adult sessions, we encourage you to share openly, listen generously, and support one another. Research may build up future therapies, but community helps us navigate today. And thank you for being a part of that community. That's for you guys. Okay, now here come the notes. You're going to be in a breakout session from 5 to 6. That means everybody in this room, you're going to lift and shift across the hallway and go into your designated spots. Okay? First of all, and the young adults meeting today are going to be meeting up near the restaurant area up in the front of the building, okay? If you're part of that, you're going to be up there. And also, we have a parent room. We've been asked for some parents want to kind of get together, especially the ones of the young adults. They're going to be meeting in salon number 4, just right over there, okay? Now, you're there. You're going to all be busy till 6 o'clock, okay? Remember, we're going to take the 5K photo, okay, at 6:20. So if you got a 5K shirt that you guys have collected over the years, make sure you wear them, okay? And then we'll probably, Jim, where are we going to be doing this at? Right outside the door in the lobby out here? Yep. We want a big picture. 6:20, if you want to slide out of your breakout for a couple of seconds and be a part of the picture, please do that. We also want to let you know that there will be a cash bar in the lobby space out there from 5:30 until 7:30, and dinner is served at 6:30 in here. That's when the doors open at about 6:30, okay? The staff is going to come in when you leave, and we're going to redo this room and make it all nice and ready for dinner. And then we've got a huge program for dinner tonight, guys. We've got all those what are those little things called? Raffle tickets. That's it. I don't know why I can't figure that out. Raffle tickets. And then we got a silent auction and a real auction with your paddles. It's going to be exciting and a little nerve-wracking for me. So you guys just go have a good time, do your breakouts. Christine, yes, ma'am. It's going to be, we'll point you in the direction. It's right across the hall, okay? You got a 50-50 chance, and you'll know if you're in the wrong place because we're going to move you, okay? Hey, no crossovers, no men in the girls, no girls in the men's, okay? Just everybody get in and do your thing. All right, you're free. Everybody, go, 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 go. And thank you for a great day. We'll see you later.
Well, good afternoon again. We hope you have enjoyed this day with us on our virtual feed here at the SPF Annual Convention and Conference. We've had a lot of different information today. We've heard from some of our researchers about the work that they're doing, as we usually do. Our federal regulators who are here with us when they couldn't be with us in Washington to talk about how we deal with the federal regulatory rules and trying to move forward toward clinical trials and other federal FDA and NIH involvement. And then this afternoon, we've heard information, over the past years, we've had folks say, the research information is good, but I don't understand all that. And can we talk about some things about how we live with these difficult conditions? And so this afternoon, we've heard from presenters about living life and how we eat and how we exercise and sleep. And we're hearing more about that tomorrow. And then finally, how we care about ourselves and care about those around us. And sometimes those of us that have these diseases can be very rough on ourselves, sometimes rougher on ourselves than anyone else. So I hope you caught some things from that can make life a little better, a little more manageable. As Tim just said, we're going to have our banquet beginning tonight at 6:30, a live auction. We've got a chance for you to be involved in that virtually as never before, and that information is on the website with the program information about tonight. Some of you may have already made arrangements to do that. In fact, some of you early on used that to buy some raffle tickets for the drawing tonight. So we'll be doing a lot of those things tonight, and that will all be on this virtual feed as well. And then in the morning, we'll begin right back. Norma or I, or both of us, will be here with you virtually about 7:45 in the morning, because we get the agenda started at 8. And our first speaker in the morning is Dr. Hande Osdendler, who many of you know because of her work with upper motor neuron issues. So a good long day tomorrow with a lot more information. And again, we're glad you're with us. If you need us, you know how to get in touch with us. And with that, you take a break, and we'll be back about 6:30 for dinner and a great program. So take care, and we'll see you in a little while.